markers, a combination of markers, so subset of T cells that discriminate CAR T cells generated from cord blood, the yellow dot, from peripheral blood, the blue dot. In particular, I want to highlight that mostly here we have a more early stage of differentiation, central memory, or even if differentiation where we're expressing CCR7. And what we found an upregulation of BTLA, CD272, in the cord blood and not in the PBMC-derived CAR T cells. In PBMC, we had the more latest differentiation expressed in CD57 and LAG3. And also, we found differential subset of T cells in T cells activated with BITS or Transact. The Transact is yellow. And in this case, we found a confirmation of expression of BTLA. We also characterize the phenotypic, the functional activity of the CAR T cells. Here, just an example. We found we simultaneously detected by fluorospot interferon gamma, IL-4, perforin, also granzyme, but was consistent with perforin. And we could detect specific activation with some differences between the different vectors, like CD28 or 41BB. Against the target cells, like Ragin, NALMC, ABVB expressing CD19, with different level of activity depending on the expression of the CD19, because NALMC express a lower level compared to Ragin and ABVB. And we didn't detect any or very negligible level of alloreactivity against PBMC. Here is the expansion. We use the GREX system. Here it was, we started with the GREX 6M, allowing more flexibility in the expansion. And we can see an efficient expansion, especially starting from 2 million, we could reach 100,000 million of cells. So the efficiency of expansion was very good and comparable to PBMC. I'm not showing you the data. And going together with the decrease of glucose in the culture and the increase of lactate that are markers that can mimic the cell count number as well. We did the transcriptomic upon activation, engagement of the CAR with the CD19 positive target cells or CD19 positive negative cells. And indeed, we could find, and we use the same system, experimental system, to do all transcriptomic, single-cell RNA metabolomic profile. And as you can see, also the spatial distribution of cells are different compared PBMC, the cord blood, the PBMC, and the target cells. And indeed, the signatures were different, completely different from cord blood, from peripheral blood, and even BITS or Transact activation. So just briefly, I cannot go into the detail of all the data we have. However, we found 1,122 differential expressed genes between cord blood and PBMC, activated with the same method. In this case, it's Transact, but the same is with BITS. And we found, for example, in cord blood, in resting CAR T cells, down modulation of inflammatory cytokines, and even antigen, the pathway associated with antigen presentation and processing, even graft versus host disease pathway. Most of the molecules involved in this pathway were down modulated. Also some cytokine. Instead, we have upregulation of the signaling IL-2 and STAT5. However, if we profile the cells upon the engagement, the antigen-specific engagement of the CAR, we have upregulation of cytokine receptor pathway, chemokine, the type 2 interferon response, and other markers associated with activated and effector function. And just as a control, you can see if we use unrelated target cells, so CD19 negative, we have a down modulation of exactly the same pathway and same molecules. So showing that what we see was specifically determined by the specific antigen activation. And here is the PBMC. I just want to highlight that if we analyze the same system for PBMC-derived CAR T cells, we had a lot of upregulation of pro-inflammatory profile, including IL-3, IL-31, IL-10, IL-9, so a more pro-inflammatory profile of the CAR T cells. And there is just a briefly analysis of a metabolomic profile. I want just to highlight the comparison between two different vectors, either activating cells with beta and transduct, and we could identify for different upregulation of some pathway like the amino acid metabolism, protein biosynthesis, glutamate, asparagine metabolite. And also for transduct, we could identify the amino acid sugar, nucleotide sugar metabolizer, lysine degradation, arginine, proline metabolism, and amino-acyl-T RNA biosynthesis. I want just to highlight that the amino-acyl-T RNA biosynthesis is involved in the development of immune cells and act as a regulator of signaling. And in addition, has been described also as one of the more prominent signaling preventing the cytokine release storm. Here, just an example of data we have. We have a lot of extensive data also for single-cell RNA-seq, where what we can appreciate that we can find here, for example, we have all the T cells not stimulated, so resting CAR T cells, either with the CD28 for 1BB, and here we have and this corresponding here where we identify the target cells. So the antigen-specific reactivity is well identified here, and we have both vectors localized in this subset, and also either with the beads and transduct as well, with the slightly different behavior. And here we have the unrelated stimulated CAR T cells with the Raji, the K562 cells, not expressed in CD19. And we have specific transcriptomic signal associated with this subset and with this cluster, can allow us to identify exactly the subset of CAR T cells involved in the antigen-specific reactivity and the molecule. Finally, we also used the zebrafish model to assess the in vivo activity of the CAR T cells. Here is a briefly example where we inoculated in zebrafish the target cells labeled with the green fluorescence, the CAR Ts labeled with the red fluorescence. Only after 2 hours, we were already able to see some colocalization, but within 24 hours, all the green target cells disappeared, so CAR T cells were able to efficiently kill the target cells in vivo. So in conclusion, we validated the usage of umbilical cord blood as a source, efficient source to generate off-the-shelf CAR T cells. We identify specific subpopulation that discriminate CAR T from umbilical cord blood from PBMC, and also all the functional transcriptomic, metabolomic, and even methylation profile associated with these cells. And we can conclude that there is a superior pro-inflammatory profile and also graft-versus-host disease-associated signature in PBMC-derived CAR T cells compared to umbilical cord blood. So the usage of umbilical cord blood could prevent some side effect, such as the induction of cytokine release storm in vivo. We are currently completing this data, integrating all the big data sets that we have. Is quite challenging, but I hope soon I will be able to present the conclusive data of this integration. Finally, also this work was performed when I was in Qatar at Sidra Medicine. And I would like to thank my team and all the collaborators from the genomic, the obstetric department, metabolomic core, the Jackson Laboratory for helping for bioinformatic analysis, and the team of Chiara Bonini, Monica Casucci, and also Attilio Bondanza for providing us the lentiviral vector and collaborating to the study. And now currently completed and performing the analysis at the San Martino Hospital in Genova. Thank you for the attention.
We have time for a couple of questions. I will start with one. You showed that you have an increased amino-acyl-tRNA synthesis pathway, but indeed you have different metabolic pathways
Yes.
they are related to some form of immune regulation, tryptophan, arginine.
Yes.
So do you know whether these pathways are activated to cool down, and are responsible for the limited inflammatory signature of these cells? This could be extremely interesting from a biological point of view, not from the application point of view, but
Yes.
I wonder whether you have done more experiments to understand.
So we didn't do functional experiment based on this data, but indeed, yes, this is true. We have this profile, and the profile is even different between usage of the 2 vector, for example, and is more associated with the 4-1BB, for example, pathway. So we are now integrating this data with the gene expression and also the methylation profile to find more mechanistics association, and for sure we will need to validate from the functional point of view.
Hi, Christina, nice talk. So I have actually 2 short questions. So one is related to the reduced pro-inflammatory signature that you have seen in your CAR T cells. And so I was wondering if you actually test these cells for the alloreactivity, and if you have evidence that they are less autoreactivity, because it's one of the major issues in using allogenic CAR T cells. And the second one instead is a curiosity, because I noticed that there is the TCR signature that is also downregulated, which again can spoke on less alloreactivity. But if you have checked then the functionality of these cells and see if it's also reduced because of the TCR downregulation.
Yes, thank you for the question. So we have assessed in vitro against allo. I just show an example, but we tested several examples, and indeed we didn't find high reactivity against allo, for example, PBMC that we use. We use also CD34 positive cells. We still need to do the in vivo validation. So we are planning to use humanized mouse model to validate in vivo the safety. So in general, based on our set of data, we also do, I didn't have time to show the data, but we validated, for example, the release of cytokines, pro-inflammatory cytokines in the supernatant, and it was exactly confirming the signature data that we have, so it was more expressed by PBMC-derived CAR T cells. So our data are suggesting that indeed cord blood can be less pro-inflammatory, more safe in terms of graft-versus-host disease. However, we need to do the in vivo final proof. And in addition, I don't know, maybe we'll still need the required to be 100% safe to do the knockout, the gene editing of a TCR before to use for therapeutic application. Okay, yes, thank you.
So we move for the next speaker is I am here, no, I think Fabrizio, sorry, because Fabrizio Mattei, and no, because here he was, he will present data about the cytarabine and the I33S22 axis and how synergize to circumvent resistance to PD-1 blockade in melanoma. Thank you, Fabrizio.
Moderator, for your kind presentation. In this study, you show the role of the cytarabine and IL-32 axis in circumvent the resistance to PD-1 blockade in melanoma. IL-32 is an interesting cytokine having important roles in allergy and cancer. In particular, thanks to its binding with ST2, it can modulate the action of different antitumoral activity in some immune cell subsets. Cytidine is an analog of the cytabine and is often methylated by catalyzing by enzyme DNMT1. DAC can act as a DNMT inhibitor, and in this way, DAC is a DNMT inhibitor that blocks DNA methylation by DNMT degradation into the genome. So, the main open questions are, can IL-33 be considered a valid immunomodulator in combined therapeutic settings against melanoma, and what is the role of this combo in the tumor microenvironment? So, in our first in vivo experiment, we see that combo did not significantly decrease tumor growth in mice and did not significantly maintain survival. But it significantly increases infiltration of CD4, CD8 T cells and eosinophils, and also increases the expression of some TIP1 related factors, and most importantly, it increases the expression of the immune checkpoint molecule PD-1. And this expression was observed in the CD4 and CD8 T cell surface. So we did a second experiment where combo DAC-IL-13 was combined with an antibody anti-PD-1, and as expected, we see that we have a tumor regression by this combination, and also a good maintaining of mice survival. This is also confirmed by increasing the infiltration of CD4, CD8 T cells and eosinophils, and the increase of expression of several effector molecules of antitumor immunity. What is the role by which this combo DAC and IL-33 interact? So we did an experiment with ST2 knockout mice that did not respond to IL-33 signaling. So we can see that DAC is the only cytokine that requires the presence of IL-32 ST2 axis to work. And this is also confirmed, but a decrease of infiltration of CD4 and CD8 T cells and eosinophils, and also by the lacking of expression of some T cell attracting and eosinophil attracting chemokines. So we also noted that in wild-type mice, DAC noticeable increases the expression of IL-33 and ST2. And we also observed that there is an in vitro dose response of DAC that increases IL-32 expression in murine and human melanoma cells. So we did a DNA B2 conversion and methylation-specific PCR by using against two promoters in IL-33 murine and human gene, and we see that that promoters appears to be demethylated. So, DAC is able to upregulate IL-32 expression to get the methylation of its upstream gene promoters in human and mice. So we used microfluidic system to study in what two tumor environments were put in competition. In the first, melanoma cells, mouse melanoma cells, were in presence of single drug or no treatment, and the second were in presence of combo. As expected, regarding immune cells, we used the wild-type spinocytes or STD spinocytes. So we see that the combo induces migration and infiltration of splenocytes, but this did not occur when the splenocytes are STD knockout. We repeated the same experiment by using a human system where PBMC and melanoma cells were improved. And we see that the combo induced the emigration and infiltration of PBMC. So we were also able to identify by confocal microscopy the presence near tumor mass of CD8 T cells, and this testifies an anti-tumor action. So we also generated bioorganics a melanoma spheroid that is vascularized by UVAC and is also induced by circulating PBMC. And we can see that the combo is the only combination that induces the high infiltration, the highest infiltration compared to controls. So, our conclusive remarks are that IL-32 increases in the tumor environment immune cell infiltration, upregulates anti-tumor immunity factors, modulates PD-1 and T cells, thus improving response to PD-1 blockade. But also DAC mechanisms are important in this context. Indeed, DAC requires IL-32 signaling for its therapeutic efficacy. DAC demethylates human and mouse IL-32 promoters melanoma cells and induces IL-33 expression and ST2 cooperation for immunocell recruitment. So our take-home message is that IL-33 is a promising immune activation for combo therapy with PD-1 blockade and DAC and possibly other GNMTI. Organ-on-chip platforms, interestingly, can help to assess immune infiltration and identify immune cell subsets in a context of preclinical studies to optimize the cancer therapy. So I wish to thank in particular Francesco Noto that realized all this work and also collaborated with Dr. Andrea Pavesi at Singapore to generate the vascularized tumor microenvironment. I also wish to thank Adele Nino and Luca Businaro for realizing microfluidic chip, and a special thank to Rom Technopol, which realized a laboratory specific for studying microfluidic systems. And finally, thank you very much for your attention.
Thank you. Really interesting data. I'm wondering about, did you profile the vessels after the combination treatment? Because it looked like in your organoids that you would actually get also wider vessels with the IL-33 therapy. And IL-33 was named the nuclear factor of high endothelial venules, so I get very excited, and I'm wondering, do you think that you get a high endothelial venule-like phenotype of the vessels?
So okay, we repeat the question.
using only UVEX. But we are setting not only the system with other cells and also other tumor cells, but also by using other types of vascularizing cells to see if this can increase the efficacy of PBMC circulation and infiltration into tumor. Okay. Thank you.
We move to the next speaker, Francesca Reggiani. Is going to talk about NK cell. So we're changing the type of cells to how to activate to improve the therapy of triple-negative breast cancer.
Grazie. Okay. First of all, I would like to thank the organizer for selecting our work. It's a pleasure to be here. So I will talk about triple-negative breast cancer that I'm sure that all of you know, also from yesterday's session, that it's a very aggressive subtype of breast cancer. And it is characterized by the lack of expression of progesterone receptors, estrogen receptor, and HER2 overexpression. So for this reason, it's not possible hormonal therapy as well as target therapy. And for a long time, the only therapeutic opportunity for this patient was chemotherapy administered as neoadjuvant therapy. However, the rate of response was very low, and only 30 to 40% of patients displayed a so-called PCR, pathological complete response, so basically the absence of the tumor at surgery. That until now is the only good prognostic marker of a good outcome in these patients. And patients who had chemotherapy failure did not have any other therapeutic options. This changed completely when the Keynote 522 study demonstrated how the combination of pembrolizumab and immunotherapy, of course, with chemotherapy in the neoadjuvant treatment of these women basically duplicate the rate of response. So this opened a very important new opportunity for the therapeutical strategy in these patients, and also underline how the proper stimulation of immune system is fundamental in this setting. So our question was, which cells of immune system are mainly involved in the chemotherapy response in these patients? So we apply a digital transcriptomic approach in the biopsies of patients that were undergoing to chemotherapy in our institute in Reggio Emilia. And we basically found out that only one population of immune cells, natural killer cells, enrich the biopsies of the patient that display a complete response to chemotherapy compared to patients that progress during therapy. So to understand better the role of natural killer cells in this context, we started the so-called STARLET study in collaboration with the medical oncology of our institute. And we prospectively enroll all the new diagnoses of triple-negative breast cancer in our institute, and we collect 2 blood samples, one before the starting of the neoadjuvant chemotherapy that in the meanwhile changed to the Keynote schedule, so with the pembrolizumab. And the second time point, we got the blood sample before surgery, so after the completion of the chemotherapy. And so we analyzed the profile of circulating NK cells, and as you can see by this FACS analysis, basically the number of circulating NK cells did not change when we look at the first time point, so before therapy, and in the 2 groups of patients that display a completely different response to the neoadjuvant therapy. However, when we look at the second time point at therapy completion, we found a dramatic decrease of circulating NK cells in the group of partial responders with residual disease, basically. But this has not happened in the group of complete responder. And indeed, we found a completely depletion in the partial responder of the NK with the immunophenotype characterized by the expression of CD64 CD16. So basically, it is the more cytotoxic and the subpopulation of NK cells with improved anti-tumor ability. So to confirm that, we perform also single-cell RNA sequencing analysis on this blood sample, and basically we confirm what we observe with FACS analysis. So the profile of deregulated genes in NK cells in the post-therapy was completely different when we compare patients with different response to the immunotherapy and chemotherapy. And indeed, the expression of the main cytotoxic marker in NK cells that you can see in the right panels is completely impaired in NK cells after therapy only in the partial responder groups, but remain quite higher in NK cells from the responder group, suggesting that NK activation is a crucial event necessary to achieve a complete response to the therapy. And we confirmed that with an analysis of trajectories to see the evolution, the functional evolution of NK cells towards a more cytotoxic phenotype. And as you can see in the second graph on the right, the trajectories of the two groups of patients with different response to the pembrolizumab and chemotherapy were quite different, suggesting indeed a less cytotoxic evolution of NK cells in the partial group of partial responder groups. So if NK activation is so important to achieve a complete response to neoadjuvant chemotherapy and immunotherapy in our tumor, how we can reactivate them, how we can improve the response of patients that are still not responding to our treatment. So to answer to this question, what we did was to construct a gene signature of 300 genes that was specifically associated with NK activation in our patients. And this signature was used to apply a bioinformatic approach using an algorithm that is called FIMO, that basically scans the promoter of the genes of our signature to predict potential upstream transcriptional regulators. So FIMO predicts a list of more than 200 transcription factors. That was, of course, too long, so what we did was to prioritize this list, and here you can see the top scoring list of transcription factor as candidate regulator of NK activation toward triple-negative breast cancer. So you can see there are some factors that are already known to play a role in NK activation, like SMA3, that last year we published a paper on Nature.com in which we demonstrate the activity of SMA3 in NK cells in another settings. But also PAX5 and X2 were already reported to be a role in NK activation in other contexts. But today, I will show you some very novel data about a new transcription factor that basically role in NK cells was completely unknown. This factor is VEGF1. VEGF1 is a zinc finger transcription factor that is mainly known for its role in endothelial biology. But actually, nothing is known about that on the biology of NK cells. So going back to our data, our single-cell RNA sequencing analysis in our patients, it was clear that the expression of VEGF1 was downregulated in NK cells after therapy completion in partial responder. So this led us to hypothesize that VEGF1 was correlated with a higher NK association. And to prove that, we performed chip sequencing analysis to assess the direct targets of VEGF1 in NK cells. And we found a lot of genes encoding from proteins involved mainly in vesicle trafficking and exocytosis. So this is quite important for NK biology because the release of cytotoxic granules is possibly the main effector mechanism, anti-tumor mechanism activated by NK cells toward tumor cells. So to prove the association of VEGF1 with NK activation, we obtain the knockdown of VEGF1 by a CRISP interference approach in NK cells. And indeed, the knockdown of VEGF1 was sufficient to drastically reduce the anti-tumor abilities and the killing abilities of NK cells in co-cultures with triple-negative breast cancer cell line. And this was associated with a reduction in the degranulation activity of NK cells. Indeed, the expression of the marker CD107A, that is a marker of degranulation, was found decrease in the cells with carrying the knockdown of VITSF1. So this data basically confirm us that VITSF1 can control the cytotoxic ability of NK cells by regulating their degranulation. But we want to confirm that, and also the association of VITSF1 in its transcriptional program with NK activation and with, of course, immunotherapy response in our patients. So we validate our findings in an independent cohort of single-cell RNA sequencing analysis, also obtained in triple-negative breast cancer patients, but treated with a different neoadjuvant therapy with a different immune checkpoint. So what we found was that the differential expression of the transcriptional program controlled by VITSF1 in NK cells was sufficient to predict a divergent response to immunotherapy also in this independent cohort patient, confirming that maybe it's important in defining the outcome of the immunotherapy. So to summarize what I told you today, I hope to have convinced you that NK activation is absolutely necessary and crucial event in obtaining a complete response to the neoadjuvant treatments, so chemotherapy and immunotherapy in triple-negative breast cancer patients. And NK activation is associated with specific transcriptional changes that can be analyzed, as we did with a bioinformatic approach in which we identify a potential novel transcription factor, master regulator of NK activation toward TMBC. And we demonstrated that VITSF1 seems to control NK degranulation and cytotoxic ability of these cells, and the transcriptional program controlled by VITSF1 is associated with higher activation of NK cells and a better response to immunotherapy in triple-negative breast cancer patients. And the take-home message of this presentation is that targeting the transcriptional program of a factor can be a novel strategy to improve NK effector function and maximize immunotherapy response in triple-negative breast cancer patients, but also can be applied, for example, to improve the effector properties of adoptive NK cell therapies that we know that are now basically a reality and an alternative to adoptive T-cells therapy. Finally, I would like to thank all the members of the Translational Research Laboratory at the Azienda Ausiliare CCS of Reggio Emilia, in particular Veronica and Elisa, the bioinformaticians that did the analysis that I show you today, and also Gloria for the helping with the patient samples, and Martina and Narita that work with me on this project. Also, I would like to thank our medical oncology unit, breast surgery and pathology unit, our collaborators and our supporters, in particular IARC, that funded this project with my first IARC grant. Thank you very much for your attention.
Thank you, Francesca. So this new role in neoadjuvant setting of NK is so important. I have a question. All your fantastic work has been done in periphery.
Yes.
I have understood.
Yes, we have also the transcriptomic profiling, the digital transcriptomic profiling on the biopsies.
Because it's really important. Because do you know when you have the tumor regression in the tumor bed regression, if NK cells infiltrate the tissue, and how is the situation at baseline of NK cell infiltration in the different responder or not responder patients, because this is really...
Absolutely. Thank you for the question. Actually, it was our question too. So what we did was to start with the biopsies, so to see if there is some immune cells that was associated with a better response, of course, also as a predictive markers of response. And we found NK cells that enrich the biopsies of patients that respond better to chemotherapy. Actually, we expected to find a lot of other immune cells, but for example, T cells are not changing between patients with different response. And so NK cell was the most prominent candidate from this analysis. And then we decided to look at the periphery also because it's more easy, of course, to monitor the landscape, the immune landscape of NK cells and of immune cells during also the therapy. But finally, we want to check at what was left of the tumor after the therapy, because of course for the partial responder it's obvious to find some tumor left at the surgery, but actually when we have the surgery of the complete responder, it was very difficult to see where the tumor was, and actually we were not able to find specific. We just had a very big inflammation, but we have no observation that a specific enrichment of NK cells. So that's why we just focus on the circulation. But of course, this is a very important question.
Quick question and quick answer.
Yeah, I'll try to be quick here. Very nice presentation. So the first one, very quick. Did you stratify for other, triple-negative breast cancer can be subclassified, and particularly the immune type of triple-negative breast cancer? Do you see any a correlation overlap between your signature and other reported signature? And the other one, how do you know that your factor ZNF1 is not expressed in the NK cells which are enriched in your sample and is in the tumor cell? I don't know if you microdissect or anything.
For the first question, the analysis is ongoing, so I don't have the answer yet because we are basically analyzing all the patients, and so I hope to answer it maybe next year. But I'm sure it's a very, very good point to analyze. And for the second one, if I got it correctly, you asked me if I'm sure that VEGF1 is only expressed in natural killer cells and not in the tumor cells, right? Okay, so actually VEGF1 expression is present also in the tumor type. So we wanted to focus on the biology of NK cells in order to see if we are able basically to reprogram them and basically improve their activity toward tumor cells. So we were not interested in what happened in tumor cell, but of course VEGF1 is very important in some tumor cells, also in TNBC, because it seems that it's also interact with oncogenes like RANKS2 and other oncogenes. So it can have also a role in tumor too.
Okay. We need to move on with the last.
Thank you.
Thank you. So
we go back to endothelial reprogramming. So we have Carlotta Tacconi will present tumor sites-dependent endothelial reprogramming limits CD8 T-cell immunosurveillance of liver cancer.
So good morning, everybody, and welcome to the last session, to the last talk of the day, where I will try to convince you how endothelial cells are important to modulate effector CD8 T-cell function in liver cancer. So first of all, I would like to introduce you to what happens usually in the healthy liver, where we have effector T cells that can exert their effector function just by staying inside blood vessels. And this happens because in the liver vasculature, we have the presence of this fenestration of this pore through which CD8 T cells can extend protrusion, allowing them to get in contact directly with the hepatocytes just by staying inside the vessels. And thereby, the effector T cells can kill hepatocytes from there. However, what we don't know is what happens in tumor growth, where the anatomical, hemodynamical, environmental feature acquired during tumor growth can shape effector CD8 T cell behavior and function. So for this, we study hepatocellular carcinoma, which is the most frequent primary liver cancer, and it arises from several different etiological factors, such as chronic liver infection given by HBV and HCV, alcohol consumption, and nowadays also metabolic diseases are increasing the incidence of hepatocellular carcinoma. We decided to take advantage of the AST mouse model. This mouse model harbors under the albumin promoter efflux stop cassette that, when excised, can give rise to the expression of the oncogene SV40 tag. We crossed these mice also with a CAG TD tomato or Zetes green mice so that we can track what happens in the tumor lesion non-invasively. We also engineered a lentiviral vector that now we call hepatocre that brings under the transthyretin promoter, which is also in this case an hepatocyte promoter, the expression of the CRE, and we inhibit the expression of the CRE in endothelial cells and hematopoietic cells thanks to the presence of these two foraminal repeats. So when we inject our lentiviral vector intravenously, what happens is that we excise the flock stop cassette, having now the expression of SV40 tag and TD tomato roseate green in our hepatocytes. And this leads to the formation of different and multiple hepatocellular carcinoma lesions that can be tracked as soon as 5 weeks post-injection thanks to the MRI, and we can see that the tumor volume and the number of lesions increase progressively over time, and finally our lesion would be either green or TD tomato positive, so that then we can track CD8 T-cell behavior in a non-invasive and longitudinal way. We then wanted now to have effector T cells that are specific for our model, and for this we decided to take advantage of the TCR1 mice that have a TCR1 that acted against the SV40 tag. We differentiated them in vitro we're using the tag peptide with interleukin-2, and then we inject them in the mice to see how they exert their effector function. In the beginning, we decided to use a very early time point, 8 weeks post-injection, when the total tumor volume was below 100 millimeter cubic, and the total number of lesions was below 25. And in this setting, we can see that the tag-specific effector T cells were able to clear the tumors, while the control effector T cells that were directed against an unspecific antigen were not. However, when we instead are in a more clinical relevant condition at 14 weeks post-injection, when the total tumor volume exceeds 100 millimeter cubic and the total number of lesions is above 25, we see that most of the mice do not respond anymore to our effector T-cell therapy. So we then wonder what was inducing the different response in these two conditions. So we went back at an intermediate time point at 12 weeks, and we found that we have both lesions that they do respond to the treatment, that are here highlighted in blue, while we have also lesions that do not respond, that are those orange ones that will progressively grow over time. And we classify them in responder and non-responder, and one of the major characteristic that they had was that lesions that were 90% of the time responding were below 10 millimeter cubic of volume, while the one that would not respond anymore were above 100 millimeter cubic. So from now on, I will talk mostly about responder and non-responder lesion. And the first thing that we thought is that maybe recruitment of effector T cells into different lesions could be different. And for this, we quantified by confocal microscopy the abundance of effector T cells that were present in the two different lesion types. And although we see that there is a significant reduction of effector T cell recruitment, we do not think that this can impact very much the tumor clearance. What we saw instead was that the activation of those specific effector T cells was different, and as here is shown in terms of a percentage of interferon gamma effector T cell specific. And we saw that activation of effector T cells was highly reduced in the non-responder lesion compared to the responder. One of the reasons why maybe these effector T cells were not able to exert effector T cell function anymore could be due to antigen presentation by the hepatocytes. So for this, we isolated hepatocytes from healthy liver, responder and non-responder lesion, and put them in culture with terminally differentiated effector T cells. And as you can see, this was not the case, because hepatocytes derived from a non-responder lesion could very well induce interferon gamma expression. So we thought about our vessels, as I showed you in the beginning, were very important for effector T cell function, and we performed a scanning electron microscopy experiment where we can see a very nice fenestration in a healthy normal condition However, this fenestre starts to decrease in responder lesion, and they're basically completely lost in a non-responder condition. So we saw that there is an important difference in the endothelium. We therefore wanted to analyze a bit further what happens into the vasculature of responder and non-responder lesion. And for this, we took advantage of single-cell RNA sequencing, where we analyzed endothelial cells from healthy liver, responder, and non-responder lesion. And as you can see already here, non-responder-derived endothelial cells mainly cluster together away from normal and responder lesion, and they start to express a series of markers that are typical of this lesion and that are not commonly found in a healthy condition. And for simplicity, I just took two different markers, like one as a marker of liver sinusoidal endothelial cells, and CD34 as a marker of this aberrant endothelium. And we saw that LIVE1, basically, it's completely gone for the non-responder endothelial cells, while they start acquiring CD34, indicating a capillarization phenotype. And this was also validated at protein level through flow cytometry. So having these two markers as a proxy for healthy liver fenestrated endothelial cells or non-fenestrated endothelium, we performed a confocal microscopy analysis to see where effector T cells that are activated sit. And what we found is that the vast majority of interferon gamma-producing effector T cells, they lie very close to LIVE1, probably fenestrated normal vessel, while they sit far away from CD34 capillarized endothelium, indicating that indeed the presence of one endothelial phenotype or the other could influence effector T cell function. And I don't have time to show you this, but we also validated the same in human spatial transcriptomic data. So, but finally, how can we restore effector T cell function? We thought about using a soluble guanylase cyclase activator that it has been used previously in liver fibrosis, for example, and to give it to the mice. So we give by oral gavage this small molecule compound, base 602770, that it's able to activate the soluble guanylate cyclase. And we see that even if we have very big lesion, already at 7 days post-treatment, we can partially restore fenestration. And importantly for this talk, the fenestration was able to restore the percentages of effector T cells that were activated in our non-responder lesion that could gain the same percentage of activation as the one that we see in the responder. So with this, I would like to conclude by summarizing the finding that I showed you so far. So basically, in healthy liver, in the responder lesion, we find liver sinusoidal endothelial cells that present a fenestration that allows the hepatocyte to be directly in contact with the blood. However, in the non-responder condition, we have a phenomenon that it's called capillarization of the endothelium. Well, we have the loss of the fenestration, and this induced the reduced density of activated effector T cells nearby non-responder vessel, leading to the loss of effector T cell function and the loss of the clearance of the tumor. So we're now thinking about reprogramming the endothelium as a new strategy and way to promote and support tumor immune surveillance. And with this, I would like to conclude with the most important slide that I showed you today, so the acknowledgement. I would like, first of all, to thank my mentor, Matteo Iannacone, that allowed me to work on this very interesting project. Our two fantastic bioinformaticians, Pietro Delfino and Chiara Laura, that performed all the bioinformatic analysis, and my former colleagues, Bianca and Federica, that worked a lot together with me on the project, and you for your attention.
We are perfect on time, so any question from the audience?
Really amazing data, very nice.
Thank you.
I have a question about these endothelial cells, because you were also doing the transcriptional profiling,
Yes.
and what you see are the markers actually of tumor angiogenesis that are usually upregulated
Yeah.
in these. like apelin and this. But I didn't see, do you also see a difference in expression of adhesion molecules and chemokines? Because that's usually downregulated.
We actually do. We see them downregulated. For example, we see that VCAM1 and ICAM1 gets downregulated, and you would not really expect that in an inflammatory environment. And we see that those vessels acquire completely different features. For example, they upregulate CD200, for instance. That is also very interesting to follow. LY6E, I know it's only in mouse, but still, it's something atypical. And they also start to express CD36 and FAP4. So they really reshape completely the transcriptional profile. Also, at the transcription factor level, they change completely. So, for example, they lose the expression of GATA4 or CMAF that now we want to try to reintroduce in the cell, but it's a bit difficult. Maybe I will talk, if I may, a bit about this later with you, because we would like to move in this direction.
It looks extremely interesting. So do you think, so is it also the recruitment of T cells into the tissue that will be different due to the adhesion molecule expression? Or do you think it's mainly the contact between
We think it's mainly the protrusion, because we also did intravital microscopy studies where we try to see how the T cells adhere and crawl along the vessel, and we didn't really see differences between a responder and non-responder vasculature. So we think they support equally the recruitment and crawling, but what's missing is the fenestration that then does not allow this effect.
Thank you.
Thank you.
It's time to wrap up. I'll be fast because you need a coffee break before the next session. If you like this session, if you like the type of interplay between scientists, the translational nature, please join us at the next NIBIT meeting. There is still time to register. It's going to be in Livorno this year. October 23, 25. Unfortunately, we don't have the flyer, but we will put a flyer. But this is the type of atmosphere we're trying to reproduce. It's the mission of our society, and I'm glad that ACC gave us the opportunity to share with you this experience. Thank you very much for attending this session.
Please, if you could have a seat, because we are running late and we have a quite packed agenda today, so we need to be really on time. So now we are going to start this session on cancer, genomics and beyond. I'm Nicola Normanno, my co-chair Giuseppe Pellicci. Unfortunately, could not make to be here today, but we designed together with him, with Ruggero and Stefania, this session with the idea to provide an example of good clinical practice on how comprehensive genomic profiling can be implemented also in Italia Reference Center, but also we wanted to discuss all the challenges, the limitations of the current approach. And for this reason, we selected two outstanding speakers. We have Giancarlo Pruneri from the National Cancer Institute Milan and Rodrigo Distman from the Vall d'Hebron Institute of Oncology in Barcelona. So it's my great pleasure now to introduce Giancarlo Pruneri, who will talk about the implementation of comprehensive genomic profiling in a reference cancer center. Giancarlo.
Me here. So what I'm going to do today is discuss with you how to build an infrastructure in order to get comprehensive genomic profiling. That would be absolutely useful, of course, to get clinical research in our environment. But I will touch base also on NGS more in general, because, of course, we have to get clinical and translational research in our daily life, but we cannot forget clinical practice. And I think that NGS is a fantastic way in order to gather information which are very useful for clinical research, translational research, and clinical practice as well. These are my potential conflict of interest. And first of all, let me outline my vision and what I think we have to build in order to implement a pathology lab which could be able to get comprehensive genomic profiling. Pathology is in brackets because, as you know, in Italy at least, the national health system is quite heterogeneous, and it is based on a regional basis. And at least in Lombardy, what is happening is that there is a decree that establishes, clearly that at least with regard to the somatic analysis of cancer, this must be carried out in a pathology lab. But of course, in other regions, we know very well that this could be carried out by other professionals, and it's absolutely fine. What I'm willing to underline is that this kind of analysis, for the reason will be highlighted later on, it is, in my opinion, very important to be very integrated with a pathology lab. So it's an interplay. There is the lab, and then there are all these infrastructure, which are absolutely essential, in my opinion, in order to get comprehensive genomic profiling and translational research. These two activities, of course, are very closely interconnected. And these are, of course, the stakeholders, which are very important and play a pivotal role in the implementation of a genomic and other omics lab in daily practice. So with regard to pathology, what is very important to underline is that NGS, comprehensive genomic profiling, is just the last. It is next, but probably is old generation sequencing. We are facing new technologies nowadays, but these are the other techniques which are very useful in order to identify predictive and prognostic factors. So when we are talking about biomarkers in the life of a patient, what is important is that we will choose the best method in order to get the gold standard analysis with regard of a biomarker. For example, in immunohistochemistry, we know that we have many troubles in harmonizing the results of immunohistochemistry, which are more or less the same troubles that we could have for harmonizing NGS results, by the way, but immunohistochemistry is very useful, for example, for ADCs. We know that antibody-drug conjugates nowadays are new drugs. There is a lot of implementation of this type of drugs in oncology nowadays, and the best method in order to evaluate this biomarker is not NGS, is immunohistochemistry. So NGS is just one of the possible techniques that we have to use, and these techniques should be available I wouldn't say in a pathology lab, but at least in an institution that is going to get precision oncology. There was a debate a couple of years ago because we were not sure, the scientific community was not sure that NGS was the best method, because we have single gene testing. We have seen that we have single gene testing technology, RT-PCR, in cytobridization, and so on and so forth. But, luckily enough, there are the guidelines which are stating very clearly that at least for a couple of diseases, what we have to do is run upfront next generation sequencing. So we are, I would say, compelled to develop this kind of analysis within our institution. And, the type of tumors for which NGS is recommended by the guidelines, they are continuously increasing. There was a debate related to the cost. This was a paper we published a couple of years ago demonstrating that NGS is cost effective. I will get back on the cost. Let me just underline the fact that we know that there is an issue related to the cost. Especially oncologists, they are usually underlining that NGS is costly. This is simply not true. We have just demonstrated, for example, with a recent paper that within the diagnostic journey of a patient, so visits, CT scan, RNA, and so on and so forth, NGS is representing 1/3 of the cost, not including drugs. So I think that the cost is not an issue. Of course, the organization of our labs and of our network is an issue, because we have to save money or using money in the appropriate way. What is the situation nowadays in Europe, in the world? Nicola published a paper a couple of years ago demonstrating that we are, still behind the task. So more or less, the use of NGS for patients, for example, with non-small cell lung cancer, is ranging from 50% to 70% of the patient. This means that for at least roughly a half of the patients, we are not able to detect the biomarkers, which are very useful for treating patients with on-label drugs. So this is something that we have to improve absolutely in our environment before, I will say, upgrading to comprehensive genomic profiling. It's like with a tennis player trying to get to Wimbledon without training in the national courts. Of course, we have to do, first of all, our duty in order to detect ACC1 biomarkers in our daily life. Let me just present this data. These data are from Lombardy region, but of course there will be for sure similar data from other regions. This is again non-small cell lung cancer. These are the biomarkers which are useful, mandatory for deciding to treat patient in second line, but very quickly in the next months in first line as well, for patients with non-small cell lung cancer. What we did is building a network which was not hierarchical, and at the end of the day, these were the results. So we were able to get in more than 2,000 patients these results related to the profiling useful for getting SCASK1 biomarkers. the results, I mean, we achieved the goal to profiling 74% of the patient. That is a quite high matrix. It is the highest matrix never ever published thus far. And this is so important because, I mean, NGS is a potential, a method that is potentially helping us in getting S-cascade variants more than one, so the S-cascade variants which are useful for treating patients within clinical trials. But first of all, what we have to do is getting the curative biomarkers. Why would we need extensive profiling at the end of the day? There are many reasons for trying to achieve this goal. Of course, large panels, I mean, they are including S-cascade1 biomarkers as well, so you can achieve this goal using next generation sequencing. And then there are other issues. We can detect mutational profiles associated with therapy resistance upon the treatment of patients with cancer, potentially germline variants, which are always included within large panels. Of course, we can get agnostic biomarkers and complex biomarkers, including mutational tumor burden, HRD, and copy number variation. Then I think that it is very important. I mean, Italy is a great country, so what we have to do is launching initiatives in order to implement our way and our knowledge of the genomic portrait of patients with cancer. There are many of them in Europe, and there is some crazy doctor that already launched this approach within the clinical practice. This is a lab that is currently ongoing in the Netherlands, and they decided to get whole genome sequencing for all the patients. Which kind of techniques should we use in order to implement our method? It is targeted NGS, whole exome sequencing, RNA sequencing, whole genome sequencing all together. At the end of the day, what most of the labs choose is targeted NGS with large panels. This is because we are compelled to get a final diagnosis, at least in my region, in 10 working days, and it would be not conceivable to get whole exome sequencing or whole genome sequencing within the clinical practice. We are in my lab trying to do that, but it is, there are many hurdles that has to be overcome. So we decided in 2022 to launch a prospective clinical trial that was very simple, running all the patients with cancer for which an NGS analysis was requested by comprehensive genomic profiling. So we switched from small panels to larger panels in order to capture all the metrics that I just shown to you. And of course, there was other secondary endpoints, the integration with different omics, and very importantly, the molecular tumor board. So basically, we start this experience because we had, we were living in an institution which was running clinical phase 1 clinical trials. So we need to discover potentially actionable target beyond the S-cascade1, so the biomarkers which are useful for the treatment with on-label drugs. So this is the structural organization. You see that you have a genomic analysis here, other omics, and then in situ hybridization and fluorescent in situ hybridization lab. There is immunohistochemistry as well, but we didn't report it in this slide because immunohistochemistry is nowadays a very routinary method. These are the guys working within this approach, this structure, and then you see, and I'm very happy that you see that pathologists, biotechnologists, technicians, they are all together. There are no the pathologists running everything. Every guy here has a responsibility in running tests. At the end of the day, this is the output. These are the output in terabyte. That is very important. I will get back on that because this is a very important issue. And then these are the stakeholders again, the electronic health record, the molecular tumor board, clinical structures, and so on and so forth. Bioinformaticians, we have 7 on them. They are repeated here. We are not all of them. This is a very important issue. I think that it is, barely impossible to develop comprehensive genomic profiling in a lab without bioinformaticians. So they are taking care of all these activities, and they are basically running vital aspects of genomic labs, and without them it wouldn't be possible to implement comprehensive genomic profiling. Don't mention other omics within a lab. And we know that in Italy this is harder because they are all of them. All of these bioinformaticians, they have contracts. They are not stabilized because the national health system, they are not recognizing them. In our region, we try to recognize them, as biologists, and we are trying to do our best in order to try to stabilize them. Why they are so important? Again, data. They are very important because, running comprehensive genomic profiling, you are generating very, big data, and big data, they must be, and all the process must be effective and safe. In order to do that, this is what they design within my lab in order to achieve these goals, which is, again, a very swift process of analysis and a safe process. And this is very important, especially nowadays, because data, they are so important for of course, for clinical practice and for clinical research as well. This is the workflow. You see that we prefer to, of course, we are going, we are trying to do all the tests in-house, and we are using 2 different platforms and a different approach. But at the end of the day, you see that with a relatively limited structure of platforms and reagents, we are able to achieve these results. These are very brand new data. I asked to my bioinformaticians to retrieve them from the files. These are you see the results here. We run comprehensive genomic profiling in 60% of the cases, but again, small panels are there in 40% of the patients. With small panels, I mean panels less than 50 genes, including less than 50 genes, with large panels, of course, more than 50 genes, but they are mainly panels ranging from 300 to 500 genes. The first result, it is quite intuitive. It is that by using comprehensive genomic profiling, you are able to detect a higher number of variants which can be useful for treating patients within clinical trials. This was, the prerequisite for this kind of approach, and it was matched for these settings, cholangiocarcinoma, pancreatic, gastric cancer. I would say that for non-small cell lung cancer, you can obtain, as I shown, very, effective results by using small panels, and this is because, the small panels were originally designed in order to detect variants within non-small cell lung cancer. This is the trend of the use with regard to the use of the different panels. Let me just underline that this is liquid biopsy with comprehensive based on comprehensive genomic profiling. You see that there is a slightly increase in the last years, but let me say that this is a very critical aspect of the especially with regard to the routine analysis, because it is costly, and it is related to the quite high prevalence of failure. because of pre-analytical flows, or because, it is quite, we are still in our training curve. This is liquid biopsy with small panels, you see here. This is increasingly, in a steeper way, and this is because this is trained by again, by the clinical practice, because that we know that especially with breast cancer nowadays, we are asked to detect ESR1 and AKT and so on and so forth in the liquid biopsy. You see that this is our use of comprehensive genomic profiling. It was very high a 2 years ago. Nowadays is slightly decreasing, and this is related to the cost and to the funding. So funds are a limiting factor for using comprehensive genomic profiling. Using small panels is much more cost effective. So, there is a balance there. And let me finally underline the fact that you see these are the small panels, which are more or less 100% in 2020. And you see that there is a slight increase in the last year for 2 reasons. First, again, because we are to run NGS with small panels in the last year in a fraction of patients. And the other very important aspect is that NGS with small panel is a rescue strategy in case of a failure of comprehensive genomic profiling. And we will see the data in a second. These are the volumes. Let me just underline that the Ministry of Health very clearly stated that a pathology or a molecular lab should run 5,000 NGS tests per year. Let me state very quite, clearly that I'm not sure that there is in Italy a lab that is able to run 5,000 NGS per year. So what we decided in Lombardy, we did, a very pragmatic approach. So we introduced a constant here based on the number of genes in the panel. So we multiplied, and so these are the effects. You see 150, and at the end of the day, by using this constant, we get more than 5,000. But this is a critical issue, and I think that we have to discuss that. Of course, we have to implement NGS. Probably we have to centralize tests in big labs in order to save money and get all the procedure more cost effective. But at the end of the day, this is something that we have to discuss with the stakeholders. This is a focus on more or less 9,000 patients. You see the prevalence of non-informative test. It is 12.5%. So we know that in our daily practice, 1 patient, optimistically, in 10 would not receive at the end of the day a result from NGS. This is because we are using formalin-fixed and paraffin-embedded tissue, as most of the labs, and FFPE tissue is the quality, not quantity, but the quality of DNA and RNA in FFPE is related to the pre-analytical steps. So we know that there are hurdles there, and this is the failure. So in this case, we try to action, and this is very important. And this is why it is important to stay at least integrated with a pathology lab. We get rescue techniques in order to obtain reliable data on at least with regard to the most important biomarkers for a single patient. And this is the oncoprint, that is a muscle exercise again of our bioinformaticians. Let me just focus on some of the possible approach. This is one. This is one of our experience. This is ovarian cancer. We launched this initiative along with our region because you probably recall the fact that when HRD became a biomarker for ovarian cancer, the labs were not prepared enough. So we centralize the analysis, and it's very important that these are the ESCAS1, that is more or less BRCA1 and HRD. But again, we were able to capture other possible biomarkers which could be, hopefully, will be very useful in second-line treatment for ovarian cancer patients, especially CCNE1 amplification. With regard to the amplification, we know that this is a very critical issue. We try, I mean, since we are screening patients by NGS, we try to evaluate whether copy number variation could be a reliable proxy for gene amplification. And so we developed specific bioinformatic pipeline in order to achieve this result, and we were successfully, at least for CCNE1 and HER2. But what I'm trying to convey here is that NGS should be validated internally, gene by gene, with regard to copy number variation. I think that, for example, HER2 will be very important because we know, for example, for ADCs, could be a screening method in order to, because you know that if you have a copy number gain for HER2, the chance to have an overexpression of HER2 is higher. So this kind of screening could be useful, but again, you must be very careful and robust in getting these results. Molecular tumor board, I'm going to finish my presentation. Towards a molecular tumor board, we know that there are deliberation, decrease, and so on and so forth. We launched the molecular tumor board in 2020. Actually, it was a homemade molecular tumor board, but then it was formalized. Let me see that this is a quite, complex approach because you have to include, of course, all these professionals within the core of the molecular tumor board, and then there is a very close interplay, especially with the oncology division, but also with all the research units, because it is very important. My institution in which research is one of the mission. It is very important, in my opinion, to have an interplay with the research groups, which could benefit that much by these results at the end of the day. So genetic counseling is an issue. I think that this is very important, and we probably will discuss about that. The report. So the molecular tumor board is not just a discussion among professionals. At the end of the day, you have to get a final report that must be structured, must be validated by the people working within the molecular tumor board. I mean, again, it is something that should be absolutely done in order to get indication for the treatment of the patient. In my institution, we are more or less discussing 300, 400 patients per year. Again, because, as I mentioned, it is an institution in which clinical trials are very important. The results, but let me underline some flows again. At the end of the day, our treatment assignment rate in 2023 was 9.6%. Now we have the new data from the Rome trial, which are very higher, from the trial of Nicola as well. But we have to try to implement that, especially because you see that 30% in clinical trials is a number that is too low, in my opinion, and we have to implement that again at the national level. But we demonstrated that patients discussed and treated, these are patients with ESCA scale variants more than one, so they were not treated by on-label drugs. You see that the patient treated, discussed and treated, they have the best survival. The structure could be very useful because as soon as you are able to get, the structure I just show you, you can try to implement a new technology. One is, of course, transcriptomics. The other could be single-cell sequencing, and this is a study which is currently ongoing within my institution. The PI is Andrea Vingiani. DSP could be useful as well, spatial transcriptomics, but in order to get results there, you must have bioinformaticians involved, and these are my takeaways. I think that at the end of the day, costs, as I mentioned and underlined, are not a unique issue. In my opinion, what really counts is organization. Thank you so much.
Thanks, Giancarlo, for this comprehensive presentation. Okay, we have time for only 2 short questions, 2 short answers. Please, Luca.
Thanks, Giancarlo. Great presentation. I'm curious, at the beginning you spoke about the potential use in the near future of whole exome and sequencing and whole genome sequencing and even RNA sequencing. Of course, the problem is turnaround time and cost, but let's say that we manage to solve those problems in the near future. Would you switch to those, and if yes, for which specific reason you would think that using whole exome sequencing or genome sequencing would be superior to simple large panels as we have at present time?
I mean, my answer is absolutely yes, provided that it could be, it could enter the clinical practice. And I think that it will be superior because, of course, for sure, RNA-seq will be very useful because it's able, as you know, to detect, biomarkers which are not included in target and NGS. And the same is for whole genome sequencing that is able to capture more complex matrix. Let me, for example, just mention gross deletions within the BRCA1 and BRCA2 gene. So this will be absolutely fantastic. I mean, again, the patient that should be evaluated by this kind of test should be the patient that will be, at the end of the day, discussed within a molecular tumor board. This is for sure.
Yeah. You have another question?
Thank you for your kind presentation. I have a question about costs of next generation sequencing technology. Do you think that the use by bioinformaticians of software based on decision-making, software based on artificial intelligence and machine learning can contribute to at least slightly decrease the cost of such technologies?
I completely agree with you. We are, all of us, we are involved in, initiatives which are including artificial intelligence softwares. So for sure, artificial intelligence could reduce the cost, the overall cost, provided that the software is not sold at 1 million per year, because this is the risk. Because the software will be provided by companies, for sure. So, it would be, again, another, balancing between cost and the possible, results that you, and I agree with you, you for sure could have using artificial intelligence methods. Let me just mention the mutational signatures, for example. At the end of the day, we do not report mutational signatures. So we analyze gene by gene and specific and very, very, very teeny pathways. But for sure, artificial intelligence will be able to help us in seeing the large picture that is underlying. But again, it depends on the cost of the software.
Thanks, Giancarlo. Thanks for your presentation. I guess that Rodrigo now can really expand also on this issue of artificial intelligence. So, Rodrigo, on this man, cancer genomics actionability, what is has been delivered and what is still missing?
Thanks for the invitation and the opportunity to speak here and switching to English. I'm not proficient in Italian, although I work in Barcelona, I speak Spanish, but I was born in Brazil originally, and part of the work I will present here is the real-world implementation of cancer genomics in more low-middle-income country. Here you can see my conflicts of interest. So what is the current status for genomics and targeted drugs? Every couple of years or so, I update this graph. So we have, when I started oncology, we had 5 biomarkers, and I could remember by heart what to do, what to order to every unique tumor type. Things have grown slightly in a linear fashion until 2018, and then we will discuss why, but probably advances in diagnostics, next generation sequencing in the clinics, also the learnings from big efforts like TCGA and others. We have increased the number of genes and tumors that we can act upon with targeted drugs. And that was close to exponential growth that now is going back to more linear. I think we still have room to increase, but it's not going to be like blockbuster drugs for biomarkers that are found across multiple cancer types that are going to change precision medicine in the next years. What happened in 2020-21 is a change in the regulatory landscape, more favorable. We started to approve targeted drugs based on single-arm clinical trials, and some of them tissue agnostic. Rare alterations across different tumor types, and you can see the examples here on the top for FDA and the bottom for EMA. Now, if you consider NCCN guidelines, it's almost like impossible to find a tumor type that has not a recommendation for genomic testing, being it for diagnostic purposes. Sometimes we do in sarcomas NGS for diagnosis and not really to decide therapy. And in other situations like hematological malignancies, we do a lot of NGS for prognostic purposes. We need to identify high-risk populations for more closer monitoring after transplant, for example. And it's clear that we need to rank the alterations in levels of actionability. So tier 1 and 2, these are the ones we care about. These are based on ESMO SCAT. We all know about them, ready to use in the clinics. You have an approved drug, and NCCN guidelines. In investigationals, they are just in the edge of becoming an approved indication. We have sufficient evidence in clinical trials that these alterations matched a drug in a tumor makes a difference for the patients. So maybe not in survival, but in response rate. Levels 3 and 4 are hypothetical. They are more based on preclinical evidence and applying the knowledge from one tumor type in a different indication. That is sometimes a little bit complicated to really measure the impact, and we will see the numbers. ESMO has published guidelines for multi-gene sequencing just in 2024, based on the number and the complexity of the biomarkers, tiers 1 and 2. This is the list of tumor types that should have molecular testing with multi-gene assays up front in the decision making. If you are in a research setting and for individualized care, maybe all patients with cancer should get molecular profiling. And when we talk about molecular profiling, more and more we care about a comprehensive profiling, large gene panels. You can go to the whole exome level or genome, but we cannot forget that for some tumor types, when we only care about a hotspot alteration in an oncogene, we can still survive with a single gene assay. If you are looking for tumor suppressors, complex alterations in oncogenes, rare events in oncogenes, you need next-gen sequencing. You need a small panel, at least 50 genes. But if you care about signatures, tumor mutation burden, if you are looking at the RNA level, red fusions and others, then you need hundreds of genes in your panel, maybe even RNA sequencing. And So let's review, with that in mind, what cancer genomics has delivered. If you apply broad next-gen sequencing, comprehensive profiling, and you have access to targeted therapies in approved indications, we have enough evidence, and this is from the real world in Brazil, that in lung cancer, the poster child of precision oncology, you can double overall survival if you compare with chemoimmunotherapy in patients that did not have tests or were tested but were negative for a driver. So identification of a driver and access to targeted therapies in lung cancer can double overall survival. But the question is, can we transfer this knowledge to all tumor types? So there are randomized data showing that, yeah, you may increase the opportunities for matched targeted therapies. So now we are talking about a different setting. These are previously treated advanced solid tumors that have already done all approved therapies, and I want to find potential rescue targeted therapies. And here, when you compare a comprehensive genomic profiling with a small panel, you can increase in absolute numbers 5 to 20% the actionability, theoretical. So I find 15% more absolute numbers, actionable drivers, if I do comprehensive genomic profiling in this setting. But in pragmatic terms, the ones that really get treated based on this finding, then the absolute increase is from 9% to 14%. It's 5%. So in my mind, I have to test with broad next-gen sequencing today 20 patients to find one that is benefiting from a therapy exposure, not a response, therapy exposure. I'm offering a new therapy based on this extra comprehensive genomic profiling. 1 out of 20. That is coming from a randomized trial. And you did very well randomizing patients to standard of care versus broad next-gen sequencing in this same scenario, previously treated advanced solid tumors, and you can increase here actionability rate. You can even show here response rates are going from 10% to 17%, so it's likely higher, but statistically better. And you see, mostly based on finding tumor mutation burden high and immunotherapy exposure, a benefit in progression-free survival. So this has also been demonstrated. We cannot forget that. But I did a manual review of the literature. I'm doing a PhD thesis now on that, and I reviewed everything published. You cannot read, but I will summarize. So there are many global prospective precision oncology initiatives, and the numbers we care about is how much actionability I find and how many patients I treat based on this actionability. And these are the median numbers, 35%, so 1 out of 3 patients. The setting really is not lung cancer metastatic that you have to test yes or yes. I'm talking about everyone else. If you get tested, 1 out of 3 patients, and 15% are eventually treated based on your profiling. So if you are pragmatic, from the entire population eligible to molecular testing, 100%, those that get informative results, that have pathogenic alterations, those that are actionable in theory, that are eligible to treatment, that finally receive the treatment and responded to therapy, it goes down to 3-4%. And it's not my estimation. This is like many trials have shown exactly this number. So, so it's 1 out of 20 to offer and 1 out of 30 to benefit. So this is the reality. If you don't do precision medicine following the rules, so if you don't have a research ecosystem to offer off-label drugs or clinical trials, if you only do because you're curious and finding like a last resource for this patient, it's not going to work. So many have published that this is an illusion. Precision oncology is an illusion. So it's too much, I mean, spending and expectation for a patient and low benefit. So I think that we cannot do improvised medicine. This is precision medicine, so we need to follow the rules, use NGS wisely, and in a network offer the best therapy that can be clinical trials. So, and the rules are very simple. So it's not magical. So some alterations have actionability in one tumor type only, PIK3CA mutations. They are actionable in breast cancer, not in head and neck. There are other alterations in the middle that are actionable in more than one tumor type, but not for all tumor types. BRCA alterations or HER2 mutation. It's not actionable in colorectal cancer. But there are some, and this is our expectation, and also this is why we fight every day for NGS and clinical trials. Some of them are tumor agnostic. They make complete difference, and then access to drugs may change lives for patients across tumor types. So what is missing? What is missing is really we working together in an environment that is favorable for NGS to be impactful. So if we only do things, out of, so as really a rescue therapy, that's not going to work. You need molecular tumor board discussion, choosing which patients must be tested at what time point, defining what is actionable together with experts, finding approved drugs, clinical trial, or some off-label indications that can have an impact. And we need to measure performance indicators of your program. You need benchmarks. There are minimum 10% of the patients that you test must be treated. Minimum. Ideal, optimal is 33%. The recommended is at least 25%. In the best case scenarios, we are doing 10%. So this is really not doing well. And we have seen already that if you are able to implement the MTB decisions, you can change the outcome of patients. I'm going to show the experience at Vall d'Hebron. Vall d'Hebron is known for a very big clinical trials unit and phase 1 clinical trials. And this is like really a perfect scenario to assess the value of genomics profiling. We have started broad panels since 2014. You can see on the right, you can see in color the number of molecular tests we do every year. It's growing and has grown a lot in 2023 when we started liquid biopsy. Now we test about 2,000 patients every year, and we do about 3,000 NGS, broad panel NGS, tissue and or liquid. We also do immunohistochemistry and smaller panels. You can see on the theoretical actionability figure the proportion of patients that we consider actionable by looking at NGS. Levels 1 and 2, this is I must act because you have an approved therapy or because I have a clinical trial with already promising data, it's 15%. And when we go to levels 3 and 4, so preclinical actionability, this goes up to 53%. So these are the numbers. So clinical trials is the mechanism of access to therapy at Vall d'Hebron. We have no opportunities for off-label therapy. So it's either approved therapy or a clinical trial. We have every year about 500 clinical trials, and about 30%, you can see, have a molecular guided approach. And this has gone down a little bit during 2020-21, and this is because there was this explosion of immunotherapy. So when immunotherapy started, the precision oncology targeted therapy drug development did shrink a little bit, but now it's coming back. And you can see in the biomarkers, like multicolor figure also, how important it is to have multiple clinical trials for multiple biomarkers so that you can then impact in patient access to drugs. And here I show the key performance indicators. No? In yellow, you see, dark line, the proportion of patients treated over the overall population tested. We started at 1%, we go in 2018-19, 7%. During the pandemics, it went down to 4%, and then back again to 10%. So 1 out of the 10 patients we test receives a targeted therapy. And in the dashed line, you can see the proportion that received approved drugs. It has gone up from 1% to 4% during pandemics, but then stable. So this means that I have 2-3 times more patients treated at Vall d'Hebron with clinical trials than approved therapies. So we invest in clinical trials so that we save money with approved drugs because they are expensive. So we prefer to pay for the test and engage with industry for trials than paying for the drugs. You know? So that was the strategy. And in the other green figure, you can see the key performance indicator from ESMO, which is what is the proportion of patients treated out of those with actionable. And by actionable here, I mean 1, 2, 3, 4, all the levels of actionability, because we have clinical trials for preclinical biomarkers. So I'm very conservative here. And we are fluctuating between 25-30%. So we are at Vall d'Hebron at the recommended level. 1 out of 4 patients with actionable alterations eventually receives a targeted therapy. And we have experienced also the literature from off-label therapies. So what is the impact of off-label therapies? The definition of off-label is very dynamic. So this publication, the largest from 2 years ago, they consider BRAF an off-label indication. Now it's a tumor agnostic. HER2 mutation in lung cancer was off-label, but now it's on-label. So you can see that, you get responses with off-label therapies, but only for very robust biomarkers in clear indications. You know? So, and if you are trying to treat patients based on preclinical evidence, you can see in the Kaplan-Meier curves, the green line is patients with level 4 actionability. This is preclinical evidence only. Response rate, 5%. Progression-free survival, 2 months. So if you treat based on preclinical evidence only, you will not benefit your patients. I rather give chemotherapy than the treatments based on preclinical evidence. You know? The testing rate, even in the US, where you have a favorable, like a private setting also to cover the cost of testing, is just slowly increasing. And so, for example, prostate cancer, it's now at 50%. Every patient with metastatic prostate cancer, castration resistant, must be tested for HRR alterations. You have approved drugs. But the reality is that even in the best scenario, many patients are not being tested. And in Europe, we have an even worse situation in terms of access to the technology. Here you can see in the graph many red squares here. These are large next-gen sequencing, liquid biopsies across different countries. It's available only in research or never. You know, so the reality is that in very few institutions you have access to all these technologies that we care about. And the cost of cancer drugs after pandemics is also increasing a lot. So you see, for biologics and small molecules, we are now paying a median annual cost for molecule of about $350,000, $450,000. And so it is impossible to sustain. It's not the cost of next-gen sequencing. We can run next-gen sequencing, including bioinformatic cost, without AI, for $1,000 US. But if you have this price of drugs, it's going to be completely unsustainable. So we have to work for access to drugs in clinical trials if you want to survive. So in conclusion, there is a linear increase in actionability for cancer biomarkers. The outcomes of targeted therapies is favorable in approved indications for tier 1 and 2, so no brain here. The benefit of off-label drugs is low if you're considering tier 4 alterations. Clinical trials are essential mechanism of access to precision oncology drugs, but you have to follow guidelines for testing and drug matching and choose wisely in molecular tumor boards. Do not give for oncologists the decision to decide on therapies for preclinical biomarkers. There are many barriers for pragmatic actionability that go beyond access to the test and the drugs. So, and we have to measure and publish the real-world KPIs, so the ratio of patients treated over those that are actionable in your institution. And you can also eventually publish in ESMO Real World Data journal. So this is a promo. I'm the editor-in-chief of the journal just launched by ESMO. And we have obviously a special interest in precision oncology in the real world. Thank you for the attention.
I guess there would be many questions on the presentation by Rodrigo. Well, I can start. Okay, Rodrigo, I guess we all agree that we should not do comprehensive genome profiling if we have no available clinical trials. Still, beyond the clinical trials that come from the companies, I guess there is also room for academic initiative. And I fully agree that this story of the actionability is difficult to address, but one of the issues is also that we still stratify patients on a single biomarker, and maybe we should go beyond this. So if you could give
the future is really going for complex biomarkers. So complex biomarkers is we using a single alteration is not enough, so you have to look at one biomarker in the constellation of alterations that you see in the tumor. So how is this HRR alteration together with the signature of TMB high? How to prioritize therapy, how to understand what is the mechanism that is really driving progression. So yeah, and this is unfortunately not something we can teach an AI algorithm to do. So that's a lot of, again, of the effort around the molecular tumor board is on the complex biomarker interpretation.
Yeah, also, there's a question. Giancarlo. Please.
Just a very quick question. I'm curious, what's your opinion in including among the actionable targets the potential germline variants that you are able to capturing within, because this is quite debatable issue.
No, you're right. I have not touched on the germline actionability. Now we have at least 5 gene alterations that if you find it is a germline event, you can give matched drug. This can be like even VHL mutations in renal cell carcinoma for specific therapies and beyond this BRCA. Yes, we have to follow the rules for reflex germline referral and testing. So every tumor board, every next-gen sequencing report must have a flag of germline actionability. It's very unusual to interpret the solid tumor NGS with a with an oncogenetics mindset. So we have to still report based on, again, the actionability as a solid tumor next-gen sequencing. But there are some genes that, depending on the tumor type and patient age, we must flag as potential germline and actionable. So we cannot officially recommend a match therapy if you have not proven that this alteration is germline. So we have to add as off-label indication and must confer with germline testing. But I completely agree that these are actionable. Yeah.
Beyond this, I guess that if you find any potential germline, you can find a family in which you can reduce the burden of cancer death related. So it's important. There's a last question.
Thanks a lot. So since you provided a less optimistic view on the percentage of patients that eventually get actionable, truly actionable alterations after ingest, I'm going to ask you the same question that I asked Giancarlo before. So do you think that the case can be made for actually, at least in some patient populations, expanding the range of genomic profiling through whole exome or whole genome sequencing? Do you see this happening in the near future, or do you think it's at present time unsupported by evidence?
Yeah, so as of today, I don't see, beyond some indications like pediatric cancer, I think in pediatrics you have to start with whole exome because you have to always do germline together, and you need to interpret, you need to look for fusion alterations that are quite relevant in pediatric cancer. So some hematological malignancies, some rare sarcomas also may benefit from broader profiling with whole exome, whole genome. But unfortunately, all the publications we have now in the literature are with application of whole exome. They are super biased, so over-interpreting the actionability. So they give chemotherapy because they found genomic instability, like a platinum-sensitive preclinical biomarker. So they tend to be over-optimistic on the impact of whole exome sequencing. I have not seen any serious study documenting that you critically need whole exome beyond a large panel. I would rather favor RNA sequencing together with DNA in a large enough panel than whole exome, whole genome sequencing. What you gain really is the copy number level and the signature. This is definitely, but right now at the copy number and signature level, very few things are actionable. They are more hypothetical than pragmatical. So I would prefer DNA, RNA to find fusions across tumor types than expanding to whole exome for selected tumors.
Thanks. I guess we need to move to the next session. Thank you again, Rodrigo. Una comunicazione di servizio, mi dicono che la room 2 non è più qui, ma è a piano 0, per cui chi vuole. Passiamo all'italiano, mi dicono, c'è Stefania Tommasi. E iniziamo, ciao. E credo che dobbiamo iniziare subito con la prossima sessione. Quindi, Pierluigi. Per favore, se vi potete sedere, in modo, siamo già in ritardo e dobbiamo iniziare la prossima sessione. Lascerei la parola a Stefania Tommasi.
Buongiorno, incominciamo allora subito questa sessione sulla biopsia liquida, che penso che sia un punto essenziale non solo per la ricerca ma anche per la diagnostica e La diagnostica clinica, infatti sentiremo ora parlare la dottoressa Del Re proprio su questo, ed è importantissima per la valutazione di biomarcatori e soprattutto per la possibilità di evidenziare questi negli stadi precoci della malattia. Io darei subito la parola allora alla dottoressa Del Re.
Grazie mille, buongiorno a tutti. Io ringrazio tantissimo Alleanza contro il cancro e Ruggero e Stefania per questo graditissimo invito. Dico la verità, quando ho visto questo titolo non ho resistito e ho approcciato questa presentazione volendovi un po' raccontare quella che è stata la storia della biopsia liquida fino al giorno d'oggi, non ovviamente soffermandoci su ogni tappa, quanto per lo più soffermandoci su quello che ogni milestone, ogni obiettivo raggiunto ci ha insegnato nell'ottica di poter traslare ad oggi, utilizzare oggi la biopsia liquida in pratica clinica. La prendo un po' ariosa, come si dice in Toscana. Questo è stato di fatto l'arrivo, il primo utilizzo della biopsia liquida in pratica clinica, che arriva, permettetemelo, sono una farmacologa, che arriva con un farmaco. Arriva con osimertinib perché era un farmaco che andava prescritto con la comparsa di una mutazione di resistenza e quindi si rendeva necessario non più biopsiare un paziente ma utilizzare metodi alternativi. La cosa interessante qual è? Ovviamente non vado nel dettaglio di questa storia che ormai conosceremo probabilmente tutti benissimo. Se vedete le curve però rispetto alla stratificazione che si otteneva valutando la mutazione di EGFR su tessuto, quella sul plasma lasciava un po' a desiderare, la vedete in basso alla vostra destra. Le curve non erano perfettamente sovrapponibili a quella dell'identificazione della stessa mutazione sul tessuto e in realtà ve l'ho messo nella curva grande in mezzo, andando a fare una serie di studi aggiuntivi su sottogruppi di pazienti abbiamo visto che forse la biopsia liquida andava interpretata diversamente rispetto a quello che ci aspettavamo. E quindi abbiamo iniziato a capire che c'erano pazienti, addirittura quelli che erano negativi per le mutazioni di EGFR sul plasma, che erano quelli che avevano la progression free survival più lunga. Come mai un farmaco che andava dato sulla base di una mutazione aveva la PFS più lunga quando la biopsia liquida era negativa? Beh, in realtà questo ci ha fatto immaginare, ci ha fatto ipotizzare una serie di cose. Ho voluto intitolare questa diapositiva come take home messages che ci ha insegnato osimertinib in realtà. Ci ha insegnato due punti cardine, due che non si sono smossi da allora ad oggi, cioè che il plasma, l'analisi su plasma, su biopsia liquida e quella su tessuto sono due approcci che devono essere complementari e che se il plasma è positivo, allora possiamo proseguire e fidarci di questo risultato. Qualora però il plasma sia negativo, dobbiamo fare una biopsia tissutale. Perché? Perché il falso negativo in realtà è l'ostacolo maggiore che ad oggi, qui parliamo di un risultato ottenuto nel 2013-2014, che ad oggi non abbiamo ancora superato completamente. Quindi attenzione al falso negativo, un falso negativo che però a volte, lo vedete, io ho riportato proprio la frase della discussione che è stata fatta in fondo al lavoro che vi ho fatto vedere, in parte potrebbe voler dire che il tumore è un tumore a basso indice proliferativo e quindi poco aggressivo e quindi indicativo di una prognosi migliore. E allora che cosa abbiamo imparato da questo primo risultato che ci siamo portati dietro e che ci ha insegnato ad applicare la biopsia liquida anche in altri tumori fino a diventare di fatto utilizzo quotidiano? Questi risultati ci hanno insegnato innanzitutto che la tecnologia è tra le cose più importanti. I risultati che vennero generati con lo studio di osimertinib valutavano la biopsia liquida utilizzando la metodica COBAS, che all'epoca non aveva una sensibilità appropriata per analizzare questo tipo di analiti. E infatti la concordanza tra biopsia liquida e tessuto era intorno all'80-85%. Oggi sappiamo che le metodiche più idonee all'analisi di biopsia liquida sono una PCR digitale piuttosto che un NGS, che hanno ovviamente una concordanza del tutto sovrapponibile, la vedete sulle rette in alto alla vostra sinistra, anche se entrambe, lo vedete riportato nella tabella, hanno dei pros e dei vantaggi e degli svantaggi che vanno da costi, quantità di analitiche che possiamo utilizzare, che ovviamente dobbiamo considerare quando scegliamo quale tecnologia utilizzare sulla base di quale tumore approcciare, qual è il quesito clinico, qual è la tempistica, qual è il turnaround time. C'è una cosa importante però che l'NGS ci dà in alcuni casi e la PCR digitale no, e che ci aiuta nell'identificazione dei falsi negativi. Con alcuni pannelli molto grandi in NGS è possibile valutare la cosiddetta tumor fraction, che è il quantitativo totale di DNA tumorale circolante che possiamo identificare. I dati ci dicono che se la tumor fraction è inferiore a 1 e non troviamo mutazioni somatiche nel plasma, allora il rischio di un falso negativo è molto alto. Se la tumor fraction è maggiore di 1, la concordanza tra biopsia liquida e biopsia tissutale è quasi del 100% e se non troviamo nessuna mutazione somatica possiamo stare, direi, abbastanza tranquilli che non si tratta di un falso negativo. Ora, come mai questo falso negativo in biopsia liquida? Perché il falso negativo dipende dalla biologia della malattia. Questa è un'altra cosa che abbiamo imparato guardando i risultati che pian piano sono stati generati. È vero, la biopsia liquida ha il grandissimo vantaggio di cogliere l'eterogeneità molecolare intra- e intertumorale, ma è anche vero che il DNA tumorale circolante subisce delle variazioni in termini di rilascio, che noi chiamiamo shedding, sulla base delle caratteristiche biologiche della malattia. E quindi parlo di carico di malattia, carico tumorale, sedi di malattia, viscerale piuttosto che ossea, piuttosto linfonodale, e quindi i tumori a grande carico di malattia, prevalentemente viscerali, come il tumore del colon retto, sono i tumori che rilasciano la maggior parte di DNA tumorale circolante e in cui più difficilmente troveremo falsi negativi. Tant'è che possiamo permetterci di analizzare da 1 ml di sangue ed essere sicuri di quello che troveremo in biopsia liquida in termini di validità del risultato se parliamo di un paziente con un tumore del colon metastatico. Se analizziamo un paziente con tumore del polmone metastatico, tumore del polmone, lo vedete nella figura in basso a sinistra, risente di moltissime variabili che impattano sullo shedding del DNA tumorale circolante, come ad esempio l'indice fibrotico, che impedisce il rilascio del DNA tumorale circolante, tant'è che i falsi negativi nel tumore del polmone arrivano addirittura tra il 25 e il 30%. Quindi attenzione quando analizziamo le biopsie liquide dei tumori a valutarne il carico di malattia e le sedi di malattia. Ma visto che la biopsia liquida era lo strumento più indicato per andare a ricercare mutazioni di resistenza, abbiamo applicato, è stata applicata ovviamente anche in altre tipologie. Il secondo tumore ad avere un'applicazione registrata di biopsia liquida è il tumore della mammella, dove andiamo a valutare la mutazione, l'insorgenza della mutazione dell'estrogeno recettore nelle pazienti estrogeno recettore amplificate, mutazioni che compaiono dopo più linee di ormonoterapie. Perché? Perché l'ormonoterapia adotta una pressione selettiva che fa sì che si acquisiscano dei cloni mutati. Non vado nel dettaglio di questo studio né delle mutazioni dell'estrogeno recettore, però passando a questo argomento mi permetto di dare un altro risultato rispetto a quello che abbiamo imparato. Quando noi utilizziamo la biopsia liquida oggi dobbiamo considerare la frequenza allelica, la VAF. E molto spesso quando abbiamo iniziato a lavorare sulla biopsia liquida la domanda che più spesso i clinici mi facevano era: sì, però trovo lo 0,02% di mutazione, è clinicamente rilevante? Per i motivi che ci siamo detti prima, cioè dello shedding che è variabile a seconda della biologia di malattia, sì, lo 0,02%, 0,001% di una mutazione nel plasma è clinicamente rilevante. Noi abbiamo una mediana di frequenza allelica nel plasma dello 0,3%, lo vedete nel grafico alla vostra destra, e se stratifichiamo, le curve che vi faccio vedere sono quelle delle pazienti che rispondono, che sono trattate con elacestrant stratificate sulla base della frequenza allelica della mutazione di SR1, vedete che le curve di fatto non si separano, quindi a prescindere dalla frequenza allelica le pazienti rispondono in egual misura al trattamento. Ma la biopsia liquida ci ha insegnato anche che noi possiamo monitorare le mutazioni durante il trattamento e possiamo addirittura predire in anticipo quella che sarà la progressione di malattia. Questo è uno dei moltissimi dati in cui si fa vedere che la mutazione, in questo caso era un EGFR nel tumore del polmone, inizia a ricomparire nel plasma e ad aumentare mesi prima della progressione radiologica. Questo è solo uno dei dati, poi in realtà sono arrivati dei trial, questo è stato il primo, l'Apple trial, che valutava lo switch da gefitinib a osimertinib, guardate il braccio B e il braccio A, sulla base, braccio B, della comparsa della T790M nel plasma, braccio C comparsa progressione radiologica. Questi erano i risultati, sicuramente significativi ma non ottimali, ma se pensate al braccio B dell'Apple Trial, che cosa vi viene in mente? Chi è un po' più addentro alla materia sicuramente ci avrà pensato il Serena 6, uno studio che ha fatto molto scalpore quest'anno all'ASCO, che prevede uno switch in termini di trattamento nelle pazienti con tumore della mammella non a progressione radiologica di malattia, ma alla progressione molecolare, cioè alla comparsa di una mutazione su plasma prima che avvenga la progressione clinica, la progressione radiologica. Queste sono le curve e anche in realtà questo studio ci insegna qualcosa, ci insegna che possiamo monitorare le mutazioni in biopsia liquida per gestire il trattamento. Questo è un altro degli esempi che vi riporto, non recentissimo, ma proprio per farvi vedere quanto è stato fatto in questi anni, dove addirittura si utilizza la comparsa o la clearance di una mutazione per fare un re-challenge di trattamento. In questo caso si parlava di tumore del colon retto con il re-challenge dell'anticorpo cetuximab. Che cosa ci insegnano questi studi? Ci insegnano che forse dobbiamo iniziare a pensare ai cosiddetti liquid biopsy resist, quindi non solo più i resist radiologici, ma un po' come fanno in ematologia dei resist liquidi. Sicuramente dobbiamo perfezionare e considerare le tecnologie migliori, ma dobbiamo ancora capire quali sono i time point rilevanti a cui monitorare le mutazioni su biopsia liquida. Ogni 3 mesi, 1 mese, 6 mesi, gli studi in letteratura su questo sono assolutamente eterogenei. E poi qual è la threshold per valutare la comparsa o la scomparsa di una mutazione in termini clinicamente rilevanti? Questo ancora è qualcosa da decidere, ci sono dei dati che ancora non sono conclusivi. Abbiamo anche imparato che in biopsia liquida possiamo imbatterci nei cosiddetti incidental findings, quindi tutto ciò che è germinale ed ereditario, ma possiamo anche trovare delle varianti che sono di origine che provengono dall'ematopoiesi clonale. Le cellule invecchiano, acquisiscono mutazioni. Il problema rilevante qual è? Che queste mutazioni molto spesso sono in geni che sono anche geni cosiddetti druggable. Infatti se vedete, scusate è un po' sfocata, però vedete che le mutazioni dell'ematopoiesi clonale possono comparire in mutazioni come CEC, ATM e DH1, che possono essere a seconda dei tumori predittive e prognostiche. Stiamo aggiornando le raccomandazioni IOM sia PEXIBIO che SIF sulla biopsia liquida, vedete l'estensione delle patologie alle quali abbiamo aggiornato, se è in corso d'opera il dato ad oggi. Abbiamo incluso il tumore del colon retto, il colangiocarcinoma, la mammella, il polmone, la prostata, la vescica, tutte quelle patologie che hanno la presenza potenziale di mutazioni che possono essere rilevate in biopsia liquida laddove il tessuto sia insufficiente, che ci possono aiutare a gestire in termini terapeutici il paziente. Le ultime diapositive sono per farvi vedere quello che arriverà molto probabilmente. Ci spostiamo dal setting metastatico, dove la biopsia liquida è pratica clinica ad oggi, al setting più precoce, quindi è tutta la valutazione della malattia minima residua con biopsia liquida. Questo è solo uno degli esempi, il Niagara Study, che è il trial che è stato fatto nel tumore della vescica. Al di là dell'intervento terapeutico su cui non mi soffermo, questi sono i risultati. I pazienti con DNA tumorale circolante post terapia neoadiuvante, post cistectomia, è un biomarcatore predittivo di recidiva e di event-free survival. Stessa cosa nel tumore del polmone, potrei portarvi molti esempi. Questo è l'EGIAN, valuta nuovamente, a fronte di una strategia terapeutica neoadiuvante ed adiuvante, l'utilizzo del DNA tumorale circolante come indice di malattia minima residua. Attenzione, perché quando parliamo di malattia minima residua nei setting precoce dobbiamo identificare nuovamente la tecnologia, quindi una tecnologia tumor informed oppure tumor agnostic, ovviamente da valutare sulla base base dell'esito che ci si pone. Su cosa si basano? Si basano su metilazione prevalentemente, parlo della tecnologia tumor agnostic, metilazione e frammentazione. Vado verso la fine, questa è la nuova frontiera della biopsia liquida. Di fatto sia la metilazione che la frammentazione riconducono il DNA tumorale circolante al tessuto di origine, e quindi è possibile con la biopsia liquida identificare non solo malattia minima residua, ma anche capire da quale tumore questo DNA ha origine. Questo è il sistema per mezzo del quale FDA ha approvato il test di Guardant come screening precoce per i tumori del colon retto. È stato approvato a marzo 2025. Vi riporto qua giù, giusto perché stiamo cercando di espandere i dati, quello che è in termini di tempo il rilascio temporale, cioè con quanti mesi o con quanti anni di anticipo è possibile trovare il DNA tumorale circolante nel plasma sulla base dei vari tumori. Vedete che c'è una variabilità notevole. Vorrei concludere questa mia presentazione con questa frase, ovviamente che parla di sogni, ricongiungendo di fatto al motivo per cui sono qui, e volendovi invitare a collaborare con noi. Io sono arrivata a Roma al Gemelli un anno fa, perché il professor Scambi ha firmato un agreement con Guardant per mettere a punto un test di biopsia liquida che rendesse il Gemelli hub nazionale per i test per profilazione molecolare. Il nome del test si chiama FPG 360, che sfrutta per l'appunto una piattaforma Guardant, ma verrà eseguita tutta la parte sia analitica che di refertazione da noi al Gemelli. Io con questo vi invito a collaborare con noi, saremo attivi dal 1° ottobre, quindi potrete mandarci test sia per pratica clinica sia per ricerca, e vi rubo gli ultimi 30 secondi, mi dispiace ma non hanno aggiornato la presentazione, però vi volevo invitare ad iscrivervi alla Società Italiana di Biopsia Liquida. Avevo aggiunto una diapositiva ma probabilmente non hanno uploadato l'ultima versione, mi dispiace molto. Abbiamo fondato da qualche mese la Società Italiana di Biopsia Liquida, alla quale vi invito ad iscrivervi. Molti membri del direttivo sono qui presenti a questo congresso, c'era la foto con i nomi, però io sono a disposizione se avete bisogno di informazioni e curiosità, resto qui fino a domani. Grazie.
Grazie Marzia, una sola considerazione. In effetti c'è ancora diciamo una grossa sfida da dover vincere per la biopsia liquida, cioè tutti quei casi in cui appunto abbiamo diciamo, l'assenza del marcatore, quindi delle biopsie liquide non informative. E quindi io non so questo pannello che state utilizzando del Guardant quanti geni abbia, ma andando ad analizzare chiaramente nella pratica clinica, andando ad utilizzare un pannello piccolo, molto spesso appunto i casi risultano non informativi, e quindi dobbiamo assolutamente vincere questa sfida.
Assolutamente. Quello che noi suggeriamo sempre quando analizziamo le biopsie liquide è non analizzare mai il dato di per sé, ma tenere sempre presente anche la clinica, e quindi il carico di malattia, le caratteristiche del paziente e soprattutto il momento temporale di quando è stato fatto il prelievo di sangue.
Sì, e probabilmente questo lo supereremo andando a stimare la tumor fraction con i nuovi pannelli. E anche da questo punto di vista, con questa storia della VAF, io sono d'accordo che non c'è una correlazione con la VAF, però noi nel polmone abbiamo poi imparato nel tempo che invece il T790M più clonale risponde meglio rispetto al T790M, diciamo, è subclonale. Quindi anche su questo credo che invece andando a rapportare la tumor fraction forse qualcosa in più impariamo, perché la VAF da solo non deve guidare la nostra decisione terapeutica, però non è che proprio tutte saranno uguali, anche da questo punto di vista. Ci sono domande? Carmine.
Grazie Marzia per la bella presentazione che hai fatto. L'hai detto, la biopsia liquida è già pratica clinica, e ti faccio proprio una domanda di pratica clinica. Noi a breve avremo, proprio nel polmone, avremo nella malattia EGFR mutata più possibilità terapeutiche che hanno problematiche di costi, problematiche di tossicità, e avremo una problematica di gestione dei pazienti. Uno dei criteri potrebbe essere correlato proprio alla clearance su biopsia liquida di EGFR. Vedremo cosa deciderà l'agenzia da un punto di vista regolatorio, perché ci sarà tutto un problema regolatorio. Il mio quesito è quali sono le barriere però, perché con Nicola stiamo conducendo uno studio a livello nazionale nel colon su biopsia liquida in prima linea, e mettere su delle reti veloci e funzionanti non è stato facilissimo. Noi siamo stati fortunati a riuscire a gestire 90-100 centri. Ma a livello nazionale quali sono le barriere che soprattutto domani il problema con il GFR polmone ce l'avremo?
La tua domanda mette il punto su una cosa molto importante, cioè i centri di riferimento. La biopsia liquida è una tecnologia che va gestita con molta esperienza. E al di là dei test convenzionali, molecolari, germinali, è difficile improvvisarsi a fare questa tecnologia. Quindi sicuramente quello che posso dire è centralizziamo, anche perché la centralizzazione ci aiuta nella gestione dei costi e nella gestione delle tempistiche. Ci sono dei turnaround time lunghissimi a seconda dei volumi dei laboratori. Punto secondo, la clearance è qualcosa di molto delicato, perché se consideriamo che il limite maggiore della biopsia liquida è il falso negativo, parlare di clearance ci mette veramente in una condizione delicata. Serve esperienza, serve la tecnologia giusta e serve molta validazione dietro questo processo. La tumor fraction ci aiuterebbe, una tecnologia alla base per valutare l'effettiva clearance che valuti la metilazione, per esempio, ci aiuterebbe. Un doppio testing con il costo dietro che si porta dal punto di vista economico. Ci sarà un bel salto da fare e sicuramente il regolatorio nella gestione dei costi di queste tecnologie deve venirci dietro, altrimenti non ci spostiamo.
Grazie. Altre domande? Una di su.
Sì, ciao, grazie. Intanto bella presentazione, perché hai toccato un po' tutti i punti critici effettivamente di un laboratorio che si occupa, che fa diagnostica su liquid biopsy. La domanda è molto specifica. Cosa non avendo la possibilità di valutare in maniera semplice la tumor fraction, e questo è il limite, è la prossima sfida secondo me. È bello parlare di 500 geni, 1000 geni, possiamo fare tutto, però in liquid biopsy noi abbiamo un problema, dobbiamo capire qual è la tumor fraction. In questo momento mi verrebbe da dire che per non brancolare nel buio il laboratorio che fa pratica clinica, pratica diagnostica, lavorerebbe molto meglio se avesse da parte del clinico informazioni cliniche banalmente. Spesso ci vengono mandati vengono inviati pazienti che hanno magari un carico di malattia ancora piccolo, quindi mi chiedo e ti chiedo se non è il caso magari di fare un po' di campagna di sensibilizzazione con i colleghi oncologi, di introdurre nelle richieste per liquid biopsy il maggior numero di informazioni cliniche possibile, perché in questo momento è l'unica cosa che ci può aiutare quando refertiamo un dato non informativo, perché in liquid biopsy negativo non esiste.
Assolutamente. Io ti dico quello che facevamo noi quando siamo partiti a Pisa nel 2013, proprio perché brancolavamo nell'incertezza più assoluta. Oggi siamo già più a nostro agio. Io avevo stabilito una sorta di test requisition form dove chiedevo la tipologia di malattia, sito delle metastasi, carico di malattia, mutazioni, qualora avessero fatto un tessuto, già presenti sul tessuto. Quando però ci troviamo in condizioni limite, quindi tumore piccolo, incertezze, un negativo, cercate sempre, a meno che non abbiate un percorso standardizzato e vincolante, di giocare sui volumi. Quindi aumentate il volume di plasma di partenza, riducete il volume di eluizione finale. Qualora vi aspettate qualcosa ma avete un risultato negativo e lo dovete ripetere. Però sì, sicuramente un test requisition form che vi aiuti sulle informazioni cliniche di trattamento e di clinica del paziente è necessario.
Ok, grazie Marzia. La dottoressa De Michelis non è potuta essere presente, quindi Proseguirei chiamando la dottoressa Costantini che ci parlerà di metabolomica, lipidomica e citochinomica.
Buongiorno a tutti, io voglio per prima cosa ringraziare gli organizzatori per avermi dato la possibilità di presentare questi nostri risultati relativi alla valutazione del profilo metabolomico integrato con altre scienze omiche. Tutto nasce dal fatto che ormai in letteratura è riportato in grande quantità come la deregolazione del metabolismo cellulare sia correlata allo sviluppo e alla progressione tumorale, infatti essa è stata poi inserita tra i cancer roll marker. La cosa importante però che stiamo cercando di fare in questi ultimi anni è quella comunque di integrare la valutazione del metaboloma con altre scienze omiche, perché solo in questo modo ci siamo resi conto che è possibile poi identificare dei veri marcatori. Tutto è nato nel 2015 quando presso i laboratori di Mercogliano dell'Istituto Pascale è stato acquisito uno strumento di risonanza magnetica nucleare in grado di effettuare studi untargeted. Ultimamente abbiamo acquisito anche una spettrometria di massa ad alta risoluzione accoppiata a cromatografia liquida che stiamo usando per far condurre studi anche targeted e speriamo di acquistare anche un gas cromatografo nei prossimi mesi, nei prossimi anni, per poter valutare anche metaboliti volatili. In questi anni abbiamo lavorato su svariate matrici biologiche, in particolar modo voglio porre l'attenzione su alcuni fluidi biologici. Infatti abbiamo lavorato sia su fluidi follicolari, sia su condensati respiratori, salive, anche feci e in particolar modo poi su campioni plasmatici e campioni serici di pazienti con cancro del colon retto e con melanoma. Oggi vi presento i risultati che abbiamo recentemente prodotto sulla sindrome di Lynch, che è una delle patologie ereditarie più diffuse, caratterizzata da difetti nel sistema di riparo del DNA, ma anche da alcune mutazioni genetiche. La cosa fondamentale è che una grossa percentuale di questi pazienti tendono a sviluppare cancro, anche se, come ho indicato nella diapositiva, in rosso, non tutti sviluppano cancro. Quindi l'aim di questo lavoro è quello di andare a identificare dei possibili marcatori che possano predire lo sviluppo di cancro nei portatori di sindrome di Lynch, tenendo d'occhio anche quelli che sono i fattori dello stile di vita, proprio perché la letteratura ha evidenziato, anche in un paper molto recente, come gli stili di vita possono aumentare il rischio del cancro in questa tipologia di pazienti. Nell'ambito dello studio Biocor, in collaborazione con la dottoressa D'Angelo della divisione di gastroenterologia del nostro istituto, sono stati collezionati campioni plasmatici di 82 pazienti, di cui 40, come è indicato nella diapositiva. Non so che cosa è successo. No, si è spento il... Ah, sono stata io? Ok, scusi. Ok, grazie, scusatemi. Di cui nella diapositiva sono presenti 40 pazienti con storia di cancro, invece altri 42 che non hanno mai sviluppato cancro. In particolare voglio evidenziare che il gruppo dei pazienti con cancer history presenta e comprende una percentuale più alta di soggetti obesi e che il 70% di questi pazienti ha sviluppato cancro del colon retto, mentre gli altri hanno sviluppato cancro della tiroide, dell'endometrio e dell'ovaio. Quindi lo scopo di questo nostro lavoro è stato quello di andare a valutare il profilo metabolomico e lipidomico mediante risonanza magnetica nucleare, il profilo citochinomico costituito da un pannello di 48 citochine, chemochine e fattori di crescita e quindi, come ho anticipato prima, il nostro aim è quello di andare a identificare quali fossero questi fattori che sono specifici del gruppo con cancer history e poi capire se tra questi fattori ce ne fosse qualcuno correlato con il BMI e lo smoking. In questa diapositiva vi presento i risultati della valutazione del profilo metabolomico, dove dallo score plot abbiamo una distinzione tra i due gruppi con una varianza del 22%. Questa differenza è dettata dai metaboliti presenti nel VIP plot e in particolare nel gruppo con cancer history abbiamo ritrovato più alti livelli di lattato, di tre-idrossibutirrato, più bassi livelli di istidina, di glucosio, di piruvato, ma anche di altri amminoacidi quali per esempio l'isoleucina e la leucina, che in letteratura sono comunque indicati come alterati nel cancro e correlati allo sviluppo e alla progressione tumorale. Anche in un nostro recente paper pubblicato su pazienti con cancro del colon-retto metastatico abbiamo infatti ritrovato che alti livelli di tre-idrossibutirrato e bassi livelli di istidina sono correlati ad una cattiva outcome di questi pazienti. L'analisi del profilo lipidomico ha evidenziato che nel gruppo dei pazienti con cancer history abbiamo livelli più alti di molti lipidi, tra quali gli acidi grassi, i trigliceridi, il colesterolo, i fosfolipidi, e livelli più bassi di omega 3 e acido linoleico, che sono riportati in letteratura come correlati a un maggiore rischio di sviluppare cancro. Proprio perché gran parte di questi metaboliti, soprattutto lipidi, sono comunque correlati al metabolismo, abbiamo valutato anche un pannello di citochine, chemochine e fattori di crescita, come ho detto prima, e in particolare voglio porre l'attenzione sul fatto che nel gruppo dei pazienti con cancer history abbiamo ritrovato più alti livelli di alcune chemochine molto note in letteratura come correlate allo sviluppo tumorale, quali per esempio i livelli della MIG, della CXCL12, ma anche un fattore di crescita HGF. Proprio perché la nostra idea, come ho anticipato prima, è quella comunque di andare a studiare anche la correlazione che c'è tra il metabolismo e l'infiammazione, focalizzando sui livelli di questi fattori che erano risultati significativi nel gruppo con cancer history, abbiamo effettuato una mappa di correlazione. Voglio evidenziare con tre colori diversi che sono poi nei riquadri che noi abbiamo ritrovato in questo gruppo con cancer history una correlazione tra fattori legati al metabolismo e fattori legati alle citochine. In particolare, per esempio, nel gruppo evidenziato in arancione abbiamo che livelli più alti di lattato e 2-idrossibutirato sono correlati a livelli più alti di trigliceridi, di fosfolipidi e a livelli più alti della chemochina MIF. Per porre l'attenzione anche sulla possibile associazione tra questi metaboliti, citochine e lipidi con gli stili di vita, abbiamo eseguito dei modelli di regressione logistica nell'idea di andare a verificare se essi potessero essere associati agli stili di vita. Per non riportare tutte le tabelle con i vari numeri, ho riassunto i risultati in questi due diagrammi di Venn, uno specifico dello smoking e uno specifico del BMI, dove vediamo quali sono i metaboliti nei due specifici gruppi che sono risultati associati allo smoking e al BMI. In particolare voglio porre l'attenzione sui cerchi blu, dove abbiamo proprio quei metaboliti, quei fattori che sono specifici del gruppo con cancer history che sono risultati correlati allo smoking e al BMI, e in particolare porre l'attenzione su quei fattori che sono evidenziati da riquadro giallo e che noi avevamo ritrovato anche nelle 3 piccole signature che avevamo ritrovato. Quindi il messaggio e la conclusione alla quale siamo arrivati con questo studio è stato quello di riuscire a identificare delle signature che sono specifiche della cancer history in questi portatori e che sono correlate anche allo stile di vita, nell'idea di poter poi andare ad utilizzare questi fattori come predittivi dello sviluppo di cancro in questi tipologie di pazienti. Quindi voglio ringraziare alla fine della mia presentazione il dottore Budillon, che è il direttore scientifico del nostro istituto, con cui è nata tutta questa storia, la dottoressa Di Gennaro, che è il nostro attuale direttore della struttura di farmacologia sperimentale, Federica, Palmina, Carlo per tutto l'aiuto proprio manuale che mi hanno dato, la dottoressa D'Angelo, come ho detto prima, e tutti voi per l'attenzione.
Grazie, abbiamo tempo solo per una domanda. Ok, grazie. C'è una domanda? No. Non mi sembra. Grazie.
Ok, va bene. Allora passiamo subito alla prossima presentazione di Martina Dameri, che ci parlerà di nanopore sequencing. Il mio co-chair mi ricorda che avete 7 minuti per la presentazione e 3 per la discussione, quindi per favore.
Grazie, buongiorno a tutti. E ringrazio innanzitutto gli organizzatori per aver selezionato questo lavoro. Oggi vi presenterò alcuni risultati del trial clinico Renovate che abbiamo svolto presso l'ospedale Policlinico San Martino di Genova. E questo progetto è finanziato da AIRC ed è in collaborazione con l'Università di Genova. Allora, il trial clinico Renovate si colloca nell'ambito della diagnosi precoce non invasiva di carcinoma mammario. Come sappiamo, in caso di sospetto radiologico, la donna è sottoposta a biopsia mammaria, una procedura che è invasiva, spesso mal tollerata, costosa anche per il sistema sanitario nazionale, con lunghi tempi di refertazione. Di qui la necessità di individuare dei biomarcatori non invasivi altamente sensibili e accurati per porre una diagnosi precoce. Noi ci siamo concentrati principalmente sul DNA libero circolante metilato. Si tratta di un biomarcatore con un elevato potenziale. Come si può ritrovare da letteratura, l'analisi della metilazione è molto sensibile, robusta, dinamica, può essere rilevata con diversi approcci, in primis gli epic arrays ed Illumina e poi tutta una serie di metodi basati su NGS, tra cui la conversione a bisulfito, la conversione enzimatica, quindi un approccio metil-sec, e poi ulteriori approcci che abbiamo appunto implementato. Noi appunto ci siamo concentrati sul CF-MEDIPSIC e su Oxford Nanopore Technology. Allora, l'approccio CF-MEDIPSIC è un approccio appunto di NGS basato su metodo Illumina. Questo approccio presenta diversi vantaggi, tra cui il fatto che il materiale input richiesto è molto basso, sono necessari tra i 5 e i 10 nanogrammi di DNA circolante, che soprattutto nel setting precoce rappresenta un importante vantaggio. Esso consiste nell'andare a immunoprecipitare le isole CpG metilate con un anticorpo rivolto contro le 5-metilcitosine e consente parallelamente di analizzare il dato metilomico, ma anche un'analisi di low pass whole genome sequencing. Ci sono ovviamente anche degli svantaggi dati dal fatto che il protocollo di preparativa è piuttosto complesso e lungo e ci sono delle limitazioni legate al fatto che, essendo un approccio Illumina, le rizze di sequenziamento sono tendenzialmente corte. L'approccio Nanopore invece consiste nel sequenziamento della molecola nativa. La preparativa, quindi la library preparation, è molto più snella, di solito non viene utilizzata un'amplificazione, quindi si riducono tutta una serie di possibili bias, permette un'analisi a livello di singolo nucleotide e combinata di metilomica, insomma epigenomica in generale e genomica. Ovviamente vi sono anche qui degli svantaggi, dati per esempio dal fatto che il materiale input richiesto è molto abbondante, per cui sono necessari circa 20 nanogrammi di DNA circolante di partenza e i dati prodotti sono molto pesanti, cioè vengono prodotti tera di dati che quindi sono difficili da gestire a volte. Molto brevemente noi abbiamo applicato queste tecniche al trial Renovate. Si tratta di uno studio prospettico singolo centro svolto al Policlinico San Martino di Genova, il cui scopo principale è la diagnosi differenziale precoce di carcinoma mammario e tra carcinoma mammario e lesioni benigne. Abbiamo quindi reclutato pazienti che presentavano una lesione radiologica di tipo BRAZ 4 e 5. Nel caso di un riscontro di cancro erano tumori molto piccoli, di lesioni inferiori a 2 centimetri. Abbiamo quindi raccolto campioni di biopsia liquida, di sangue, di plasma e di urine. Abbiamo utilizzato i campioni derivati dalle provette PAX-Gene per fare un'analisi di CF-MEDIPSIC. Qua molto brevemente la casistica raccolta: abbiamo raccolto 366 pazienti che rispettavano appunto i criteri di inclusione. Da questi abbiamo derivato 64 pazienti con carcinomi mammari precoci e 115 pazienti con lesioni benigne. Abbiamo inoltre raccolto anche una casistica, sempre in questo ambito, di 100 pazienti, cioè 100 donne sane con mammografia negativa. Per quanto riguarda l'analisi di CF-MIDIP-SIC sullo studio Renovate, possiamo vedere che sulla sinistra ha appunto riportato la distribuzione delle RIZ di sequenziamento e possiamo vedere come questa distribuzione è perfettamente conforme con la morfologia del DNA circolante di partenza con il tipico picco a 167 paia di basi. Il coverage ottenuto è intorno al 2 per, in linea con quanto atteso, e sulla destra invece è presente un volcanoplot che ci mostra, sull'asse delle X log fold change, sull'asse delle Y la significatività, ci mostra come vi sia un generale trend di ipometilazione nel cancro, cosa che appunto è attesa con quanto ritrovato in letteratura, ma in generale questo ci dimostra che anche nel setting precoce la metodica è in grado di rilevare un segnale di metilazione. Successivamente abbiamo implementato l'approccio Oxford Nanopore, però per far questo siamo partiti da una casistica diversa, una casistica di carcinoma mammario avanzato, sulla quale era già stata svolta un'analisi di bisulfite sequencing. Come si può vedere nel pannello A abbiamo analizzato questo è un esempio di 2 campioni che sono stati analizzati sia con bisulfite sequencing che con nanopore, e possiamo vedere come sia la tumor fraction che in generale la morfologia del cariogramma sono assolutamente riproducibili e sono praticamente identici. Nel pannello B appunto il livello di correlazione nella tumor fraction espressa tramite le 2 metodiche è molto elevato. Nel pannello C possiamo vedere che lo stessa elevata correlazione si osserva anche nella frequenza delle isole CpG metilate stimate con le 2 metodiche. Fatto questo abbiamo per ora su un numero ristretto di campioni fatto un'analisi con nanopore sullo studio Renovate, e quindi anche in questo caso siamo riusciti a vedere che Oxford Nanopore è in grado di rilevare un segnale di metilazione in un setting precoce. Qui per esempio abbiamo visto che a livello del cromosoma 21 è presente una differenza nelle isole CpG tra le lesioni benigne in rosso e i tumori in azzurro. Quindi per concludere possiamo dire che si tratta di risultati preliminari, comunque promettenti, nel senso che entrambe le metodiche sono in grado di rilevare un segnale di metilazione nel setting precoce. Entrambi gli approcci ovviamente presentano dei pro e dei contro, sia dal punto di vista concettuale che dal punto di vista tecnico, metodologico e analitico. Come prospettive future l'analisi bioinformatica è ongoing, per cui non solo dovremo analizzare l'aspetto della metilazione ma anche la frammentomica e gli end motives. Sarà necessaria una comparazione formale tra le 2 metodiche, nonché una probabile poi validazione su coorte esterna e un downscaling magari su isole CpG metilate specifiche. Con questo concluso, ringrazio tutto il gruppo di laboratorio, il professor Zoppoli, il mio PI, e tutti voi per l'attenzione.
Grazie anche per essere rimasta nei tempi. Domande? Io comincerei a chiedere una cosa, però nanopore, ecco, il vantaggio è che fai veramente una multiomica, perché puoi fare come number variation, puoi vedere praticamente fragmentomica e metilazione in una singola analisi. Uno dei problemi però è non abbiamo mappe di metilazione in qualche modo standard, e l'altro è anche la mole di dati. Ci vuoi dare un po' di consigli? No?
La faccenda dei dati molto grandi è il principale problema che stiamo affrontando, perché come abbiamo visto prima nella presentazione del professor Pruneri, che ho letto 150 tera all'anno, una corsa di nanopore occupa fino a 2 tera, quindi effettivamente questo è uno dei principali limiti. Sicuramente è invece molto interessante non solo l'analisi di genomica metilomica, ma per esempio consente anche di analizzare marcatori non convenzionali, per esempio 6-metiladenina, 5-oxoguanidina, che altri approcci non hanno, come dire, implementato. È più una questione di gestire questi dati molto importanti. La corsa di sequenziamento comunque è anche piuttosto lunga, perché sono 72 ore, quindi non si discosta molto da un'illumina, ecco.
Altre domande? Abbiamo una domanda lì, ti pareva?
Scusate, se posso una domanda. Era una domanda tecnica più che altro. Quindi, avendo confrontato nell'early stage sia l'approccio CFMDIP e quello nanopore, qual è l'opinione su quale delle due tecnologie sia più sensibile o richiede un sequenziamento più profondo? Ad esempio Nanopore avete comunque fatto un sequenziamento 1-2 per o è servito di più?
Allora, con Nanopore varia molto sulla base di quanti campioni si pullano insieme. Per cui nel momento in cui vengono pullati per esempio una dozzina di campioni, il coverage rimane sull'1-2 per e come dire, il segnale viene rilevato ma un po' più basso. L'ultima slide che ho presentato dei campioni benigni e maligni era un campione per una flow cell, quindi vuol dire costo piuttosto elevato, e lì siamo anche un po' più alti rispetto al MEDIP, arriviamo anche a un 5 per di coverage. È tutto sì una questione di bilanciare, insomma, tipologia di analisi, costi e DNA circolante di partenza anche.
Ultima domanda, il bioinformatico.
Sì, mi collego sempre a quello che ha detto la collega. Quindi nella fase avanzata siete riusciti a pullare più campioni insieme in quanto la quantità di DNA di partenza era maggiore e migliore?
Allora, sì, allora è stato più, quella è una ragione pratica, però di base sì, il DNA circolante era maggiore, era più concentrato sicuramente.
Invece nell'early vi trovate in una fase più critica?
Ci troviamo in una fase un po' più critica perché il problema principale era capire se riuscivamo a vedere segnale, diciamo bioinformatico, nel setting precoce, e poi sì, anche dal fatto che 20 nanogrammi richiedono 4-6 ml di plasma, quindi vuol dire più di una provetta di sangue per paziente. Quindi...
L'ultima, quando prima hai accennato a volte amplifichiamo prima.
Sì.
Quindi anche su Nanopore avete inserito degli step?
No. In
Okay.
questo caso il protocollo è proprio da sequenziamento nativo, quindi non c'è amplificazione. Vengono legati i barcode, insomma, solita preparativa, non c'è amplificazione. Era solo per dire che altri protocolli invece prevedono anche l'amplificazione, partendo per esempio da DNA, da RNA, da cDNA, alcuni protocolli richiedono amplificazione, anche in quel caso.
Grazie, ci dobbiamo fermare qui. Grazie molte.
Grazie.
Bene. Allora, ora il dottor Del Bene ci parlerà della liquid biopsy nel glioblastoma.
Buongiorno a tutti. Sì, innanzitutto grazie appunto dell'opportunità di poter presentare il nostro lavoro. Il focus è quello delle vescicole extracellulari nel glioblastoma come strumento per biopsia liquida. Il razionale è chiaro a tutti: il glioblastoma è il tumore più frequente e più aggressivo del sistema nervoso centrale per via di 3 pilastri che sono l'invasività, l'eterogeneità intra e intertumorale e la capacità di modificarsi nel tempo per rispondere ai challenge. Il primo step per implementare una medicina di precisione, come tutti noi sappiamo, è quello di una biopsia liquida che possa permettere di studiare questo tumore e di colpire ogni paziente e ogni tumore in un momento specifico della malattia. Nella nostra visione le vescicole extracellulari sono il substrato preferibile perché permettono una lettura veramente multilivello a partire da livelli più bassi come la concentrazione e il size, per poi passare a livelli più approfonditi come la caratterizzazione del cargo. Il GBM ne rilascia una quantità smisurata che è tracciabile a livello interstiziale ma anche nei fluidi corporei e soprattutto appunto da esperimenti in vivo sappiamo che gran parte della concentrazione delle vescicole a livello plasmatico è proprio di origine tumorale. Il paziente glioblastoma presenta un enrichment della concentrazione a livello plasmatico se lo confrontiamo sia con controlli sani che con pazienti affetti da altri tumori del sistema nervoso centrale. In questo secondo ambito però questo confronto risente molto dell'eterogeneità legata alle metastasi cerebrali. Le metastasi cerebrali non sono una patologia di origine cerebrale appunto, ma una malattia sistemica con un carico sistemico che deve essere tenuto conto a livello proprio clinico nell'interpretazione di questi dati, perché è intuitivo che un tumore metastatico con un alto carico di malattia sistemica ha una produzione di vescicole che è superiore a quello di una localizzazione singola cerebrale. La concentrazione delle vescicole permette anche di seguire l'andamento nel tempo e la risposta alla terapia. Nel post-operatorio c'è un crollo a livelli paragonabili a quello dei controlli sani e che poi è seguito da un re-enrichment durante l'evoluzione di malattia fino a livelli paragonabili alla prima diagnosi nel momento della recidiva. Il secondo livello di lettura è quello del sites. I pazienti di glioblastoma hanno delle vescicole che mediamente sono più grosse rispetto a quelle di controlli sani e di pazienti affetti da altri tumori del cervello, e questo dato rimane anche nel confronto diretto tra glioblastoma e brain metastasis. In quest'ambito specifico questo livello di informazione si può aggiungere a quello della concentrazione, aumentando quindi l'accuratezza dell'interpretazione della biopsia liquida. Come nel caso della concentrazione, dopo l'intervento le vescicole ritornano ad avere dei sites paragonabili a quelli dei controlli sani, per poi riavere un riallargamento a livello della recidiva. Passando al cargo, il problema, noi abbiamo affrontato più o meno i tipi di cargo, diciamo gli acidi nucleici e le proteine. Il problema del cargo è che risente tantissimo della diluizione dell'informazione, sia per quanto riguarda il DNA, sia per quanto riguarda l'RNA. Non è informativo all'atto pratico ad oggi nelle nostre mani, perché l'informazione, soprattutto per quelle varianti poco espresse, si diluisce nel mare magnum di vescicole che derivano da tutto il resto dei processi biologici. Quindi sia per il DNA sia per l'RNA meno, perché comunque è più frequente, noi possiamo perdere delle varianti, soprattutto in quelle forme appunto poco espresse. Discorso differente invece per quanto riguarda le proteine. In collaborazione col gruppo della professoressa Bonaldi abbiamo isolato circa 2000 proteine dai nostri campioni, e prevalentemente legate ad adesione, a secrezione e a EV, con grosso overlap con i database pubblicati riguardo alle proteine nelle vescicole, ma l'aspetto più interessante, andando a vedere quelle disregolate nei glioblastoma verso controlli sani, sono quelle pathway che avevamo già identificato in passato, che sono legate a complemento, coagulazione, e che avevamo già identificato come signature. Da qui siamo passati a cercare di identificare una signature che fosse di superficie, che fosse presente nel preoperatorio e poi non presente nel post-operatorio, e che potesse permettere, che fosse arricchita a livello del tessuto tumorale nei confronti del sano. Qui si può vedere appunto l'analisi di massa, che poi abbiamo validato per flow cytometry, per ELISA, e nei grafici in basso i confronti in silico con il database del TCGA. L'aspetto interessante di questa signature è che da una parte ha un valore prognostico, i pazienti che la esprimono vanno meglio, questo sia nel TCGA sia nella nostra corte, ma soprattutto potenzialmente può permettere di isolare le vescicole di origine tumorale e quindi di focalizzare lo studio su queste, quindi superando quel problema che è la diluizione. Quindi concludendo, la concentrazione sites delle vescicole di per sé funzionano come biomarker, sono reliable e possono permettere sia la diagnosi sia il follow-up del paziente. Dall'altra parte il cargo, come mostrato, è specifico per il glioblastoma e ha una signature proteica, signature che potrebbe essere utilizzata per isolare le vescicole, per focalizzare lo studio su di queste, superando quindi quel problema che è la diluizione dell'informazione. Grazie.
Grazie. Ci sono domande?
Grazie per la interessante presentazione. Uno dei problemi principali quando si estraggono vescicole dal plasma è la presenza di contaminanti lipoproteici che hanno densità e o dimensioni assolutamente paragonabili a quelle delle vescicole. Voi quindi quali protocolli di isolamento avete adottato nel vostro studio? Grazie.
Siamo partiti come tutti dalle ultracentrifugazioni differenziali per poi arrivare alla ACC e la cromatografia e il TRPS per la quantifica, che in effetti è molto più specifico del nanosite, proprio nella caratterizzazione della vescicola.
Sì, dati interessanti, poi anch'io penso che il cargo gioca un ruolo fondamentale. Ti volevo chiedere, prima domanda, se poi rispetto al cargo avete distinto anche per sites, perché ovviamente l'extracellular vesicle, come tu hai presentato, è una popolazione molto eterogenea, con origine anche diversa, e il cargo può variare. Noi per esempio stiamo studiando la differenza di cargo tra le large vesicles e gli esosomi che hanno, diciamo, anche un altro significato. Allora questa è la prima domanda. E poi, poiché nel glioblastoma sono stati descritti queste larghissime vesicles denominate oncosomi che erano state già isolate nella prostata, se voi avete fatto un focus anche su queste che poi per sites sono più facilmente isolate.
Diciamo che è un ottimo punto in effetti. Noi qua ci siamo rivolti alla popolazione bulk di tutte le vescicole, quindi senza un focus specifico. Vero è che i large oncosome in letteratura sta uscendo moltissimo su questo, ed è vero potenzialmente. Di per sé ci sono anche altre esperienze su invece vescicole molto più piccole. È un discorso che noi non abbiamo ancora adressato in quel senso. Il nostro focus è su identificare qualcosa che possa identificare le vescicole tumorali, proprio per superare la tumor fraction, come superare. L'idea è quella.
Perfetto, grazie. Passiamo alla prossima presentazione, abbiamo quasi recuperato. Abbiamo Giorgia Marisi che ci parlerà invece della profilazione molecolare del DNA circolante estratto dalla bile. Giorgia.
Buongiorno a tutti, è un piacere essere qui oggi con voi per presentarvi e condividere con voi i primi risultati preliminari dello studio Bile Biopsy, un progetto che si inserisce nel filone di ricerche sulla biopsia liquida nei tumori epatobilio-pancreatici. Come sappiamo, il carcinoma duttale pancreatico del pancreas e il colangiocarcinoma sono due tra le neoplasie più maligne e aggressive e con prognosi infausta. E nonostante i progressi scientifici, la sopravvivenza a 10 anni di queste patologie è circa il 3%. La diagnosi precoce è complessa, pochi pazienti vanno incontro a chirurgia e le chemioterapie ad oggi disponibili mancano di bersagli molecolari precisi. Quindi in questo contesto l'identificazione di biomarcatori precoci e un approfondimento molecolare dei meccanismi alla base di questa patologia sono fondamentali per migliorare l'approccio clinico di questi pazienti. In questo contesto la biopsia liquida, in particolare con l'analisi del DNA tumorale circolante, offre grandi possibilità. Se finora il plasma è stata la fonte maggiormente studiata nel campo della biopsia liquida, la bile stessa nei tumori biliopancreatici potrebbe rappresentare un campione minimamente invasivo e facilmente ottenibile mediante un drenaggio biliare. Con questo razionale nasce lo studio Bile Biopsy, uno studio prospettico, multicentrico, finanziato dal PNRR e coordinato dall'unità di gastroenterologia ed endoscopia digestiva dell'unità di Forlì-Cesena diretta dal dottor Carlo Fabbri, con la collaborazione del nostro istituto l'IRST, il Policlinico di Palermo e l'azienda ospedaliera di Cagliari. L'obiettivo primario di questo studio è la profilazione genomica completa del DNA circolante biliare in confronto con l'analisi del DNA circolante da plasma e il tessuto bioptico. La finalità di questo studio è appunto dimostrare il valore, la rilevanza clinica di questa fonte alternativa di biopsia liquida con ricadute sulla stadiazione, diagnosi, prognosi e scelte terapeutiche. L'obiettivo secondario dello studio è eseguire anche un'analisi del microbioma biliare e intestinale al fine di studiare la biologia come impatta sulla biologia del tumore. Al momento sono stati arruolati 70 pazienti che sono andati incontro a drenaggio biliare. Al momento del drenaggio vengono raccolti quindi campioni biliari e plasmatici il DNA circolante sia da plasma che da bile è stato estratto da 2 ml mediante kit Qiagen, è stato quantificato col Cubitat High Sensitivity e analizzato mediante Agilent Bioanalyzer, e la profilazione genomica completa è stata eseguita mediante il kit TrueSight Oncology 500 ctDNA versione 2 di Illumina su piattaforma NovaSeq 6000. Questo kit è un pannello ampio che permette la detezione, l'identificazione di varianti somatiche in 523 geni. Per la prima volta abbiamo ottimizzato e applicato questo kit al DNA circolante biliare, confrontandolo direttamente con i risultati ottenuti da plasma. Dal confronto diretto abbiamo notato che la quantità di DNA estratta dal campione biliare risulta 16 volte più alto circa confrontato con la quantità estratta dal campione di plasma. E come si può vedere dai profili al bioanalyzer, il DNA biliare risulta arricchito in frammenti di maggiori dimensioni. La profilazione genomica completa è stata eseguita su campioni biliari plasmatici in 15 pazienti, al momento 13 PDAC e 2 colangiocarcinomi, e nei tumori pancreatici il gene più frequentemente mutato è il KRAS, che si evidenzia una chiara distinzione tra il campione biliare e il campione plasmatico. In particolare abbiamo evidenziato 10 varianti in 10 pazienti sul DNA biliare, di cui 6 non rilevabili nel campione plasmatico. Le varianti riscontrate sul KRAS erano la G12V, la G12D e la Q61H. Non solo, la VAF e la variante, quindi la frequenza allelica delle alterazioni di KRAS, risultava significativamente più alta nel campione biliare rispetto al campione plasmatico. Interessante anche la distinzione clinica. Nei pazienti metastatici abbiamo notato concordanza tra l'identificazione delle varianti di KRAS nel plasma e nella bile, mentre nei pazienti localmente avanzati o resecabili le alterazioni di KRAS venivano rilevate quasi esclusivamente nel campione biliare. Nel DNA biliare altre alterazioni sono state identificate come mutazioni di P53, SMAD4, CDKN2A, RID1A, spesso non rilevabili nel plasma. Queste alterazioni indicano come la bile può essere una fonte molto più ricca di informazioni sul profilo molecolare del tumore. Un altro esempio, P53, abbiamo riscontrato alterazioni in 9 campioni biliari e solo in 5 campioni plasmatici, sempre con una VAF significativamente più alta. Per concludere, i nostri risultati preliminari mostrano che il DNA biliare sembra essere più rappresentativo e abbondante di quello plasmatico, è in grado di rilevare alterazioni spesso invisibili nel plasma e potrebbe essere interessante nei pazienti non metastatici in quanto riesce a detectare varianti in un setting più precoce. Quindi crediamo che la bile potrebbe essere una nuova fonte di biopsia liquida nei tumori biliopancreatici. Vi ringrazio per l'attenzione, ringrazio il mio gruppo di ricerca dell'IRST, il gruppo di gastroenterologia ed endoscopia e di Forlì e Cesena e tutti voi per l'attenzione.
Domande?
Complimento perché è molto interessante, perché questo è uno dei problemi maggiori che abbiamo sugli studi di tumori delle vie biliari, dove molte volte sul tessuto abbiamo grosse difficoltà tecniche in particolare. I quesiti sono due. Il primo è 90 pazienti, è una bella casistica, voglio dire, sicuramente una casistica consistente. C'è una relazione tra positività nel test, sangue, vie biliari, rispetto al carico tumorale, non soltanto rispetto alla malattia localizzata e avanzata e con quali stadi avanzate, e se c'è una differenza sulle alterazioni molecolari che sono individuate nei due setting, cioè setting tessuto, setting bile e carico di malattia, perché a noi questo è un dato veramente rilevante per la pratica clinica.
Allora, al momento le analisi sono in corso. Per questi 15 pazienti in cui abbiamo già fatto la profilazione genomica abbiamo analizzato anche il tessuto. C'è concordanza col tessuto, a volte sì, a volte no, ma sono in corso, abbiamo completato tutte le analisi delle library, è in corso l'analisi bioinformatica, quindi sarà molto interessante approfondire. Grazie per l'osservazione, ma al momento non abbiamo dati sul carico mutazionale.
Abbiamo provato a estrarre anche l'RNA dalla bile?
No.
Perché poi sono il problema delle fusioni. Che
sì.
Sicuramente i kit su DNA circolante le vedono, ma non le vedono in maniera efficiente, quindi questa potrebbe essere la prossima frontiera, soprattutto per i vie biliari.
30 secondi, ci sono problematiche tecniche nella bile rispetto al sangue?
Ma abbiamo dovuto ottimizzare, per questo kit molto ampio, non abbiamo provato pannelli più piccoli, abbiamo dovuto ottimizzarlo, cioè l'abbiamo considerato un po' come un tessuto paraffinato, l'abbiamo dovuto frammentare col Covaris, mentre il DNA plasmatico è già corto, quindi abbiamo dovuto fare delle accortezze, però è facilmente, insomma, l'analisi è facilmente eseguibile.
Perfetto, se non ci sono altre domande direi di passare alla prossima presentazione, grazie. E abbiamo Milena Orbini che ci parla di nuovo di metilazione su DNA tumorale circolante.
Buongiorno a tutti, ringrazio per questa opportunità di poter presentare i nostri risultati sull'analisi del profilo di metilazione target utilizzato sul plasma di pazienti oncologici per rilevare il DNA tumorale circolante. E come abbiamo visto, la liquid biopsy ha rivoluzionato il metodo di ricercare biomarcatori nel cancro in modo non invasivo e soprattutto l'analisi del DNA circolante è uno dei biomarcatori più utilizzato perché è in grado appunto di associarsi con l'outcome clinico, con la diagnosi, per l'early detection, per la sorveglianza del paziente. Però è ancora in discussione quale sia il metodo migliore per la rilevazione del DNA circolante al fine di ottimizzare il bilanciamento tra i costi e sensibilità del metodo. La metilazione si è visto avere una potenzialità di maggior sensibilità rispetto all'analisi mutazionale, però generalmente approcci di metilazione di profilo whole genome sono costosi e possono richiedere comunque dei laboratori ben preparati e attrezzati, quindi non facilmente utilizzabili da chiunque. Noi abbiamo testato questo metodo di metilazione target fornito da Agilent Technologies, che utilizza un pannello di 3481 DMR. Sono state disegnate dalla ditta in modo che siano pan-cancer, quindi specifiche e in grado di rilevare il DNA tumorale circolante da diverse tipologie di cancro. Noi l'abbiamo, inoltre la facilità anche di questo assay deriva dal fatto che può partire da un input di DNA molto basso, fino a 3 nanogrammi, e la parte di analisi è fornito un software cloud che permette di fare l'allineamento, fare il conteggio della metilazione, delle CpG e quindi stimare un methylation index score che rappresenta il carico del DNA tumorale circolante presente nel campione. Noi abbiamo studiato una casistica ampia, circa 200 campioni di plasma appartenenti a 3 tipologie di cancro che rilasciano poche quantità di DNA tumorale circolante, quindi campioni di pancreas, di gastrico localmente avanzato e di mieloma multiplo. Il nostro obiettivo quindi era valutare questo assay, vedere quanto la sua sensibilità nel rilevare il DNA tumorale circolante e la sua utilità per monitorare la malattia durante il trattamento. Il kit a livello di qualità ha performato bene, noi l'abbiamo utilizzato sia sul plasma che su alcuni campioni di pancreas di tessuto, ha lavorato bene su entrambe le matrici. Quello che abbiamo visto è che l'input influenza il numero di citosine metilate che si riescono ad analizzare, il numero quindi di regioni di EMR che sono analizzabili poi per l'analisi, però il methylation index score che viene fornito non ha influenzato dall'input di materiale utilizzato. Inoltre abbiamo visto che se si analizza il profilo globale delle DMR analizzate è possibile stratificare i campioni in base al campione che è stato analizzato, quindi riusciamo a separare in PCA il tessuto tumorale da quello adiacente e i campioni di plasma che sono qua su in alto e sono distribuiti lungo la prima componente principale in base al tumor content, quindi al methylation index calcolato dal software. Però non solamente riusciamo a distinguere il methylation index, riusciamo anche a distinguere dei profili di DMR, di metilazione, di regioni specifiche in base al tipo di patologia che stiamo analizzando, quindi siamo riusciti a identificare delle DMR che distinguono i tumori solidi che abbiamo analizzato, il gastrico e il pancreas, da quelli ematologici. Per entrare brevemente nel pancreas, l'analisi è stata più difficile perché il pancreas rilasciava estremamente poco DNA circolante. Inoltre avevamo una casistica di pazienti il cui plasma era stato raccolto pre e post chirurgia in alcuni pazienti che avevano fatto chirurgia solamente o in pazienti che hanno fatto neoadiuvante. Quello che vediamo è che la chirurgia non ha avuto effetto sul quantitativo di DNA tumorale circolante, mentre un effetto si riesce a vedere tra i pazienti che hanno fatto neoadiuvante o no. Per quel che riguarda il gastrico, Qui avevamo pazienti trattati con neoadiuvante, il campione era stato raccolto pre e post neoadiuvante. Anche qui vediamo che la neoadiuvante ha un effetto significativo abbassando il tumor content, il methylation index, e tra l'altro questi pazienti li avevamo testati anche con un pannello di 500 geni mutazionali e vediamo una buona concordanza con il dato di methylation index calcolato dalla 6 di metilazione. E inoltre, dato clinico importante, si è visto che i pazienti che avevano un calo significativo dopo la neoadiuvante sono anche quelli che poi hanno risposto al trattamento. Viceversa, in quelli che non avevano risposto, questo calo non era così significativo. Ancora, nel mieloma multiplo, noi abbiamo visto come il profilo delle metilazioni di questo pannello è in grado di distinguere non solo il methylation index, ma quindi lo stadio della malattia, distinguendo le mieloma multipli smoldering, che sono quindi le early diagnosi, da quelle new diagnosi che vengono posizionate a livello intermedio e quelli dei stadi più avanzati di relapse. E quindi riuscivamo a stratificare i pazienti in base al methylation index. Infine, il dato interessante è stato che, visto che avevamo identificato delle DMR specifiche per il mieloma, se noi ci andiamo a costruire un methylation index basato su queste DMR specifiche per il mieloma, riusciamo a aumentare la sensibilità dell'assay e riuscire a discriminare meglio gli smoldering, quindi gli early stage, rispetto ai pazienti a donatori sani di controllo. Quindi, per concludere, questo assay sembra robusto, facilmente implementabile, ti calcola un methylation index che correla con il contenuto tumorale e quindi anche sullo stato della malattia, ed è possibile, bisogna ragionare su cercare di fare dei pannelli o dei sottopannelli, insomma, per riuscire ad aumentare la sensibilità dell'assay. Sembra possibile e va controllato e validato su casistiche più ampie. Vi ringrazio per l'attenzione, ringrazio i miei collaboratori e qualche spunto per degli special issue. Grazie.
Bene, grazie. Domande? Bene, al di là però del methylation index che può servire per individuare o non individuare DNA circolante, in realtà la cosa interessante è studiare i pattern di metilazione. Ci sono modifiche, per esempio, tra pazienti early, avanzati, prima e dopo terapia neoadiuvante, perché insomma questo ci potete dare qualche insight anche dal punto di vista di approccio terapeutico.
Dobbiamo ancora approfondire questo dato, sicuramente abbiamo appunto intenzione di andare a confrontare sia che cosa si modula tra i diversi stadi di malattia nel mieloma, ma anche appunto pre e post trattamento nel pancreas e nel gastrico, anche se ammetto che qualche limite questo assay ce l'ha, perché essendo disegnato soprattutto per rilevare la presenza di DNA tumorale, al momento la più grossa differenza che si viene a vedere è quella appunto dello spegnimento in generale della metilazione di questo pannello, però l'approfondiremo.
Perfetto, grazie. Se non ci sono altre domande io lascerei a Stefania, che è stata davvero stoica,
chiedo
stava
scusa,
molto
ma non riesco
bene,
nemmeno
è
a
stata
parlare.
bravissima.
No, fai tu.
Va bene, allora grazie a tutti, adesso ci siamo guadagnati il pranzo, siamo riusciti a rimanere quasi in orario, quindi grazie agli speaker e a tutti quelli che sono rimasti. Ancora buon proseguimento.
Arrivederci.
This is my declaration of interest. So my lab, as the introducer said, studies cancer evolution. And we have two main lines of investigation. One is understanding tumor initiation already from pre-cancer conditions. And the second, which I will be talking about today, is how the interaction of cancer cells with the surrounding environment impacts on their evolution and how this is important for response to therapy. And this is exactly what is represented here in a very simplistic way. So all the mechanisms exerted by the tumor cells to evade the immune pressures actually have an impact on how the tumor evolves. And I will be focusing today, among the many ways this coevolution occurs, on the expression by the cancer cells of receptors that can bind with molecules on the T cells, blocking their ability to attack the tumor. As you know, this is a way of quite successful way of treating tumors, at least some tumors, and it has a lot to do with genetics of the tumor, because the tumor mutational burden, so the load of somatic mutations acquired by the tumors, is one of the first tumor agnostic biomarker for eligibility to immunotherapy. And the very logical explanation of this is that the more mutations the tumor has, the more likely is that neoantigens will be produced and presented for the attacking to the immune system. So, but as we will see, as almost everything in biology, very simple, and in life, I would say, very simple explanations do not really work. So, tumor mutational burden is also quite relevant in colorectal cancer because it divides colorectal cancer biology into two main subtypes: mismatch repair proficient, which are the majority of colorectal cancers. They are also known as MSS stable or non-hypermutated. They have a low tumor mutational burden and in general a low immune infiltration. And then there is between 5 to 15, according to the different reports, of colorectal cancer, which are instead mismatch repair deficient. They have a high tumor mutational burden and in general, high immune infiltration. So what happened in 2020 was that thanks to a big phase 3 clinical trial, KEYNOTE-117, TMB, so immune checkpoint inhibition, anti-PD-1 immune checkpoint inhibition, has been approved as first-line treatment for mismatch repair deficient colorectal cancer in the metastatic setting. However, only the response is variable, and only around 15% of patients will have durable benefit from this treatment. So we were lucky enough because we could access to the samples of the KEYNOTE-177 trial, and we, back in 2021, performed a multi-omic analysis of the tumor genetics and of the surrounding microenvironment to understand why some patients will respond and some other will not. So the first surprise was that within mismatch repair deficient tumor, there is no association between tumor mutational burden and response. And there is no, and probably the cause is that it's not true that more mutations a tumor has, more CD3 infiltration it will have. So there is no direct correlation. What we found instead was that the proportion of predicted possible neoantigen coming from all these mutations, which were clonal, so present in the majority, if not all, the cancer cells that will present the same possible neoantigen, was associated with a higher response rate. And this coupled quite nicely with the fact that the clonality of T-cell receptors in responders was higher. So probably there was the expansion of a neoantigen that was then recognized by the same T cells. Now this has been seen in many cancer types, and is recognized that probably the emphasis on TMB being a biomarker of response was a little bit too risky. So then the question was, what is then predictive of response to immunotherapy, at least in colorectal cancer? We ran an imaging mass cytometry screen to define the composition of the tumor microenvironment, at least of the main population of immune cells. And at the time, there were no software for the analysis of this data, so we developed our software, which is open source and is not only applicable to imaging mass cytometry, but to all imaging-based technologies. So if you want to give it a try with your own data, you are free to use it. So what we saw was that within mismatch repair deficient tumor, there were particularly two populations of CD8 T cells, one that was mostly cytotoxic, and the other that was mostly proliferative, so Ki67 positive, that would express PD-1 and were higher in mismatch repair deficient tumor. In the responders, then we also found a subpopulation of T-cell activating antigen, class 2 antigen presenting molecule, macrophages that will express PD-L1, which were significantly higher in responders. And so we wonder whether these two populations of immune cells were interacting with each other, which we proved by multiplex immunofluorescence. So basically, it's the microenvironment around the tumor that seems to be relevant for response to immunotherapy, at least in colorectal cancer. But then the question was, how is the tumor immune microenvironment determined? So what determined the composition? To address, or at least to start to address this question, Amelia and Kalum in the lab performed multi-regional, meaning at the invasive margin and at the core of the tumor, laser capture microdissection of both colorectal cancer that were deficient and proficient. And the aim was to separate the stroma from the tumor. Not a single cell resolution, but almost a single cell resolution. So what we saw was that as predicted or as expected, the majority of immune cells were higher in mismatch repair deficient than in mismatch repair proficient tumor, and there was a high positive correlation between the level of infiltration of all this population, meaning that a immune hot tumor will be hot for almost all immune population. There is co-infiltration. But what was unexpected, because, as I said, Amelia performed laser capture microdissection, was that in the epithelial compartment, we still could see particularly 3 populations of immune cells that were higher in mismatch repair deficient tumor than proficient. And these were the intraepithelial immune infiltrates that were impossible to separate via laser capture microdissection. Now, this is known for cytotoxic T cells, but our pathologists, at least, needed to be convinced by staining that also NK cells and a class of inflammatory macrophages could be highly found in the intraepithelial compartment. So the presence of this population also triggered a response in the tumor, particularly high interferon alpha and gamma response and a high activation of the STING pathway. So what then we did was taking the signature, the transcriptional signatures of these immune populations and their response in the tumor, and cluster all our samples irrespective of their genetics. And we found 2 main clusters that would differ for the expression of interferon gamma, and therefore we call these 2 clusters interferon high and interferon low immunophenotype. These 2 clusters are composed of mismatch repair proficient and deficient. Of course, as expected, the interferon high was enriched in mismatch repair deficient tumor, and the interferon low was enriched in mismatch repair proficient tumor, but both genetics were present in the two immunophenotype. And in fact, they didn't differ for tumor mutational burden. However, the mismatch repair, the interferon high immunophenotype was enriched in responders to immunotherapy, suggesting at the point that what we call immunophenotype, so the composition of these tumor microenvironment and the response of the tumor could identify responders. At this point, we wanted to investigate more the interaction between immune cells and cancer cells, particularly the intraepithelial population that we were interested in. So we did cosmic single-cell spatial transcriptomics. And here I show the antibodies that we used to complement single-cell segmentation. And then really to study the crosstalk between a cancer type, Pietro, a very good PhD student in our group, developed a computational approach to investigate cell neighborhood, and in particular to group cells within the cancer ecosystem according to who is around them, which is very, very powerful, because then what we could do was to divide, for example, macrophages that are near to T cell from all the other macrophages, T cells that are near to cancer cell to all other cancer cells, and cancer cell next to T cell to all other cancer cells. And of course, you can do this type of game with any cell type that you are interested in. And why this is interesting? Because you can then do differential gene expression between, for example, T cell near to a cancer cell as compared to the rest of T cells. So what is the cancer cell modifying at the transcriptional level, at the phenotype level to T cells? What we saw for T cells was that the T cells near cancer cell had a cytotoxic and exhausted phenotype, but what was probably more interesting was that both macrophages and T cells, and cancer cells near to T cells started to express antigen presentation machinery. And while this is not so surprising for TAMs, because they are professional presenter, it was quite surprising for epithelial cells, because epithelial cells are not antigen presenting professional cells, but they still can respond to an interferon high environment induced by the T cells. So this was all quite nice, and I should already point out that both epithelial cells and TAMs would express CD74, which is a chaperone involved in, as I told before, class 2 antigen presentation. That was the same molecule that we also tested in our previous study, and so probably meaning that these are the populations that we are identifying. So this was all quite exciting, at least for us, but it was all computational. And being a computational biologist myself, I know that you need to validate your data with an orthogonal approach. So what Emma did was to initiate co-cultures of epithelial cells with activated T cells or adding to the media interferon gamma, and then check whether epithelial cells would start to express antigen presentation. This is only qPCR, but we also did RNA-seq, just to say that indeed in two different colorectal cancer cell lines, CD74 was induced by the presence of activated T cells, also by interferon gamma, and when we use an interferon gamma inhibitor, although not always, but at least in one cell line, it was reduced. So it is possibly dependent by interferon gamma, not only by interferon gamma, probably by something else, but it is something that is definitely stimulated by activated T cells. Then what we are doing now is to move from co-culture, or not to move, but to complement co-culture with the manipulation of patient-derived tumor explants, because they will have the entire ecosystem, or almost the entire ecosystem. I mean, some population will die, but still it's a more complete representation. So Amelia started to establish patient-derived tumor explant in the lab. They have similar immune composition that the original tumor, and she treated these tumor explants with interferon gamma. And what we saw was that there was indeed an increase of epithelial cells that would express CD74. So the tumor explant treated with interferon gamma will start to change their immunophenotype versus at least antigen presentation. We don't know what will be the role of this antigen presentation, if they need it or if it's just a response to interferon gamma, but this is quite an interesting question to address. So back to why this was relevant for, or can be relevant for, response to immunotherapy. So as I told you, the interferon high immunophenotype is enriched for patients who respond to immunotherapy. So if this immunophenotype would be enriched in CD74, so would be highly expressing CD74, this could mean that CD74 is a marker for interferon high immunophenotype and therefore could be used to predict response. At the transcriptional level, this is what we found. So CD74 is more expressed in interferon high tumor of both mismatch repair deficient and proficient subtype. And therefore, we went on to develop a score, which is very similar to the scores used in clinic for PD-L1, which, by the way, doesn't work in colorectal cancer, so cannot be used in colorectal cancer. So we developed a score that measures from an immunohistochemistry staining the proportion of stromal CD74 positive cells over all stroma cells. And both in our original cohort as well as in a validation cohort, it was predictive of response. More interesting than that would be to identify mismatch repair proficient colorectal cancer patients responding to immunotherapy, because immunotherapy is not given to mismatch repair proficient patients, which constitute 90% plus of all the colorectal cancer patient. So this was something quite difficult to prove because there are no patients treated with immune checkpoint inhibition. However, there are now a number of clinical trials that use combination of immunotherapy with targeted therapy or with chemotherapy. One of those was released while we were revising the paper, so we could actually check, at least at the transcriptional level, from geomics data whether CD74 was more expressed in responders than in non-responders, and it is, both in the stroma and the epithelium. But actually, most interestingly, is that the really great hub of clinical trials led by Chiara Cremolini at the University of Pisa, they are really fantastic for coordinating clinical trials in colorectal cancer and collecting samples. So they published recently a phase 2 trial for mismatch repair proficient tumors, testing an anti-PD-L1 agent with 2 other agents. It's a quite complex clinical trial, so I will not go into detail, but just for you to remember that there is an experimental arm where the immunotherapy is given and the control arm when the immunotherapy is not given. In general, these patients respond quite a lot, even without the immunotherapy. So these are not easy data to analyze. However, with the help of Chiara and her team, we could see that CD74 would be predictive of a longer patient-free survival only in the experimental arm and not in the control arm. So it is important that microenvironment only for response to immunotherapy. So this is then the summary of results, which Pietro actually nicely imagined would be similar to a picture from Kandinsky, where TMB marks the phenotype of mismatch repair deficient tumor, but not immune checkpoint response to immunotherapy. And this is because it seems that the composition of the tumor microenvironment is quite relevant for response to immunotherapy in colorectal cancer, and in particular the local microenvironment that needs to be composed by NK cells, CD8 T cells, and this population of inflammatory T cell activating macrophages. This tumor microenvironment can be recognized by the protein expression detected via immunohistochemistry of CD74. So my last slide is then what comes next. So still, we don't know what triggers this immunophenotype. If it's not the origin of the tumor, it must be something else, which we still don't know. How frequent is across other cancer types, and is also predictive of response to immunotherapy in other cancer types. Does it change during tumor evolution? So does an interferon high immunophenotype tumor preserve this immunophenotype throughout its evolution? And can we induce this immunophenotype or interfere with it to increase the patient response to immunotherapy? So we will be busy for a few more years. So, and with this, I thank the 3 people that dressed like this worked on this project, and Emma, who joined lately. This was really a collaboration with our pathologist of reference, Manuel, with all the clinicians who provided and identified the samples, in particular Chiara, who was instrumental for the mismatch repair proficient cohort, the fantastic facilities at the Creek, and all the funders for their money. And thank you very much for your attention.
Thanks, Francesca, for this wonderful talk. We have time for a few questions from the audience.
Thank you, Francesca. It was great. So, I mean, I have a question regarding the mutational status of this tumor, whether, regardless known mutations such as STAT1, JAK-STAT pathway, but if other mutation can, help to further, let's say, stratify population in order to check.
Yes, you would imagine so. As you can imagine, we did whole exome sequencing or targeted resequencing, and then measure the frequency of mutations, for example, in the CASTIG pathway, as well as a lot of pathways. We can't see any enrichment, so but at the transcriptional level, it is more active, which is probably a response more than a trigger.
Thank you. It's a very nice talk. I just was wondering, because in our system, when we do spatial transcriptomic analysis, it's a different model, it's CAR T cells. But when the CAR T cells goes in proximity to tumor cells, we see a number of interferon-regulated genes that are upregulated. So I was wondering whether the CD74 is the best one, or you search for others?
I mean, for us, it was good because it has very good antibodies. It stains very well. And it's clearly, you can clearly identify and distinguish it in the stroma and in the tumor. CD74 per se is not important for response to immunotherapy. It is a marker of the tumor microenvironment that is, or the ecosystem that is favoring response to immunotherapy. So, in my opinion, any other marker of this microenvironment, ecosystem, because it can be expressed also by cancer cells, will be equally good. However, for example, antigen presentation genes are not good markers. So it is a good compromise, but by any means is important per se, CD74.
If I may, along
Yes.
the same line, have you tried to use, instead of a single gene, gene panels, maybe through data-driven approach, using your spatial data?
So you mean for
classifying?
For classifying, not so far, and I think we are not possibly going that way, because ideally, if we are far from any clinical application of our assay. However, we are going hopefully in that direction. If it works, if it is confirmed to work, this is very simple, because any diagnostic lab can do immunostaining. And in principle, it's also very simple to measure. Now, as I said at the beginning, simplicity in biology almost never works, so there will be some problem along the way. Let's see.
Hi, Francesca, great talk. So just a quick question. Do you know if the interferon gamma signature that you reveal is induced in an antigen-specific context, like the T cells need to recognize the tumor antigen, or is antigen independent? So since the tumor burden is not like really accounting for
That's
this
a very good question. I suspect is antigen independent, because when we do co-cultures, these are completely, are cancer cell lines or organoids plus human-derived, donor-derived T cells. So they don't recognize each other. So I think and still we see upregulation of antigen presentation. So I think it's antigen independent, but I don't think we have the formal proof.
Thank you.
Thank you. Thank you very much. It's a pleasure for me to co-chair this session with Dr. Zahn. I have the duty to report you the greetings of our president, Maurizio D'Incalci, that I am substituting now, and I'm very happy that CKCC has prepared this joint session, whose next speaker is Dr. Giulio Caravagna from the University of Trieste, and please, Giulio. He will talk about clonal evolution using sequencing data.
Thank you very much. Thanks to the organizers for inviting me. Thank you for the name. Sorry. Yeah. Well, anyway, there was my disclaimer of conflict of interest, which is none, so it's okay. Thank you. So our group, I'm a professor of computer science, and we are a kind of a heterogeneous group of people that have background in mathematics, physics, computer science. We have biologists, and we have 2 clinicians. And we basically work thinking of tumors as ecosystem of populations that evolved over time. We try to do equations that describe the evolution of these processes, and we try to parameterize our models with sequencing data. The output of our work, besides trying to solve some interesting clinical questions, as I'll try to convince you today, are tools that you can take and run on your data. So whenever you see something that looks like this, it's basically one of the tools that we've used to do the thing I'm showing in the slides. The principle that we use is to think about tumors as evolutionary processes. And in this presentation, I'm going to show you 2 projects that we're working on. First one is a little bit more like standard biostatistics in some sense. So look for biomarkers. It might look less evolution in some sense. The second one is instead digging in the evolution of one particular patient. So in the first part, we look at low resolution sequencing data, classical targeted panels. In the second one, we look at high resolution whole genome sequencing data. But in the first part, we look at hundreds of thousands of patients, and in the second one, we look at a single individual. So We try to challenge a little bit the classical idea of mutations as biomarkers, in which one is interested in maybe distinguishing a stratifying group of people based on the presence of your particular mutation, your favorite mutation, the one you can target. That is like a starting point, we do believe, but it is obviously missing from the overall picture the effect of aneuploidy, which is the presence of somatic copy number alterations that might be mapping on top of those mutations, because every mutation is, of course, present in configurations that depend on the karyotype at a specific mutant locus. And that is something sometimes difficult to be extracted from targeted panels that are used commonly in the clinic, because they are missing basically a matched normal that we often use to pick up the signal for copy numbers. And in general, the two things don't talk, no? So you have a reporting of an allelic frequency of a mutation, you have reporting of a gain or loss, but you are missing an understanding of the exact relation between the two quantities. What I think it would be interesting instead to think in terms of gene mutant dosage, which is a quantity that can be defined by computing the ratio between the number of copies of a mutation over the total ploidy at the mutant locus. So something that looks very simple to be computed. And that if you could compute, and we can, could allow you to stratify your group of patients into people that have low gene mutant dosage compared to people that have high mutant dosage. Of course, think of this. This is a situation where you have a mutation, you have a copy gain, right? But basically you have a single copy of the mutant here, whereas here you have an amplification of the mutation. So if we're talking about an oncogene, for instance, this scenario might be much more oncogenic than this other one. So the idea would be to try to pick up this type of signal from low resolution data. We have developed a method that you can find on this medRxiv paper that hopefully will be out in Nature Genetics soon, that takes in input sequencing data. Basically, you just need to use the coverage that you have a particular mutant locus. You don't need to have anything like row sequencing, reads. You just need to take the number of reads, basically, that you have a particular position, and uses some type of mathematics which is called Bayesian inference to estimate a full distribution probability over all the possible values of M and K. So in some way, which I'm not discussing here, is capable of computing exactly, accounting for the uncertainty of the measurement, a distribution over the values of M and K that are the ones that give you this gene mutant dosage ratio. So you have a distribution over the gene mutant dosage ratio. This is something that is not difficult to do analytically, but is something that is difficult because of the data. So the sequencing data in a targeted panel is something that is not super easy to manage compared, for instance, to whole exome or whole genome assays. So in order to make the model stronger, because we were using these Bayesian statistics, we could encode prior biological knowledge in the process. So we have taken over 10,000 tumors from public cohorts, PCOG and Hartwig. Those are very famous cohorts of whole genome sequencing data where we know the values of M and K for these patients, and we could in some way, abusing a little bit the language, pre-train the model to use this information to get good estimates. So this is something that is very nice and allows you to go back to your data in your computers, even if you don't have raw sequencing data, and compute the gene mutant dosage for each one of the samples that you have already sequenced, even in the past. And this is something that also makes the model very fast. So in I would say, a couple of hours, we could analyze over 60,000 samples, recently released by two cohorts in the United States, the MSK impact and the Gini DFCI cohorts. They have really a lot of tumor types. Most of the samples are metastatic. And so we could use this to test whether this whether this stratification would be actually prognostic. We found positive results that I'm trying to summarize here and show you a few examples. On the left, you see a comparison of a classical mutant versus wild type stratification in the context of KRAS mutations in prostate cancer. And you will be seeing, obviously, that when we switch to simplifying this curve, the blue one, into the stratification, you will see that there is an obvious increased risk, so more prognostic power, when you see high mutant dosage for KRAS mutations, which is very important if you think about the fact that now we can target those. And this type of analysis resists obvious confounders like tumor mutational burden or the fraction of genome alters, so it looks something that is very interesting. There are even other type of scenarios, like scenarios in which, for instance, the classical mutant versus wild type analysis is not prognostic, but when you start introducing these classes, instead you see a certain kind of prognostic power in the notion of mutant dosage. The example here is PIK3CA mutation in the context of certain BRCA subtypes. And you'll be seeing, obviously, that high mutant dosage for BRCA is prognostic compared to wild type, whereas the simple analysis was not. In these cases, you also see something that looks very surprising, the potentially protective effect of low mutant dosage, like having few mutation copies would favor the prognosis, but of course this might be a limitation of the fact that we're looking at a single gene, and therefore what we call here wild type might have many other type of mutations. The beautiful thing is that we find, for instance, 13 different markers that the standard analysis would be completely neglecting. And of course, because we have the presence of metastasis, we can actually also measure the probability of metastasis as a function of gene mutant dosage and whether there is any association with specific metastatic sites based on particular classes of mutant dosage. And we find 24 situations in which the gene mutant dosage stratifies the tendencies of metastasis, and also we find specific patterns of metastasis which are enabled by specific configurations of gene mutant dosage. So there might be underlining transcriptomics, epigenomics explanation, but the key point might be that you could action this type of information based on the result of this type of assays. So these concludes the first part of the presentation, where I wanted to show you that thinking in some kind of, I would say, advanced machine learning terms is possible to encode biological processes in the way we reason about very simple data. But here I was working with tens of thousands of patients. Now I want to work with a single patient, but with high resolution data. And I want to answer a question that is very different from the one here. I want to make a prediction about the past of the disease. Let me clarify this with you. Imagine you have a classical patient that has been sampled twice, diagnosis and after a certain type of treatment, any treatment that you like, and you might have access to the genomics data, sequencing data for this patient. And imagine that you have basically your, whatever is your favorite algorithm, you can understand what are the somatic mutations, the copy number alterations, you know the clinical data, you know everything about this patient, right? And you have a clear understanding of what is the potential elephant in the room that drives the resistance here. You know, you have your favorite mutation, your favorite copy number, and you think this is the reason why this patient is relapsing to this treatment. A very simple question, which is simple to ask but difficult to answer, is understanding whether the presence of the relapsing clone, which is here written in green, is something that was born during exposure to any of these treatments, or you have reasons to believe that it was already present before you started the treatment. These are obviously two clinically distinct scenarios that might require, if you knew the answer, you would probably act in a different way, but these are difficult to understand because they require you to infer, to say, statistically decide where this green dot gets positioned, whether it's going to sit here or it's going to go before this diagnostic sample. I don't think it's possible to answer this question in every possible scenario. For instance, epigenetic, relapses, plasticity are very difficult to be managed. But I think we have found a smart way of dealing with this type of scenario in the context of strong genetic bottlenecks that define these green populations. We work in the context of hematological cancers, even if we have developed this method and tested it also in the context of two solid tumors, glioblastoma and neuroblastoma. But in the presentation here, I'm going to focus precisely in the context of leukemia, and it's a collaboration with the group of Luca Bago, which is an immunologist in San Raffaele. He provides us the sample and the clinical expertise, and Giovanni Tonon that let us sequence, because we decide the sequencing scenario ourselves, the experimental design, what kind of data. Before doing the equations, we decided we need data done in this, and that way, so we could do equations in a certain way. So these are interesting patients. These are 11 patients that we have sequenced. We have generated about 50 whole genome sequencing samples at very high coverage. We're talking about 100x reads per genome position. We have sequenced the recipient of the transplant, which is the therapy I'm going to talk about right now, and the donor. In this context, these patients have a particularly interesting clinical history. They start with the AML diagnosed, and they do usually several cycles of chemotherapy. Maybe some of you are clinicians that treat these patients and they know better than me. But the key point is that some of these patients will qualify for transplant and undergo a transplant which is based on the simple principle of triggering a graft versus leukemia response in the context of this disease. And I'm focusing on patients which are selected based on the HLA mismatch compared to the donor. So the idea is to mismatch the HLA of the recipient of the transplant, trigger a response, and hope that this, of course, is going to deplete the leukemic expansion. But these patients also get other type of treatment. Sometimes they relapse. About half of these immunological treatments fail. And of those that fail, 1/3 of them will show up with a strong copy number event, a strong genetic lesion, a strong bottleneck, which consists in reshuffling the HLA locus, losing the mismatched alleles, which is obviously the most simple way of evading this type of treatment. So we have looked at these patients, taking longitudinal data, this diagnostic, pre-transplant and post-relapse, let's say, relapse samples. We have looked for obvious evidences of somatic mutations that might explain these type of relapses. I remind you that we want to time the HLA copy neutral OH. And here you will be seeing for every patient the classical drivers of any AML that we've been seeing so far. I mean, nothing particularly special. Maybe your favorite driver is here. The key important information is that the set of driver mutations that we find in these patients are stable when we compare before and after transplant. It's like the leukemia has basically reached a sort of a peak of fitness in some sense in terms of somatic mutations and just need to have the extra hit to evade negative selection induced by the transplant. But essentially, the set of mutations are rather stable in most of these patients. And this is something that allows us to make some simple mathematical simplifications when we want to think about these processes. Because we have sequenced whole the genome, so we have basically each one nucleotide in the genome of these patients, we can look at the distribution of somatic mutations in these patients. So when we do that, this is an example of one of the patients, UPN06. You'll be seeing on the top row here, you see a sample collected before the transplant, and here you see a sample collected after the transplant. And obviously, it's not very difficult to notice that there are 4 sticks of C2A substitutions in those particular flanking bases. This is a classical plot from the Sanger where you see the notion of mutational signatures that are associated with the relapse sample. And we have taken our mathematical models that are able to use this type of signal from all the patients to extract the mutagenic processes. And besides obvious cosmic annotated mutagenic processes, we have found a bunch of other signatures, which are this one green, red, and 2 blue here. Some of them we don't know what they are. For instance, this. One of them we knew because that's a mutational signature of cytarabine, the type of chemotherapy that these patients get before qualifying for transplant. And this one is the interesting one because it's the one that is exactly matching these 4 sticks. And I'll tell you a little bit about what this is. We have asked ourselves what is the distribution of these mutational signatures throughout our sampling strategies. And of course, diagnostic samples are just plain leukemias. If you work in large-scale genomic studies, plain leukemias usually show up with these 2 clock-like mutational signatures, nothing very, very fancy. Certain samples before transplant, but after chemotherapy, they have indeed the signature that we associate with chemotherapy. But there is obviously this signature which is present prevalently at relapse samples. We dig in the clinical history of these patients and we realize that these patients, all of them, do one thing. So after transplant, to counteract the reactivations by cytomegalovirus, they're treated with an antiviral. This antiviral is called the ganciclovir. Maybe you know better than me. But this antiviral is mutagenic, and we didn't know about that. We actually discovered because we did an experiment cultivating cell line and comparing lines of cells exposed to this antiviral to the MSO, and we could replicate the presence of this signature in the lines of cells that had been exposed to the antiviral. We wanted to publish this a couple of years ago, but then there was another paper that published it before us. But so this gives us strong evidence that mutagenic process in vivo is linked to one particular type of treatment. And this is something that is very helpful for us because it allows us to make some computation, some mathematics, which I'm not discussing here, but essentially allows us to take a phylogenetic model for the patient and transform that into a set of molecular clocks. The idea is that for each one of your patients, because you've used whole genome sequencing data, you are capable of writing the evolutionary process as a very neat and clean phylogenetic model, where you have a branch of mutations that are associated with the relapse sample here. And as you notice here, this, as any phylogeny, this one has a length here that represents the number of mutations. And also you have mutagenic processes, so those mutational signatures mapped on top of the phylogenetic stick. And you can use this information to derive a set of clocks that basically inform you about the time t0, where you think there was the first event of HLA coupling neutral loss of heterozygosity. This is something that is kind of complicated, but the intuition is to merge multiple type of somatic events, multiple type of clocks that are explained by the presence of these mutations. You're going to have clock-like mutagenic processes happening consistently throughout cell division. You're going to have therapy-related mutations, like the one associated with ganciclovir. And you're going to have also structural arrangements around the HLA locus. That is what you want to time. So the mathematics of this is complicated, but the beauty of that is that it's easy to use. You take this, you put in our tool, and you take all the patients, and you get basically something like this. These are all our patients. They are aligned. 0 is when they have received the transplant. So anything that has, on the right of that is after transplant, on the left is before transplant. And what you see here, this kind of weird shape here, is exactly this probability distribution, is when the model thinks that the event has happened. So in this patient, for instance, UPN01, the transplant happened here, and about, I would say, 100 days after transplant, there was the event happened of losing the mismatched HLA allele. Certain scenarios are uncertain for the model. Sometimes the model thinks that the clone is pre-existing the transplant, but in most of the scenarios, the model thinks that this is largely de novo. So you would think that it's something that happens after the transplant. So this is interesting, but it's a prediction, and you would like to get some more information about that. So what you do, you look at the overlap between annotations of ganciclovir, which we know is mutagenic, and the time of origin estimated by the model. There is sometimes they're kind of very close in time. They're very, really, really close. Looks like they're almost overlapping in some of the scenarios. So that you are, of course, moved to think there might be some causal explanation linking this particular drug and the probability of this type of relapse. So we went back to our colonies and experiments, and we checked what does this mutagenic process do. So this mutagenic process do basically double-strand break inside these cells. Whenever the damage is integrated, when cells divide, they get this double-strand break. And this is something that is strongly obvious from a bunch of assays that we have done, and is something that is specific to ganciclovir. But the beautiful thing is that we discovered that this type of damage gets repaired by homologous recombination. We see that with immunofluorescence, we see that with bulk RNA sequencing, we see that with long reads and haplotype reconstruction. In a number of different ways, we come to the same conclusion. This damage gets repaired by creating 2 sister cells which have the opposite and equal homozygous HLA alleles. So basically, one of the 2 is completely evading immune control of the transplant. It's something that is systematic. So you find that there is an obvious evidence ganciclovir causing mutagenic creating a mutagenic processes, causing double-strand break, and eventually causing something to get repaired by homologous recombination. This does not happen necessarily on chromosome 6, where the HLA sits, but happens throughout all the genome. But the key point is that in this particular context of this mutagenic, immunological pressure, when you get that on the HLA locus, this gives you immune evasion. So the epidemiological question that comes naturally after all this is whether this type of event, giving ganciclovir to a patient, actually increases the general probability of relapse. And luckily that's, no is the answer. So in general, ganciclovir does not necessarily increase the probability of relapse to this type of treatment. But in the context of specific patients which undergo mismatched HLA transplant, which is called haploidentical transplant, ganciclovir increases by a fold 6 the probability of relapse. So in a particular cohort that we could assemble in San Raffaele, the validation of a cohort that we could assemble in China, and with the cohort we assembled in Spain, which are not shown here, basically we could see that in general, ganciclovir increases the probability of relapse by HLA loss in the context of haploidentical transplant. And of course, this, I think, concludes the evidence that we needed to suggest that another type of antiviral should be driven. I'm coming to the conclusion here that basically I tried to show to you something that might be a little bit exotic, the way that we think about these processes with these equations, which I didn't show, but you can believe me, are just underneath, allows us to make inferences about the clonal composition of the disease or something that has happened in the past, no? The time of origin of one particular expansion. The background of all these approaches is what we call model-based machine learning. So the fact that we think about the probabilities in terms of dynamical systems, some kind of equations that basically tell us if the state of the system is this, in the future is going to move in this direction, and in the past probably has moved in this direction. That's the way, no? We try to switch from a static cancer perspective to a dynamic temporal perspective. The tools that we develop can be used on your data. There might be some caveats, might not work on your designs, but we're happy to help you if you want to try them. And I think that you can see that this kind of bioinformatics is not just the standard bioinformatics, but it's the type of bioinformatics that is done to solve clinically relevant problems. So maybe this can help you in your daily research. Thanks.
Thank you, Giulio, for your beautiful talk. There are questions from the audience? No? I will start with one, and regarding gene dosage, a type of metastasis. Mm-hmm. If I understand well, you check different type of tumor and different type of metastatic sites. Mm-hmm. There are a relation independent of the tumor by which the mutation dosage.
Yeah, you ask if any mutant dosage for a gene leads always to the same type of metastasis. Yeah. I don't think we check it like that, but we have a huge table of predictions, and we could definitely, it would be easy to check. On the top of my head, I think I remember they are tumor specific, but that is something that we could check very easily.
Giulio, for what you call stability, so the conservation of drivers after relapse, do you know whether this is actually due to clonal hematopoiesis rather than a foundation? So what is the age of your patients? Did you check?
So the age is rather different. There's some which are, so there is, I think, as far as I understand, over a certain age it's difficult to qualify for transplant, so I would imagine some of these patients are rather young. But I don't have actually an estimate of the presence of CHIP in these cases. It might be something that might be worth checking. I agree with you, but they don't even not all of them have the classical CHIP mutations though. So maybe it's not something that would be. There were a few of them that did, but not all of them. So yeah, it might be something interesting. Thanks.
If there are no other questions, thank you, Giulio.
This will conclude this session. Thanks all the speakers again, and we move to the
next one.
Yeah, thank you.
I will stay here and invite Federica for the co-chair.
Gli diciamo che preferibilmente in inglese, chi proprio non capisce, dico io, dai.
Scusate, una domanda al volo, siccome io presenterò dopo e voi siete miei, e la presentazione sarà in italiano perché non
Sì, siamo in ritardo. Sì.
Okay. Welcome to the parallel session on clonal evolution and resistant to therapies. I'm Federica Di Nicolantonio from the University of Torino Candiolo Cancer Institute, and I'm delighted to invite our first speaker today, who is Andrea Sottoriva from Human Technopole, who is going to talk about measuring and predicting cancer evolution. Thank you.
All right. Thank you very much for the kind introduction, and thank you very much to the organizer for inviting me here today. All right, today I'm gonna tell you a bit about what we are doing in the lab. So we're a lab that is interested in measuring and predicting cancer evolution, and we have moved a lot from, we've done a lot of work on genomics, and now we are slowly moving towards epigenetic profiling of cancers, particularly colorectal cancer. So I have nothing to disclose apart from funding from different academic funding institutions. All right, so this is the typical plot that you see when people talk about cancer evolution. So people have known for a long time, more than 100 years, that tumors change over time. They transition from benign to malignant to metastatic, and finally they develop resistance to treatment. But it is only in the last maybe 20 years that really we have consolidated this idea, this paradigm, that the way they change, the rules by which tumors change over time are the same that regulate the change of species over long periods of time. So Darwinian evolution that you see on the right-hand side can also be used as a paradigm to describe the change and adaptation of cancers to different microenvironments and to treatment. And in my lab, my background is computational biology, we try to answer the question of, can we measure this process in humans, and can we predict it if we can measure it? Can we forecast the future course of the disease? So in evolutionary biology, this is philosophy. Can you predict future evolution? Stephen Jay Gould in the 70s is a famous evolutionary biologist, asked the question, if we roll back the tape of evolution, would we see birds and dinosaurs and bipedal mammals? And that sparked a debate. People were really, there were two camps. Some people say, oh yeah, there is a lot of convergence in evolution. Look at whales and fish, they look the same, they come from different lineages. And many other people said, no, it's completely contingent to random events. The dinosaurs got extinct, they would not have gone extinct if the meteorite didn't hit the planet, blah, blah, blah. Now, I will try to argue that in cancer, there is such a level of convergence of evolutionary forces, and in the context of drug is even more clear, that there is hope for predictability because of this convergence of forces. And on top of it, predicting evolution in cancer is clinically relevant, so it's really useful for us to predict the future course of the disease. All right, so we spent now I look back at all these papers and I feel like really old, and like 10, 15 years developing computational methods to analyze cancer genomes, okay? And we focused a lot on cancer genomes. We work a lot also with Giulio that you just heard, because, first of all, because there is a lot of evolutionary theory, so mathematics in the context of molecular evolution, which is a big field with a lot of solutions, and these solutions could be adapted and changed to work in cancer. And of course, in evolutionary biology, when you think about molecular evolution, it's all about genetics, right? That's most of the traits we pass on to the next generation. So most of the methods, if not all, they describe genomes and not epigenomes or anything else that we observe in cancer. So we focused on developing methods to identify forces of evolution, what we call neutral evolution, which I don't have time to investigate today, to tell you today. Positive selection, that's Darwinian evolution, classical Darwinian evolution. So that means, the clones that have a selective advantage of a heritable alteration, they grow and they come to dominate the cellular compartment. Negative selection, for example, by the immune system, so some clones die off or are removed by some force. And then we do subclonal reconstruction, and we're also interested in predicting whether evolutionary measurements and evolutionary forces, they make forecast of the disease. Do they predict survival or progression? They should, right? If evolutionary biology is the theory that determines cancer biology, they should, and in fact they do. And so we have made quite a lot of papers about it. But in the course of the years, we received a lot of criticism from molecular biologists and cell biologists. They all told us, yeah, but you are looking at only genomes, and the cancer is not only genetics. And what about epigenetics and transcriptional profiles that change? What is the impact of plasticity and cell transcriptional profiles? So transcriptional programs. Now, plasticity is a very hot topic. Nobody knows exactly what it is. Nobody agrees on what the definition is. I'm going to give you my definition. So for me, something that is plastic, that means it can change without Darwinian evolution, without a heritable mechanism. So if it is epigenetic, that mechanism, and that mechanism that drives, I don't know, resistance, is an epigenetic mark which is heritable, that is not plasticity. To me, plasticity has to be something that is non-Darwinian, because I can deal with anything that is Darwinian even if it's not genetic. Okay, the theory handles everything. Remember that Darwin, which formulated the theory of evolution, knew nothing about heritability. Everything he said about heritability was wrong. He knew nothing about how the traits are passed on. He had no idea. Mendel, of course, a contemporary of Darwin, clarified all those, crazy thoughts he had. All right, and then, again, as I said before, do evolutionary measurements predict progression? So all these things that we measure, are they useful in the clinic? I don't have an answer to that, so don't ask me now. But what we did back then was, okay, we did all these analysis of cancer genomes and developing methodologies to analyze those. Can we can we design an experiment that will inform models that contains multiple layers of information of cells? Some of them may be heritable, some of them may not. Okay, that will inform better models. That was the idea. So basically, this was the experimental design. We took colorectal cancer, which is our favorite cancer of choice. Colorectal cancers are the archetypal solid tumor studied for evolution because they are easy to sample. You can cut during surgery a whole piece of a colon. There is a cancer in the middle. You can sample spatially the tumor in the same way we sample existing species or mitochondrial DNA in humans across the globe to track their evolution and their migration. We do the same in cancer. And the advantage also of colorectal cancer is that you can spatially sample them accurately and you can isolate back then, and it is now, but it was easy to isolate single crypts or glands. Doing single cell multi-omic sequencing back in when we started 2015 was difficult to have, even today, having reading whole genomes of cancer cells and transcriptomes and epigenetic profiles like chromatin on the same cell. It's not possible today if you want to generate a decent data set. Back then, so we had to do this single crypt or single gland isolation. It's really painstaking, but because they are tiny clones, single cell in origin, tiny crypts, you could pluck them out and do multi-omics. So we did like whole genome, ACC, and RNA sequencing. And to cut the story short, what we found was, and this I'm really simplifying, lots of things. One thing you find is positive selection for mutations in chromatin modifier genes. So genes that have something to do with epigenetics have genetic mutations in them. Okay, fine. This was described also before. The other thing you find is recurrent epigenetic alterations at the level of chromatin accessibility. So promoters and enhancers in cancer cells had different accessibility than the normal counterpart. And it's very nice in colorectal cancer, you can sample the normal very well. In other cancer types, you can't. Okay? And you find a lot of changes between the cancer and the normal, and between the adenoma and the cancer, and the adenoma and the normal. But particularly, you find changes in cancer driver genes. So the first thing you do is you get a long list of cancer driver genes described by genetic studies, and you find that even in the absence of a mutation, these cancer driver genes have changes in their promoter or enhancers. And in fact, for example, the most common loss of promoter in a cancer driver gene was CDC6, which is a known tumor suppressor gene in colorectal cancer. So this tells you that epigenetic alterations can act exactly like genetic alterations. We know they are heritable because we sample really far away location in the genome and they have exactly the same profile. So the parsimonious explanation is that they have a common origin. So if things are very similar, very far away, consistently, usually they come from the same place. So they have similar heritage. That is, similarity by lineage, which is at the very core of evolutionary biology. Okay, adenomas and carcinomas. But now to move to the stuff we are doing now, okay, the question was, are all these epigenetic alterations really functional? Are they a cause or a consequence? how stable they are. Can we measure them at single cell resolution? We couldn't do a great job at single cell resolution with crypts. Now we can, okay, and several other questions. All right, so we set up a whole new study in collaboration with the National Cancer Institute, Istituto Nazionale Tumori, with Filippo Pietrantonio. We take samples routinely from colorectal cancer resections and liver metastasis resections, and we do both whole genome sequencing, but also single-cell multiome. So we extract single cells, and we look at RNA and chromatin from the same cell, which is like a common assay that everybody can do. You just like, it's a plug and play almost, if you prepare your sample correctly. And we also derive organoids. Organoids are very important because every time you design a model that makes any prediction on human data, it's very hard to validate the model in some model system. And now we can, if you trust organoids, they are not the best thing that you can have, but they work quite well. Patient-derived model systems, you can tweak them, right? So you can manipulate them. And I'll come back later with that. All right, so the first thing you do is you check whether what are the epigenetic alterations in the cancer that are different from the adjacent normal, or in this case, well, the distant normal, so the normal on the same patient. Okay, so you sample normal cells that are far away from the cancer, and you look at their epigenetic profile. And what you find is that if you look at cancer driver genes, you find quite a remarkable level of recurrent alterations. On the left-hand side, you see promoters, and on the right-hand side, you see enhancers, okay? The shade is the level of clonality, how many cells in a certain sample have that alteration. And what you find is that, you say in the columns, you have primary colorectal cancer and metastasis. You find that, for example, there is a high recurrence of certain gain in accessibility in promoters in known cancer driver genes, or loss of promoters in some tumor suppressor genes, and the same for enhancers. So you find a long list, which is even more recurrent than genetic mutations, of changes in accessibility, which is something we also found in a similar way in EPIC before. And then you also find quite a lot of other recurrent alterations, which are not cancer driver genes. They are something else, we don't know. They are very recurrent. The question is, they are so recurrent, they look like, I don't know, to me they look too recurrent, okay? There are so many changes that are recurrent. So is it just the tumor microenvironment that changes the epigenome of these cells? And basically there is a cascade factor in which a lot of genes are involved. So if you read a lot of these genes, I don't know if I have no idea what they do. So many of them are really difficult to make sense of. And I'll come back to that. So of course you can do all sorts of correlation between the data. So you find that quite a lot of the epigenetic changes are accompanied by change in expression because you have chromatin accessibility and RNA-seq from the same cell. So you can check whether if you take a tumor suppressor gene in which there is loss of accessibility to the promoter. So the promoter is normally the normal of the tumor suppressor gene is open, so the tumor suppressor gene is transcribed. In the cancer is closed, there is no accessibility to the promoter, and therefore, and in fact, you see the expression of the tumor suppressor gene going away, okay, that you observed, and the other way around. Okay, you can also look at transcription factor activity. So instead of going per locus and say like cancer drive, like you want to do cancer genetics where you look at, okay, there is a gene and I look at the promoter or I look at mutation in the gene body, you can measure transcription factor activity. So you look at accessibility of transcription factor binding sites of a transcription factor and ask the question, does this transcription factor, for example, something related to Wnt signaling, is it more active in the cancer or less active in the cancer? You find a lot of interesting things that have to do with immune response and Wnt signaling. Some of them are really obvious. Okay, you find a lot of developmental transcription factors involved in developmental genes being reactivated, homeobox genes, that's has also been described before. So there is also an epigenetic rewiring of the cancer epigenome. All right, then the question is, because I'm conscious I have, what, 20 minutes? So how many minutes did I burn so far? 13, okay, very good, very good. All right, so the question is, epigenetic alterations, are they stable? So when you look at when you do spatial sampling of a tumor, if things are the same and are very far away apart, the parsimonious assumption I told you before is they come from the same, there is memory of that. So they are similar by descent, okay? So for example, I don't know, primates are similar because they have the same descent, even though they are far away. chimps and orangutans. They live in different continents. Okay, so the same here. But here we also had organoids. So organoids, of course, they are synthetic. They are manipulated model systems. So when you put them in matrigel, they don't retain. They retain a lot of the genome, the chromosomal composition, and the mutations remain quite a lot of the same, depending on how long you culture them. And then we look at their epigenome, and you see here on the top. So on the bottom, you have the actual. Maybe I should point at something. Oh, that's tiny. Okay, so this is two cancer cases. At the bottom, you have a tissue. Okay, when you look at recurrent epigenetic alterations in cancer driver genes, this is at the level of enhancers, for example. And then at top, you have the organoids. So the ones you had, you keep them, and of course you add. There is certainly additional epigenetic dysregulation in an organoid. You cannot expect an organoid to look identical to the original tissue, but more, the take-home point is that a lot of what's present in the original cancer is retained over even cultures, okay, not just between samples. All right, and then, and I get to the very end of it, the question really was, what's the chicken and the egg? Is it that all these epigenetic changes in the cancer are a consequence of the environment where the cancers are, or are they really, like heritable events, like a mutation that happens in the cancer and is selected? Okay, so the nice thing in colorectal cancer is you can sample a lot of the gut. So you can sample the normal distant colon that you see up here, and you can sample the adjacent cells of the normal colon, okay, that you sample with the cancer. So basically, this is a bit of a human experiment in which you grew normal cells inside or next to the cancer microenvironment, okay, versus cells of the normal gut that are far away. And you compare these two. So you basically ask the question, what does the epigenome, so the genome of the normal epithelial cells next to the cancer is normal, okay? They don't have APC mutations, they don't have driver genes, very rarely they have. But what about their epigenome? And it turns out that even though they're transcriptionally, they look similar but not identical to the adjacent cell. This is a typical single-cell RNA-seq UMAP plot. So in yellow, you have all the cancer cells. Cancer cells are always different in a UMAP plot because you have copy number changes. And then you have adjacent in green, adjacent cells to the cancer, normal adjacent cells, and in blue you have distant normal epithelial cells. They are relatively similar, but they are not exactly the same. And in fact, when you look at their chromatin profile at promoter and enhancer regions, they have some gained promoters. For example, in FOXQ1 and other known, well, some of these genes are cancer-related. They also have the same type of epigenetic alteration the cancer has. So the cancer has, if you look at cancer versus distant normal, you find FOXQ1 promoter gain. Okay, what does that do? Well, it turns out that even the normal cells next to the cancer have it. Okay, so there are quite a handful, not an enormous amount, but quite a few of epigenetic changes that are evidently not somatic evolution. They are not a heritable new event that happens in the cancer, because if it was, they would have had to happen at the same time, independently in the cancer and the normal cells next to them. So the only explanation here, the parsimonious explanation, is that there is a convergence of these two events. So evidently the microenvironment or the place where they are rewires the epigenome. Same for other highly recurrent alterations. All right, so in summary, okay, epigenome cancer evolution is really evident, okay? Somatic changes are recurrent and are stable, and now we are in the process of validating this. The question is, okay, there are a lot of epigenetic changes in cancer driver genes. I mean, is the cancer dependent on these drivers or not? Okay, and of course, this is about CRISPR-I screening, when you actually change epigenetically some accessibility or promoter enhancers, and you see whether, in the case of organoids, of course, we cannot do it in humans, you check whether there is a dependency of this cancer into these epigenetic states, and that's what we're doing now. Of course, they occur in the absence of a mutation, so sometimes they happen like a driver. Okay, this I said before, so there is a lot of, there are focal alterations, but also global alteration at the level of transcription factor binding sites, so there's certainly some level of rewiring at the global epigenome level, which is not very clear. And the funny thing is that there is epigenome rewiring, so some heritable epigenome rewiring in normal cells adjacent to the cancer. And so we need to be paying attention that that's certainly not a cancer driver gene. So if you target those, I'm not sure it's a good target for a drug, for example, if it's just the environment of the cancer inducing epigenetic rewiring. All right, I'll stop there, and I'm happy to take any questions.
Thank you, Andrea, for this outstanding talk. Are there any questions from the audience? If not, oh yeah, there's one there.
Thank you, Andrea. Fantastic talk. I was wondering whether you could speculate whether some of those changes are in kind of inflammatory memory, and if you, when sampling regions distant from the tumor, you saw differences, or I don't know if you or someone else, in terms of inflammation, so that whether they have differently exposed to inflammation. And that's why at some point even the normal next to the tumor has these changes.
Yeah, that would be an obvious one, yeah. So the two obvious candidates, one is inflammation, but we'll look into it a bit better. The other one is Wnt signaling. So when we were picking crypts back in 2016, we picked a lot of crypts that looked like cancer. They were huge, okay, and we thought they were beautiful crypts. We sequenced their genome, was normal. And of course, you likely have Wnt signaling that upregulates stem cell population, so the crypts look crazy, but they are normal. And probably is also that one a component, because it's just diffusible factors, right?
Okay, thank you. Any further questions? Yeah, there's one over there.
Nice talk, Andrea. Just curiosity, can you estimate which proportion of these stable epigenetic changes that you detect in the tumor and its microenvironment depend on DNA methylation changes, which are also, DNA methylation is a well-known established way to lead to stable epigenetic changes. But I didn't get from your talk whether you used a technique which assessed DNA methylation or not.
No, we didn't. So we can do single-cell methylation. The problem is you have to do single-cell on plates, and it works, but it's quite laborious. But yeah, that's a great thing. Then you would have a proper molecular proof that that there is a stable marker in the genome. So it's something we could even do at the bulk level. I don't know if it's really necessary to do single-cell methylation, but it's an excellent point. Yes.
Okay, thank you. If not, I have a quick one. If I spotted correctly, among the genes that surprisingly had a low promoter was ERBB2, which is instead a well-established oncogene in colorectal cancer and in GI cancers, malignancies in general. Would you comment on this? I find that rather intriguing.
Yeah, I have no idea if it's noise, and basically, I don't know. None of these have HER2 amplifications. So I don't know. I think in the context where there is no HER2 amplification, I think it doesn't matter, or I guess I don't have a good
Could it be noise, as you mentioned, like a false positive coming out of the background?
For sure, a lot of this, more than false positives, in the sense that there is strong evidence in the data that they are there. But a lot of these, they are like a list of cancer driver genes. Very often, the cancer driver genes, they do nothing in the specific context they are in. Yeah. Right, so there are cancer driver genes you see, and half of the time they are not real drivers. So I bet that a lot of these long lists are real. That's why we are doing the part with CRISPR-I,
the functional validation. Yeah, we're really looking forward to see some functional data associated with this, some of these changes that you found, because they will really point us towards the right direction, I guess. Thanks.
Thank you. We move to the next speaker, that is Vincenzo Corbo, that will talk about the role of extra chromosomal DNA in pancreatic cancer. Thank you.
Grazie. Okay. Vediamo se... Okay. So I have nothing to disclose. I don't know if this is working. Now I know why you put me here. It's not moving. Yeah. So you want me to, so that we can put up a fight with Andrea, since he has just said that he doesn't believe that plasticity is linked to something which is heritable, and actually I put this word plasticity.
linked to something that is actually editable, which is extra-chromosomal DNA, although this is not following Mendelian rules, so it's a little bit different. I do agree with Andrea that plasticity also for me is something environmentally imposed. And that's what I'm trying to show you today, so nothing to disclose. So just a brief introduction on the disease that we are focusing on, almost obsessed about, which is pancreatic cancer. As you probably all know, this is a devastating disease, has a very peculiar biology, and I think that what captures really well the uniqueness of this disease is actually just a simple histology, because as you can see, if you look and compare the normal pancreas towards the malignant, what you see is very few neoplastic glands immersed in a desmoplastic reaction, which you can, for example, identify by staining with alpha-SMA. So PDAC is no different than any other solid tumor. It's a complex heterocellular system, but has a significant expansion of the stromal compartment, which creates very hostile environment with reduced oxygen tension, nutrient availability, elevated fibrosis and mechanical stress. And to further complicate the situation, we know that within the same tumor mass, we have this partially confined subtumor microenvironment, which creates a great intratumor heterogeneity. So what we really think in our lab is that the reason why these cells are so aggressive is actually because they have been shaped by this very hostile environment. So our interest in understanding how these microenvironmental stressors will shape the evolutionary trajectory of PDAC cells. And that's why we turn our attention to extrachromosomal DNA at some point, because this ecDNA have been around since the 60s, more or less, but now the, let's say, attention to this alteration is revived thanks mostly to new computational methods to be able to detect their presence from whole genome sequencing data. And what we think is quite interesting is that these large megabase chromatinic circles, they can contain oncogenes, regulatory elements such as enhancers and promoters, but what's really important that they lack centromere. So during cell division, as far as we know, they replicate once per cell cycle, but they do not align to the mitotic spindle. Therefore, during mitosis, they are unevenly distributed to the other cells. And as you can imagine, this will create an extreme variability within the same tumor, and which is what we probably agree in a way with Andrea, is also that the environment can rapidly change the outlook of the genome in few cell divisions. And so this really non-Mendelian pattern of inheritance, which makes them very interesting to us. And so our question was how this phenomenon contributes to pancreatic cancer progression. And so if you think about, there are two problems here. One is related to our ability to detect extrachromosomal DNA, because as I mentioned before, the neoplastic cellularity in an average pancreatic cancer is very low. And if you use bulk whole genome sequencing, single cell is not even possible at the moment, then you know that the signal is very diluted and you may not pick up extrachromosomal DNA amplification because they're heterogeneous, even if there is high level of amplification. And the other problem is that if you really want to understand whether this alteration can contribute to the development and aggressiveness of this tumor, we have to model the dynamics of extrachromosomal DNA. And something else that we agree with Andrea, we use organoids, which I think it's at least better than 2D as a model. And so we took advantage of models that we have generated within the Human Cancer Model Initiative, which was launched in 2016 by the National Cancer Institute, Wellcome Hub, and Cancer Research UK, and we sequenced them. We use whole genome sequencing, short read sequencing, and then we use a computational tool, which is called Amplicon Architect, that will reconstruct the structure of the amplicon and classify these amplicons into linear, breakage, fusion bridge, complex, or circular. And when we did that, what we actually saw is that at least the prediction of ecDNA positive tumors is very high, higher than expected, which is in this pie chart. I don't know if it is working actually, probably not. This pie chart here on the left, if you compare this piece of data with the one that we obtained from tissue. Again, for the problem of cellularity, if you go to tissue, then the frequency of ecDNA goes down. And then we look at the differences between ecDNA positive and ecDNA negative tumors. The ecDNA positive tumors, they show biallelic inactivation of p53, almost invariably. They are, of course, enriched for
oncogene amplification as expected. There is no statistical association between ecDNA and chromatopsis. At first this was surprising to us because chromatopsis is a mechanism supposed to lead to the formation of extrachromosomal DNA. There was a statistically significant association with whole genome duplication events. And also both at genomic and transcriptomic level, this tumor were characterized by replication stress. And so we found this ecDNA to be quite prevalent, but what Amplicon Architect does is a prediction of circularity. So you have to validate the presence of extrachromosomal DNA. The first thing that we did, we focused on MYC because this was the most recurrent event in our cohort, and now that we have expanded this to other cohorts, actually we can confirm that MYC amplification of ecDNA in PDAC are quite prevalent. And we could validate the presence of ecDNA, or at least infer the presence of ecDNA through whole genome sequencing in the parental tissue. As you can see from these circles, they are relatively simple. They contain contiguous regions from chromosome 8, where MYC is located, and also validated their circularity by circle-seq, which is a very tedious procedure where you basically get rid of the majority of the linear DNA, and then you are kind of left, or there is an enrichment of circular species that you sequence, and this is here. And then we also were capable of validating the interconnected breakpoints of the circles by a simple capillary sequencing, which was very useful for us because we then decided droplet qPCR probe that we could use to distinguish copy number of MYC from the chromosome and MYC on extrachromosomal DNA. Finally, of course, since we have organoids, we can prepare metaphases and use old-fashioned cytogenetics and can prove indeed that you can see there are a number of spots outside of the chromosomes, and then the cultures are quite heterogeneous, right? So these two cultures that we named VR01 and 06, as you can see, not all of them contain ecDNA. When they do, and this is interface fish, these spots tend to cluster in confined regions of the nucleus, and this is due to a phenomenon that has been described by the Paul Michel lab, which is clustering. Basically, these circles interact with each other. And more importantly, as you can see here from this distribution of MYC spots per cell, the distribution of copy number within the same culture is extremely heterogeneous. You have the majority of cells with low number of ecDNA per cell and very few cells with extremely high level of extrachromosomal DNA. So this extreme intratumor heterogeneity related to the copy number of MYC actually translates into extremely wide distribution of MYC dosages in the same culture. So if you compare ecDNA positive tumor, the one on the left, versus organoids derived from tumors containing linear amplification of MYC, what you notice is that on average the expression of MYC is higher, which is kind of expected. But then when you normalize by copy number, then the situation is very different. So you have one organoid, VR06, where the expression of MYC is indeed supercharged, and the other one is actually very low. And this was a new finding because the dogma in the field was the expression of an oncogene from the extrachromosomal DNA is always supercharged because the chromatin is relaxed, because they form these clusters. So in addition to the cis interaction, they establish trans interaction between circles that further push the expression of these oncogenes. But what we found is that it really depends on the regulatory landscape of these circles. Indeed, we went back to the circles, and we realized that in VR06, the one showing supercharged expression of MYC, indeed there is the lack of PVT1 promoter, which is a known element that insists to restrain MYC expression. And if you look at the expression of PVT1 and MYC in VR06 and 01, this completely makes sense. Now we have further proof that this is happening in additional model. So we can see cDNA. We found that the expression of the oncogene does not scale linearly with the copy number, but it's actually depending on the regulatory landscape on the circles. The question for us was whether these could actually afford cancer cells an adaptive mechanism to environmental stress. So what we decided to do, we took advantage of previous publication from Toshiro Sato's lab showing that not all PDAC cells can withstand a wind-deficient environment. So we removed basically wind ligands from the organoid culture medium. And I'm not showing in the interest of time, but we show that MYC is a bypass mechanism to the requirement of wind into the culture medium. So as expected for increased fitness for cells having a lot of MYC, what you see just 3 days after removal of this wind ligand from the medium, if you perform an immunofish, then you see that the MYC high cells are enriched in the proliferative fraction.
But something that I wanted to point out is that even like in plus Wnt are responding, which means in the absence of a selecting pressure, you already see that the cells with extremely high level of MYC are actually in the non-proliferative fraction, which was an indication that there is some sort of fitness cost imposed by elevated burden of ecDNA, and we will come back to that later. So consistent with this positive selection of ecDNA, if we keep growing these organoids without never passaging them in the absence of Wnt ligands in the medium, what you see is that the fraction of ecDNA positive cells go to 100%, so all of them now contains ecDNA, that the copy number of MYC on average is way higher, and the distribution of copy number states shift towards the right, which means that these cells rearrange themselves around the new optimum of copy number, because of course now they have superior fitness. But even they do that, the distribution of copy number state is anyway extremely wide. But again, as I told you before, this actually comes with a cost. Indeed, if you look at cells, they have rearranged around a new optimum of copy number, which are these WRI. I don't know if you actually can see anything from these slides, but you have to believe me on that, where cells where there is a huge level of MYC, of course, on extrachromosomal DNA, there is this replication stress that you can measure in different ways. So there is engagement phospho-CHEK1, HERPA2, FTD-BP1 gamma-H2AX. And more importantly, this is ecDNA specific, because if you increase MYC expression exogenously through a lentiviral vector, first of all, these organoids lose completely extrachromosomal DNA because they don't need it anymore. And of course, you don't see the same engagement of the replication stress. Actually, the only marker that goes up is phospho-RPA2. And it's also oncogene agnostic, because we took advantage of models from Andrea Ventura. He used the Krilox system to generate this endogenous extrachromosomal DNA. And if you look, these are colorectal cancer cells where we can, by using antibiotics, elevate the presence of ECMDM2, and you see same relationship. ecDNA goes up, this genotoxic stress goes up. And so if this is true, what you expect is, of course, if you neutralize the selection pressure, there is negative selection. And that's actually what's happening. So the first thing that we did after just 3 days of neutralization of the selection pressure, this is what you get. Now the proliferative fraction of these cells is enriched for MYC low copy number. According to this, the fraction of ecDNA positive cells goes down, MYC copy number goes down, and the distribution of copy number states goes towards the left now. So again, they're rearranged around the new optimum of copy number. Why we think it's very important? Because of this concept of ecDNA fitness landscape. So the genotype to phenotype mapping in ecDNA-driven tumor is very difficult. It's very difficult because the ecDNA doesn't mean that you have the same amount of MYC, that you have the same amount of extrachromosomal DNA. And this is kind of captured here. This is individual organoids from ecDNA positive versus negative, where you see the extremely wide distribution just of MYC copy number. And of course, since this relationship between ecDNA and fitness is not linear, not fixed, we call this as a fitness landscape. But the point is why this is actually important from a, let's say, clinical perspective. So we kind of started to test this concept using different type of drugs. I'm just showing you a pan-RAS inhibitor, the one from Revolution Medicine, because we said to ourselves, okay, what do you expect to see? Right? MYC is a known mechanism of resistance to RAS. So we expect to see elevation of extrachromosomal DNA and MYC level. And so the first example, BR06, the organoid with a supercharged expression of MYC, this is actually true. So if you use a pan-RAS inhibitor, you see a shift towards the right of the distribution of copy number, all the cells are ecDNA positive, and the copy number goes up. But if you actually look at the other organoid, the one that had to elevate to 200-300 of copy to get to the same level of MYC. Actually, this is the other way around. It's actually, as you can see here on the far right, so basically it's dropping the percentage of ecDNA positive cells. The distribution of copy number states goes to the left, and the average copy number of MYC specific for the extrachromosomal DNA goes down. So that's why it's very important to, characterize these circles really well. So just to conclude, what we found is that the ecDNA are quite prevalent in pancreatic cancer, around 20% with putative, because we need to validate all of them. Amplification on ecDNA. MYC is clearly the most important one. What we also found is that this transcriptional output does not scale linearly with copy number, so it really depends on these complex rearrangements on the circle. I have no time to show that this circle can further evolve over time, which is kind of scary.
We also found that the relationship between ECDNA and fitness is not linear, not fixed. It really depends on the context. So if you are in a context, they have a different fitness. And of course, something that I did not present in this of time, that MIC expression provides a mechanism for rapid environmental adaptation and drives also morphological changes. So I'd like to close this by, of course, acknowledging the work of the people in the lab, all of our collaborators, including Andrea and Carmine here, although I have no time to show their contribution to our job. And thank you, of course, for the attention.
Thank you.
Okay, sorry.
Thank you for your beautiful talk. There are questions.
Hi. Very nice presentation. I just had a question about your extrachromosomal DNA with having MIC inside. So since you showed that it has like an impact also in the level of MIC that is expressed by the tumor, do you know if the amplification, if the piece of DNA that gets into the extrachromosomal DNA also contains regulatory elements like the NME or other super enhancers that are normally at the same locus?
Yeah. So, yeah, they do contain regulatory elements, and we also, I have no time to show, we also see that the two different extrachromosomal DNA behave differently in terms of clustering. So again, the one with the supercharged expression of MIC show clustering, and when you treat with JQ1, you can dissolve the cluster, which is, reported or at least suggested, and the drop in MIC expression is very high, so you reduce the expression of MIC. The other one, we do see clustering, but I think it's more due to the number of copies of ECDNA rather than actual interaction between the circle, because the clustering is BRD4 mediated, and so since there is this conflict between PVT1 and MIC, what we believe, although we don't have the actual data, it's just a speculation, there is no actual clustering in the other one. Indeed, when we treat with JQ1, we don't see the same drop in MIC expression. So it's really heterogeneous. We now have another organoid, which is a metastatic one, which is actually similar to VR01. So it's a huge number of ECDNA per cell. But if you normalize by copy number, expression of MIC is low. And indeed, we look at PVT1 and MIC locus, and it's preserved. So it kind of makes sense to us, at least, that this is the model that we have in pancreatic cancer, because others have shown different things.
Thank you, Vincenzo, for the great talk. Now, in light of everything you said and the fast-moving field of ECDNA biology, are they a good target, a drug target?
Not the way they're doing right now, because what they're doing, they're targeting the ECDNA high, right? Because they are exploiting this genotoxic stress. And I think it's pointless, because at the end of the day, because what we also see is that if they don't need ECDNA, right, they just lose it.
Or if it's highly plastic, then it tells you something
there
that the dependency is not
So
there.
what we would like to do, and I think we are starting to do this in collaboration with Francesco at some point, Yori was here somewhere, we would like to map the fitness of the ECDNA low, medium, and high of the different, I don't know if we can do that as a continuum, because we're also doing single nucleus multiome, because we would like to create an atlas of ECDNA fitness landscape, matching copy number with expression in the different environment, to learn, the different vulnerabilities, right? But I think the way they're doing it now, I think it's useless, because if you target, it's like targeting one population knowing that already they're going to shift and you're going to reach for the ECDNA low, which show literally no genotoxic stress, because if you have few copies of ECDNA, you have nothing. So yeah, I don't think it's the way to go.
Thank you, Vincenzo. I have one quick question. How do the fitness of your MIC extrachromosomal amplified clones correlate with the dose of KRAS inhibitors that you've given?
That's a good question. So the again, depends on the dosage. So if the dosage of MIC is very high, the cells tend to be insensitive in a way, so they kind of adapt very easily to the treatment. When we see this change in fitness in when they have to elevate too much the level of extrachromosomal DNA. So that's why I think it's not like, okay, there's an extrachromosomal DNA amplification. If you don't know the MIC transcriptional output and all the rest, I don't think it's a good idea to go after that, right? So you need to know when to treat and with what and who to treat. I think it's going to be difficult based on the different type of extrachromosomal DNA. So there is no one fits all, even if it's ecDNA.
No, do you actually, the reason why I was asking is whether, of course, this is the dynamic process. And indeed, if you're thinking about the response to KRAS inhibitors could be transient, you could already start thinking about re-challenge strategies after an intermittent line.
Yeah, that's
And the knowledge of extrachromosomal DNA in that instance will be a good biomarker to select patients to re-challenge or not to re-challenge with a KRAS inhibitor.
But you need to know, how these ecDNA are behaving, because otherwise...
Exactly. Yep.
I think we have to move on. Thank you very much.
Okay, so we are now moving on to the selected oral communications. Now, abstract presenters were asked to prepare their slides in English and their talk in Italian. It's really up to them how they wish to present. I invite all of them to present in English if they feel confident in that. And this is for the sake of our English-speaking guests. But of course, if you prepared your talk in Italian, then okay, that's fine. Go ahead. Welcome. The first speaker is actually Giulia Cosentino, who is going to deliver her abstract on miR31-3P inhibition as a strategy to increase response to trastuzumab in HER2-positive breast cancer.
Okay, thank you. I'll go in English, but be patient with me. Okay, so first of all, I would like to thank you for yes, for the opportunity to present my data. So my data is I will present the data concerning miR31-3P in the context of the response to trastuzumab in HER2-positive breast cancers. So 15 to 20% of breast cancer present the overexpression or amplification of the oncogene HER2, which activates downstream pathways of proliferation and survival. And the standard therapy for this subtype of breast cancer is trastuzumab. Trastuzumab is a humanized monoclonal antibody which binds to the extracellular domain 4 of the receptor. It blocks the downstream pathway. It induces the internalization and the degradation of the receptor, and it also triggers the anti-tumor immune response. So unfortunately, important portion of patients do not respond to this treatment. Although new anti-HER2 agents have been already tested, and also combinations treatments have been tested, it is important to predict response in order not to overtreat the patients and design alternative strategies. So to do this, we would like to propose the use of microRNAs, both as biomarkers and as therapeutic tools. So recently we have identified a signature that predicts response to neoadjuvant trastuzumab, starting from samples from the NeoAlto clinical trial. And this signature is composed of two microRNAs, only two microRNAs, that are miR31-3P and miR3823P. In particular, we then focused on miR31-3P to see whether, besides being a biomarker, it could also have a biological role in our context. Since miR31-3P is negatively associated to response to neoadjuvant trastuzumab, we decided to inhibit its expression in a HER2-positive breast cancer cell line resistant to trastuzumab. And so, I don't know if I can. Doesn't work. Okay. On the panel, on the left panel, you can see that his down modulation in a 2D model induces a reduction in cell viability and HER2 activity, but it doesn't increase response to trastuzumab. On the contrary, when we inhibited miR31-3P expression in an in vivo model, we could see that we could increase the response to trastuzumab. And a fundamental difference between the in vivo experiment and the in vitro experiment is the presence of the tumor microenvironment in the murine model. And although we performed the experiment in immunocompromised mice, the innate immune cells are still present. And so our hypothesis is that our microRNA maybe could modulate the activity of the innate immune cells. So we went back to the samples from the neoadjuvant clinical trial, and we could see that the expression of miR31-3P anti-correlated with activated immune cell subsets. And we then performed an ingenuity pathway analysis on RNA-seq data extracted from HCC 1954 cells, that the same cell line that we used in vivo, inhibited for the expression of miR31-3P. And we could see that the first pathway that was activated upon miR31-3P inhibition was interferon alpha-beta signaling, which is an immunomodulating pathway. And so we decided to validate this data, selecting 3 of the main genes involved in this pathway, so interferon alpha-1, IRF7, and FKB, which are 2 transcription factors for interferon alpha. And as you can see, both in non-treated and treated cells, we could see an increase in their levels upon miR31-3P inhibition. So encouraged by these results, we then performed a very preliminary cytotoxicity assay using cell lines of NK cells and macrophages, conditioned with the medium of miR31-3P overexpressing HER2-positive breast cancer cells. And as you can see, the activity of these immune cells is reduced upon conditioning with miR31-3P expressing medium. So our idea is that when we inhibit the expression of miR31-3P in the tumor cells, this induces a higher release of interferons in the tumor microenvironment, which helps the activation of the immune cells and increases the efficacy of the treatment. So these are very, very, very preliminary data, but our idea is that we could use our signature to stratify the patients into putative responsive and putative resistant, and propose to the putative resistant patients the combination of trastuzumab and the inhibitor of miR31-3P. So I would like to thank my supervisor, Dr. Marilena Iorio, all my colleagues of the lab, and all the collaborators and you for your attention.
Thank you, Giulia. And any questions from the audience?
I have one. And did you know how this MIR31 is secreted? If it goes in exosome, and can you follow the microRNA to see if it indeed translocates in the recipient cell?
This is something that we would like to do. We know that MIR31-3P has already function in immune cells, directly in immune cells. So we actually don't know whether MIR31-3P could start from the immune cells and then could act in the cancer cells, or the other way around. But we know that it is secreted and it can be secreted, but we actually do not know yet. But it's something that we are interested in doing. Thank you.
Okay, thank you, Giulia. I think in the interest of time, we should move on. Thank you.
We now have Valeria Gelfo that will talk about interleukin-1 in microenvironment response to EGFR neutralization. Valerio, sorry.
So good afternoon, everybody. First of all, I would like to thank the organizer for choosing my presentation, my project for this presentation, because in my project we are studying the role of interleukin-1 in the establishment of EGFR blockade in colorectal cancer patients. In the clinic, one approach is the use of cetuximab, which is a monoclonal antibody, again is EGFR, and patients are selected for the patient are all RAS wild type for the employment of this drug. As you can see from the graph, I don't know if I can point in some way. If you can see from the graph, at the beginning of the therapy, the patient responds well, but unfortunately, after 6-18 months from the beginning of the therapy, there is a failure of the therapy. From our previous data, in 156 xeno patients, we can see that the patients that respond worst to the therapy, because the xeno patients are treated with cetuximab, there is an increase in IL-1 alpha, IL-1 beta, and IL-1 8. Also, in an independent course of 1,051 patients, we can see when these patients are stratified according to the consensus molecular subtype classification, that the patients with a higher expression of IL-1 receptor are also the patients with the worst prognosis, with the worst survival. These patients also belong to the consensus molecular subtype 1, which is characterized by an immune infiltration. Stemming from this observation, first for IL-1 sequestration, we employed a recombinant decoy developed with the colleagues from the Weizmann Institute of Science, and this recombinant decoy is able to sequester both IL-1 alpha and IL-1 beta from the medium. If you from the panel A, you can see if we employ in a model of cetuximab responsive cell, if you employ both cetuximab and human trap, so we sequester IL-1 from the medium, you can see a decrease in cell proliferation. Also, the same result, we obtain the same result in a 3D environment. So we asked what happened when we we block EGFR with cetuximab in responsive cells. As you can see from the panel 8, after 24 hours from the treatment, there is a peak of production of IL-1 receptor, IL-1 alpha, and IL-1 beta. So we asked what IL-1 actually do at the EGFR axis. As you can see from the panel B, first there is an activation of EGFR when we treat with IL-1 and also the downstream pathway, but more interesting, there is a stabilization of EGFR itself. So we asked, so we used, there are the videos, why they don't start, I don't know. There is a way to start the videos? No? Okay. So, but yes, trust me, we use this approach. We basically we marked the cetuximab with a red fluorescent molecule that is able to activate the signal when the pH is below 5. So this is a molecule sensible to pH. The cetuximab. So when we treat the cell with this molecule, the molecule, we can study actually the internalization of EGFR, and when we treat the cells, the sensible cells with IL-1, there is actually a decrease on the signal, meaning that there is a decrease in the degradation, first in the internalization and then in the degradation of the receptor. So the same here, it's maybe they start in this case, I don't know why. Okay, so in the unresponsive context, when we sequester IL-1 in unresponsive cetuximab cells, there is a rescue, let's say, of the phenotype, and there is, in this way, an increase of the degradation of EGFR. So we moved to the in vivo. In this case, we used nude mice. We developed a tumor unresponsive to cetuximab, as you can see from the panel A, and then we start the treatment. Combining cetuximab and TRAP. As you can see from the panel B, even if the effect is mild, there is a significant decrease of the tumor volume and also the survival of the mice. So we used the slice of these mice, and with a machine learning based approach, we detected the type of tissue of the tumor. So we detected the tumor, the necrosis tissue, and the immune cells, and we could see that when we sequester IL-1, there is an increase of immune cell. So we moved to the immunocompromised mice. So we basically did a syngeneic inoculation of cell in wild-type mice, and so we used the murine version of the human trap in order to sequester IL-1 from the tumor microenvironment. And what we can see, this is a very striking result. What we can see is if we use this cell that continuously produce a human trap, so we continuously sequester IL-1 from the tumor microenvironment, there is there is no tumor basically. So the tumor is completely abrogated. This, of course, is translated in the survival result, which is very prolonged. So in conclusion, IL-1 induces an increase in EGFR in the downstream pathway in cetuximab responsive cells, and decrease EGFR degradation. Also IL-1 sequestration reduce tumor volume and prolong survival in immunocompromised mice, and there is a complete abrogation of the tumor in immune-competent mice. On the right, you can see the basic model of cetuximab working when there is the acquisition of resistance. In the end, I would like to thank my teammate, and also thank you for the attention.
Thank you. Any questions?
I don't know if, okay, clinical trial are still ongoing, so we will see the effect of the blockade, yes. But the point is that in clinical we still use very easily the combination, because the drugs are already approved. So cetuximab and anti-IL-1 drugs are already approved. So this is something that it is easy to do in the clinic.
Thank you, Valerio. Do you have evidence that high levels of interleukin-1 associate with poor response to cetuximab in patients in public data sets?
Actually, we didn't so far do this evaluation.
They are available, so you...
Yes, this is a very good suggestion, and this is something that we can do, actually, yes.
I think in the interest of time, thank you, Valerio, for your presentation, and we move on. Thank you. It's now my pleasure to invite the next speaker, Giulia Schiavoni, to present her abstract on dual HER2 blockade triggers ILA-H STAT signaling to drive adaptive HER2 loss and therapeutic resistance in breast cancer. Thank you.
So, first of all, thank you for giving me the opportunity to present this data here today about a resistance adaptive mechanism that involve interleukin-8 and STAT signaling in breast cancer HER2 positive. So first of all, just a brief introduction. You know, HER2 is one of the most important biomarkers here in this context, and is also one of the targets for who has been developed specific molecules. For example, the monoclonal antibodies against HER2 are now used both on an early onset as a combination in neoadjuvant therapy, but also in advanced setting as first-line therapy and treatment. So for this reason, HER2 remains so much important to study, and also it's really important to understand the mechanism involved in its evolution during cancer progression. So previously in the lab, we identified that first-line treated patients with the double HER2 blockade with trastuzumab and pertuzumab has worse prognosis in response to the second-line treatment with T-DM1, that is an ADC, so an antibody-drug conjugates against HER2, compared to those patients that received trastuzumab only. And this is due, as we confirmed from immunohistochemical samples, for a reduction of HER2 expressions that we also confirmed through preclinical models of resistance in which HER2 delocalized from the membrane. So since also previously we demonstrated that ACC1 is one of the most deregulated RTK protein in double resistant models, we wondered whether ACC1 is also involved in HER2 regulation in this context. And so for the reason, we investigated the transcriptomic public available database, both for cell line and patient cohorts, filtered to be non-amplified for HER2. And we observed that the true transcripts positively correlate. So the more we have ACC1 transcript level high, the more is HER2. And we confirmed this data also from the protein point of view in a small cohort present in our institute from immunohistochemical analysis. So at this point, we wondered how ACC1 regulates HER2, and we overexpressed it in HER2 non-amplified cell models, and vice versa. We silenced it in HER2 amplified models. And as you can see, when we overexpress ACC1, we also observed an increment in HER2 expression, both at the protein and transcript levels, and on the contrary, silencing it, we have the opposite results. And so we'd assume that ACC1 should regulate HER2 from a transcriptional point of view. And to confirm this data, we just exploit luciferase assays by exploring the transcriptional activity of both the canonical promoter of HER2, and as you can see from here, ACC1 overexpressed in cell lines has an increased expression in the two canonical promoter HER2 by transcriptional activity. And since ACC1 is known to interact with STATs, transcription factors, we also investigate their role in this regulation. So we overexpress STAT1 and STAT3 in our cell models, and we identify that they have a repressor activity on the HER2 promoters. And currently, with this, silencing ACC1 in the HER2 amplified cell models induce an hyperactivation of the STAT pathway by their phosphorylation. So we thought that having a little bit more attention on HER2 locus should be a good idea. So we investigate the ENCODE database, and we identified some transcription motifs associated to STAT transcription factor, both on the canonical promoter of HER2, but also through the CTCF motif analysis by CHIA-PET data. We also identify two putative regulatory regions that should control HER2 locus, and in particular, the RRR1 seems to be a transcriptional regulatory hub for HER2 locus. We confirmed this data with chip analysis, and as you can see from here, those cell lines that overexpress ACC1 have less STAT3 and STAT1 localized both on the transcriptional regulatory region and both on the canonical promoters, so confirming that these two factors have repressory activity over HER2, and since upstream of STATs transcriptional factors, there are cytokines signaling involved, we performed a little screening, and we identified that interleukin-8 is down-modulating and is less secreted by ACC1 overexpressing cell line. So we can conclude from here that STAT1 and STAT3 acts as transcriptional repressor at regulatory sign to RBB locus. Then we wondered if ACC1 should control the pharmacological resistance acquired by the double blockade resistance models, and as you can see from here, the double resistant models show less ACC1 expression and also less HER2, and that we also confirm by immunohistochemical analysis both on pre-therapy and post-therapy tissue sample from patients. So trying to investigate which was the mechanism behind this kind of regulation, we performed RNA-seq on our resistant cell models, and we filtered the data set for the upregulated genes common to the two double resistant cell lines. And we noticed that in this context, we have genes involved in inflammatory response, and here there is also interleukin-8 coherently to which we observed before. And so with an ELISA assay, we observed that double resistant cell models secrete more interleukin-8 that control cell, And also, this is dependent from ACC1, since when we overexpress this protein in our resistant models, we have a reduction in interleukin-8 secretion. Also, interestingly, the silencing of interleukin-8 in double-resistant cell models also rescue the transcript levels of HER2 in our cell models compared to the control. So, giving a look on the downstream pathway, we firstly confirmed that double-resistant cell model have an increased activation of the STAT pathways, and it is dependent from ACC1, since when we silence ACC1 in the trastuzumab-resistant cell only, we can rescue the STAT hyperactivation through DEM phosphorylation, similar to what we observed in TP-resistant cell models. We confirmed this data with CHIP, since, as you can see from here, STAT3 and STAT1 are most localized on the regulatory region and the canonical promoter of RBB locus in resistant cell models. And interestingly, when we inhibit interleukin-8 signaling by repertaxin, which is an inhibitor of the canonical receptor of interleukin-8, or through its neutralizing antibody, we are able to rescue sensitivity to trastuzumab deruxtecan in double-resistant cell models. So from our cytokine screening, interleukin-6 didn't seem to be deregulated in the resistant cell models, but we knew that it has a really important role in breast cancer progression. So we decided to investigate if it could be involved in HER2 regulation, and we treated our double-resistant cell model with recombinant interleukin-6, and as you can observe from the immunofluorescence, you can see that HER2 is delocalized from the membrane. And studying the correlation, taking advantage of the transcriptomic database we used before, we observed a negative correlation between these two cytokines, transcript levels, and their BB2 genes. So our model is that there is cytokine networks in a resistant context which hyperactivate a JAK-STAT pathway that end with a transcriptional repression of RBB2 locus, giving as a conclusion less availability of HER2 in the membrane. And so we hypothesized that the inhibition of this axis should rescue HER2 expression in the membrane, so increasing the trastuzumab deruxtecan and second-line treatment ability. So we firstly identify that effectively the inhibition of this JAK-STAT pathway should rescue the membrane HER2 expression on the membrane, and then in the end, the combination of this JAK-STAT inhibitor with the trastuzumab deruxtecan increase its efficacy in interleukin-6 stimulated double-resistant cell models. So this is just the model I showed you before. The idea is that an autocrine and paracrine cytokine networks may be involved in HER2 regulations, and that inhibiting this pathway should increase the trastuzumab deruxtecan sensitivity. We are just working on assess this autocrine and paracrine mechanism just in a deep way through co-culture, and to assess also the validity of our hypothesis in a clinical study using HER2 positive early onset patients cohorts, evaluating the levels of these two cytokine from immunohistochemistry, but also liquid biopsies. So thank you for your attention, and just a special thanks for the people who contribute to this work, in particular to Dr. Giulia Bond, Rossella Loria, and Aurora Puce.
Thank you. Thank you very much. Unfortunately, we do not have time for questions, so we move to the last presentation that will be from Michela Sgarzi, that will talk about the crosstalk between MET and YAP in amoeboid-like phenotype migration.
Good afternoon to all of you, and thanks to the organizer for selecting my abstract. In this presentation, I will show you some very new preliminary data about my project that right now is focusing on the role of MET and YAP in sustaining the acquisition of an amoeboid-like phenotype in cancer cells during migration. This study actually has its basis in a previous work that we published in 2023, where we analyzed many gastrointestinal cell lines with a constitutive activation of MET, including LOVO cells, that in this presentation I will refer to as MET plus cells. In these cells, we found deep rearrangements of the actin cytoskeleton and of the nuclear architecture that were completely normalized by the silencing, the gene silencing of MET by CRISPR-Cas9. And then going on with this study, we eventually found that these aberrations were not of the entire cytoskeleton, the entire actin cytoskeleton, but were selective of the perinuclear actin fibers that sometimes are collectively indicated as perinuclear actin cap, as they form a dome-like structure above the nucleus to regulate the nuclear shape. And from a mechanistic point of view, we found, in parallel with some other research groups, an involvement of YAP in the proper alignment of the perinuclear actin. And indeed, the activity of YAP was impressively decreased in MET hyperactivated models. The reason why we were so interested in the fact that the actin cap was restored upon MET silencing in cancer cells was that usually in cancer the actin cap is completely disrupted. It's indeed a feature of only normal cells in interphase, where it prompts the acquisition of an elongated or squamous phenotype, and when it regulates the directionality of cells during persistent and classical mesenchymal-like migration. On the contrary, when in cancer cells, for example, when the actin cap is aberrant or completely absent, we have a typically rounded phenotype and a random and unpolarized cell motility that can be defined as amoeboid-like motility, as you can see also in this video taken directly on our cell models. And the amoeboid-like motility is faster, but it's characterized by completely unpolarized displacement of the cells. And so we started to think that maybe, as MET is very important, as you all know, in the metastatic dissemination, maybe the reason why MET hyperactivated cells were inducing a misalignment of the actin cap was to induce the amoeboid-like migration that recently was introduced in the classical paradigm of AMT as a final step, as the acquisition of an amoeboid phenotype compared to the classical mesenchymal phenotype is actually very important for cells migrating in complex and three-dimensional microenvironments that needs to squeeze, where they need to squeeze and to pass through constrictions. And the amoeboid state seems to be very useful in this context, for example, during intra and extravasation. Moreover, the loss of the actin cap itself prompts a nuclear expansion, a nuclear vertical expansion that decreases nuclear stiffness, and the soft nuclei are even more deformable, and this again is very important for cells that need to squeeze in complex microenvironments. So basically our idea is that MET hyperactivation prompts the actin cap loss in order to gain a phenotype where we have soft nuclei and an amoeboid-like migration that is very important for the migration through constrictions compared to the classical mesenchymal migration. To prove our idea, we started a collaboration with a reading group in this field, which is the Lammerding Laboratory at Cornell University, where we produced some microfluidic devices where we can force the cells to pass through narrow channels of 1 or 2 micrometers wide, and these channels, these sizes are limiting for most of the cells, and we can compare the confined migration to the standard unconfined migration in a controlled chamber where the channel have the channels are 15 micrometers wide. Then we can monitor in time-lapse the migration of our cells, and by using, for example, the NLS-GFP marker, we can even monitor the nuclear envelope rupture and repairs, which are transient processes that usually happens during confined migration that are non-lethal but can induce an increase in the DNA damage and in genomic instability. So basically we tested our cells in the devices after the transduction with the NLS-GFP marker, and as you can see from the video and also from the chart on the right, the MET silencing deeply affected the cell capability to pass through the constrictions, and so very few cells were still able to pass through the constriction and to squeeze through the microchannels. As I mentioned, we also investigated the involvement of YAP, so we already have some models with the YAP constitutive activation that was able as MET silencing to rescue completely the perinuclear actin cap in cancer cells. These cells in the devices, again, behaved as expected, so we have deep impairment in cell capability to pass through the constriction, both in terms of total number of passing cells, but also in terms of transit times. So the few cells that still can pass through the macrochannels were actually much slower compared to the unconfined migration. Finally, we wondered whether it was possible to induce a misalignment of the actin cap in normal cells, in this case epithelial cells, MCF10A, that usually have a perfectly aligned actin cap in the apical plane of section of cells. And by inducing TPR-MET, by inducing the expression of a constitutively active form of MET, in this case a fusion protein, TPR-MET, we were able to disrupt the actin cap together with a decrease in YAP protein stability and nuclear vertical expansion. And by testing these cells in the device, we were able to push their capability to pass through the constrictions, both in terms of cell number and in terms of decreased transit time, so the cells were faster in passing through, but impairing their nuclear stability, nuclear integrity. So the nuclear envelope ruptures were actually increased in the TPR-MET model. Finally, we tried to impair directly the actin cap in order to see if it was a mechanism that was actually actin cap dependent and it was not due to some other effects after manipulation of MET and YAP signaling pathways. And so we employed some siRNA directed against some link complex protein, SUN1 and 2. The link complex is the supramolecular structure that is supposed to connect the perinuclear actin fibers to the nuclear envelope, and so the impairment of the link complex itself is sufficient to induce the loss of the actin cap. And these cells in the devices, as you can see very well also from the video, but we also have the quantification, were much faster in migrating through the constrictions. Again, with an impairment of the nuclear integrity, so an increase in the nuclear envelope rupture events. So basically, our idea is that MET hyperactivation in cancer cells induce YAP inhibition and the loss of the actin cap in order to prompt an amoeboid-like motility that is particularly useful for cancer cells when need to migrate in complex microenvironments, so for confined migration, at the expense of nuclear integrity. But this event itself is actually prompting DNA damage accumulation that is intrinsically protumorigenic, as you all know. On the contrary, YAP activation or MET inhibition can prompt the alignment of perinuclear actin cap fibers and a typical persistent mesenchymal-like motility that is not effective in confined environments. With this, I have concluded, and I would like to thank all the Professor Loriola's laboratory where I belong, and all our many collaborators, and you for your attention.
Thank you very much for your talk. There are any questions?
So thank you, very interesting. And so I wonder whether you have investigated the relationship between MET and YAP in terms of signal transductions. This is one question. Second question, if I got it well, you induced cell migration with EGF, epidermal growth factor.
Okay.
So no experiment with HGF or?
Okay, so for the second question, we need to use EGF with MCF10A cell because they are normal cells, so they don't tend to pass through the constriction, they need a gradient. So basically, we have a first chamber where we seed the cells without EGF, and the second chamber we put EGF so that we can push and prompt the cell migration through constriction, otherwise they will not migrate. For cancer cells, we don't need to do that, so that's why we haven't used HGF, and moreover, our models are hyperactivated for the MET signaling independently of HGF due to some post-translational modifications or amplification of the gene, so HGF will not work. And in general, cancer cells in the devices behave as they tend to migrate just because the second chamber is empty, so there are no cells and there are more nutrients in the media. For the first question, this is exactly our point right now, so we need to find a way to connect actually MET and YAP, because at the moment we don't have actually an idea, so any suggestion is very welcomed. So yeah, we have a lot of sequencing data that we need to analyze that maybe will help us in this fishing expedition to find the signaling that connects MET and YAP in this context.
Thank you. I have a little curiosity. Did you check ever at microtubule network in your ameboid cells?
Yes. Actually, tubulin is not affected. There are no differences. It's something very specific for the perinuclear actin.
Okay, thank you.
Thank you very much.
Okay, thank you very much for attending this session. And sorry for being a little bit late. We're now leaving the podium for the next session on pancreatic cancer. And again, the Italian Cancer Society wish to thank Alleanza Contro il Cancro for the invitation to organize this joint symposium. Thanks.
Vai Aldo, comincia tu. Sì, cominciamo, sì, sì, sì.
Facciamo precisi tra di noi. Benissimo. Ascolta, io una roba che posso bloccare, voglio, come sempre.
Tu sei al posto di Militano.
Però quanto è? Perché noi abbiamo calcolato 20 minuti, facciamo anche di meno.
Anche un quarto d'ora perché ne abbiamo poi 3 da 10. Un quarto d'ora, l'ho detto pure a un quarto d'ora 20 minuti, poi alla fine.
No, ma io mi blocco a un certo punto. E sempre può modulare, però non sapendo che
Un quarto d'ora.
intera.
No, poi adatta il giorno prima per il giorno dopo.
Per cui dimmi.
Quarto d'ora. Sì, ti faccio segno. Vai Aldo, partiamo. 45, siamo in perfetto orario.
Sì, parti tu.
Comincia tu, poi mi dai la parola a me, così poi ti presento io, se no non ci facciamo. Vai Aldo.
Allora, benvenuti a tutti e grazie per essere qui presenti e cominciamo questa sessione sul cancro pancreatico che introduco brevemente, nel senso che passo immediatamente la parola a Carmine Pinto perché c'è stato un piccolo cambiamento di programma.
Grazie, siamo un po' stanchi, quindi cercheremo di mantenere il più possibile i tempi per dare poi anche alla sessione dopo di poter essere frequentata. Parliamo di tumore del pancreas nella città del tumore del pancreas, quindi sarà un bel momento di discussione. Io non occuperei più tempo e darei immediatamente la parola al professor Scappa, direttore della patologia dell'Università di Verona, che parlerà su pancreatica e cancer eterogenesi, deconstruction, complexity. Tutta tua, Aldo.
Appunto, come vedete, doveva parlare Gioacchino Natoli, ma non c'è, per cui mi hanno rigirato la palla e io vi posso raccontare le cose che appunto abbiamo fatto noi qui in questi tempi. Queste sono le mie disclosure.